Hello students, welcome to NSC Biology. This is Neelavati, your biology lecturer. Today I am going to start one more new chapter from class 11th, that is nothing but biomolecule. I know it is a difficult chapter for many students, but don't worry, here I am with you to help you. If you watch the video from beginning to end, trust me, you can solve any MCQ. Okay, of any competitive exam, either it is a state entrance or your NEET exam, any MCQ you can easily solve. Now, before starting the chapter, I need to share two things with you. The first thing is, many students after watching few videos on YouTube, they ask a question, ma'am, we want to watch rest of the videos of your channel, but where it is? They don't know about membership. That is fine. For such student, here I want to tell them that I have done complete syllabus of both class 11th as well as class 12th. That means overall 32 chapters are there, right? So I have done all 32 chapter videos. All are once again one shot only. That means total 32 videos you could able to get. But only thing is, if you want to watch this video, you have to become a channel member just by paying rupees 199 per month. That's it. So here I am going to give 100% guarantee that if you watch all these 32 videos, you can easily understand the concept. Once you understand the concept, easily you can understand even NCERT lines also. Because I have even read the NCERT line in the chapters. Okay. So once you thorough in the concept as well as in the NCERT textbook, easily you can solve any MCQ. Okay. So there is no doubt in getting 360 out of 360. That is one thing I wanted to share. The second thing is, okay ma'am, we do agree that we will score 360 by 360 if you watch all this video. But the question is, where is the time? How to manage time? Because NEET is not just dealing on biology. Even we need to cover physics and chemistry, right? How to manage time for all those things if you just keep watching your videos, right? Yes, dear, I can understand. I do agree. Because compared to biology, yes, physics and chemistry are difficult. So that's why here I have a small tip for you. If you follow this, it would be easy for you. Okay, what is that? Look at here. What I said, from both class 11th as well as class 12th, total there are 32 chapters, right? So, in my channel, I have done all 32 videos and all are once again one shot videos, right? Total 32 videos. So, average time, if you calculate from all this video, average time, we can get 5 to 6 hours per chapter. It means some chapters might be having 7 hours or 8 hours. For example, if you take a biomolecule only, it could be around 7 or 7.5 hours. So like that, if you take some other big chapter like a molecular basis of inheritance, principle of inheritance, so there the chapter duration will be more. But all chapters are not like that. There are certain chapters, they are very short, like ecosystem, biodiversity, cell cycle, cell division. So their total duration is around 2 hours, 3 hours or within 4 hours. So that's why I'm telling, if you aggregate together, if you take an average, we can get hardly 5 to 6 hours per chapter. Roughly we can calculate like that. Okay. Now, look at here. If you spend 1 week per chapter, if you take one chapter, you just keep reading that chapter by watching the video. And if you finish this chapter by the end of the week, then you will become perfect in that chapter, right? So what you have to do? You have to just spend only 1.5 hours per day. In that also, you can beautifully plan. So how? 1 hour you can spend for watching video as well as to read the NCERT. And half an hour you just practice MCQ, because practicing MCQ is also very, very important. So for that you won't need time. Much time definitely you don't take, because after understanding the concept, within 15 or 20 minutes you can solve MCQ, trust me. You can try also if you don't have confidence on my words. So like that, if you do for all the chapter, so within 32 weeks, because 32 chapters are there, no? So within 32 weeks, just by spending 1.5 hours per day, you can finish whole syllabus. That means in 8 months, we can finish full syllabus, correct? So still you will be having plenty of time to cover physics as well as chemistry, right? So if you follow this tip, definitely it would help for you to score 360 out of 360. Listen to me. Just by paying hardly around 1,500 to 2,000. Because per month you need to spend 199 rupees to watch all those videos I said. Correct. So if you plan it for 8 months also, or even for 1 year also, hardly it would take around 2,000, right? Need not to spend lakhs together. So if you could able to score out in biology just by paying 2,000 rupees, why can't you do that? So hope this tip would helpful for you. So now, without wasting time, let's enter into the chapter that is biomolecule. Yes. Okay, biomolecules. Here, I will tell you few things which are actually given in NCERT. NCERT has given one line. What is that? All living and non-living things has the same elements. All living and non-living things has the same element. See, it seems to be quite surprising. Where the living things, where the non-living things, how they could able to show the similarities. It is really a surprising matter, right? If I just take an example of a living thing, it can be of any source. It can be a plant source, like a vegetable, fruit or anything. So it's a plant source. Or even if I consider any animal source, say for example, animal liver or any meat piece, anything you can take as animal source. Then NCERT says, you just subject these material for chemical analysis. Like that, you can take even non-living thing also. Non-living thing could be a rock. It could be soil. It's a part of earth crust only, no? So, you just subject these things also for chemical analysis. That means you just subject it for analyzing the chemicals present inside it. Then, you are going to get a same element. Element means, here when you subject it for the process, we are going to get what? The same carbon, hydrogen, oxygen, phosphorus, nitrogen, sulphur, these things only. Then, we came to know that yes, we are the part of the nature. We are the part of the universe, because whatever the things present in the universe, whatever the things present on the earth crust, the same material present even in us also. Then what is the speciality? Why we look so unique? Why our body nature behaves so differently compared to non-living things? This is because these material are these elements. Let me name it as elements, which are so present inside the living or non-living, it will be in a different form. It means, suppose if I consider these elements only in the non-living things, non-living thing says, I have a combination of these two, these elements in inorganic form. Very simple. Just focus here. You can understand. Inorganic form means what? It could be carbon dioxide. It could be in the form of water. It could be in the form of certain ions. So these are only inorganic forms. So like that, if you just test the presence of these elements inside our body, that is in the living organism, let me write here. In living beings, it will be present both in inorganic as well as in organic form. Okay. So inorganic means even in our body also we have elements. We have certain ions like a calcium, potassium, carbon dioxide, water. Everything is there in our body also. So like that, if you just look at inside our body, we have a lot of organic material. Organic materials are nothing but our lipids, our carbohydrates, okay, the proteins, nucleic acid, these things only, organic molecule, right? So in this chapter, we are studying in detail about these organic molecules. These organic molecule itself, we call it as what? Biomolecules. Understood? So biomolecule means, so hope you have understood this much. Now, if I enter into the definition of biomolecules, I can say a simple definition that all organic carbon-containing molecule found in a cell are nothing but biomolecule. Let me explain it once again. What I said, organic molecule present inside a cell, right? Let me take a cell. Imagine this is a cell. Cell is made up of plasma membrane, correct? So, what do you think the plasma membrane made up of? Plasma membrane also made up of lipid and protein only, right? Lipid and protein. So this lipid and proteins are organic molecule. That is nothing but biomolecule. Now I will enter inside the cell. Here there is a nucleus. So nuclear membrane is also made up of what? Nuclear membrane is also made up of lipid plus protein only. And inside we are going to see the chromosome. Right? So chromosome once again made up of histone, protein, as well as the DNA. DNA is nothing but the nucleic acid. It is also biomolecule. Now if I take any organelle which are present inside, for example mitochondria and all, mitochondrial membrane is also made up of lipid and protein. Okay, inside enzymes are present. So these enzymes are also protein. That means if I just keep analyzing the cell or the constituent of the cell, I mean material present inside the cell are nothing but the constituent, all are made up of biomolecule itself. Okay, that means organic molecule itself. In these organic molecule, like lipid, protein, carbohydrate, whatever the things that I am mentioning, these molecule will be having carbon inside them. Okay, so such a carbon containing organic molecules are called what? Biomolecules. But our cell not only restricted for biomolecule, I mean organic molecule. We know here is a cytoplasm. Right, it is a liquid part. So inside the cytoplasm, we have ions like a calcium, we have ions like a magnesium, we have potassium, okay, outside sodium, even inside also some quantity of sodium will be there. So these are inorganic molecules. Then what did you understand from this? Our cell consists both organic as well as inorganic molecule, correct? Now the question is, how we need to test it? Okay ma'am, you said our body or our cell, of course our body is made up of cell only, it consists organic and inorganic molecule. How we come to know? How we come to know that these are having these molecules? So for that we need to do certain tests, right? That is nothing but chemical analysis. How we do chemical analysis? Let's see. But before going to that chemical analysis part, we need to understand the types of biomolecule. Okay? Just make a note along with me or else you will get confused at the end. Just make a short notes within a textbook, or even you can make a notes also as a reference note, so that whatever the points that I am going to write here on the board, you just make a note in that. Right. So now, after finishing the class, when you just go through the notes, no, it will help a lot. And even if you read NCERT also, you can imagine the things just by looking at the notes, whatever the information that I gave. So really, it does help a lot during the examination. Okay, please do that. Now let's move on to the types of biomolecule. Here the biomolecule can be classified into two categories. One is called biomicromolecule and another one is biomacromolecule. Macro means what? Big. Micro means small. Microorganism, we are going to say, no, they are very, very small organism. Macro means very big. So let me take a simple analogy to make you understand. Can you imagine a wall? Okay, or else I will draw here. So let me consider these are the bricks. This is one brick. So like such many bricks I need to join to construct a wall, correct? One brick, two brick. So like that I need to place one above the other. So now, do you consider it as a wall? No. Why? Because you say, ma'am, simply they are aligned, but it is not a wall. If I just put a touch here, if I just touch this, mass of this brick, it will definitely fall. So that's why what we need to do, we need to bind it. Correct? So how we are going to do that? With the help of a cement. Correct? Let me consider it as a cementing material. So we need to bind like this. Now it is very strong. Now it is very strong. Now I can say, oh, this is one wall. Okay. Now, listen here carefully. If you consider this wall as a polymer, polymer means many monomer have joined to become a polymer, then obviously this brick would be monomer only. Now, if I ask the question simply, which one is having more weight? Whether a wall or a brick? Obviously, what a nonsense question, ma'am, it is. Brick is smaller. Right? So like that in our body also, many small molecules, just like a brick, they are going to join together and they become a big wall. Okay, so now I can consider this entire wall or a big wall as a macromolecule. And here, to make this macromolecule, there are small micromolecule has to join. Okay, so here if I take an example of a macromolecule, let's go back to the previous slide. Macromolecule, I can list protein as a macromolecule. Okay, it's a very big chain. So to make this protein, it requires a monomer called amino acids. Amino acid. Let's consider this is one amino acid. Okay, so like such many amino acid is required here. Okay, this is one amino acid. So 1, 2, 3, 4, like that many amino acid should get joined here so that it will become a protein. So here it required a peptide bond just like cement. Okay, so now this protein become a macromolecule because it has a many smaller units called micromolecule. Okay, so like that I can take even an example of a nucleic acid. So nucleic acid is something like this, you know, DNA. So here the monomers are present. So these monomers which are so present here you can say these are the monomers that are joined by a bond. These monomers are nothing but nucleotide. Okay, suppose if I have to become a big carbohydrate chain, a big sugar molecule, then I need monosaccharide. A small sugar in it, they join to become a big sugar molecule called carbohydrate. So, protein, nucleic acid, carbohydrates, these are formed by having many monomers. Many monomers in the sense amino acid for protein, nucleotide for nucleic acid, monosaccharide for carbohydrate. So now, based on their weight, we are going to classify them as a micromolecule, as a macromolecule. Now, if I just take one amino acid, its molecular weight will be very less. Like that nucleotide, monosaccharide, they show very less molecular weight, that is around 80 into 800 Dalton. Remember, it is very important. It shows the molecular weight around 8 into 800 Dalton. So like that, if I just measure a protein, it will be having more than 1000 Dalton. It could be 10,000 Dalton, more than 10,000 Dalton. So like that, if I measure nucleic acid, again it will be more, carbohydrate more. That means if I summarize it for the molecular weight of a macromolecule, I can write it will be having molecular weight more than 1000 Dalton. More than 1000 Dalton. It can be range up to 10,000 Dalton or even more. But generally we say a molecule which is having less than 1000 Dalton, we just go into the micromolecule group. Okay, I am going to check on one more molecule. I am just going to measure it. Its molecular weight. It is having more than 1000 Dalton. Then I will say we just come into the group of macromolecule. So here based on number of monomers, if only 10 monomers, 20 monomers, 1000 monomers, so like that, based on that, their weight can range from 1000, 10,000 or even more. Understood? So this is what the basic difference between the micromolecule and macromolecule. Now the question is how we are going to how we are going to analyze this is a micromolecule, this is a macromolecule, on which basis, on which chemical test you are going to analyze it. So for that, we need to go for a chemical analysis. Okay? Yes. How we are going to analyze? So for this purpose, take any tissue, either it is a plant source or an animal source. Take any vegetable piece, take any liver, take any meat, anything you take care. Okay. So now you subject it for chemical analysis. Now how we are going to take? In which container you are going to take? So for that NCERT says, don't worry, take a mortar. So this is nothing but a mortar. So in this mortar, you take any piece of vegetable, meat or anything. Now you take a pestle. So in kitchen, your mom is going to use this, no? pestle and a mortar to grind certain things, especially some spices and all, for grinding purpose, to make it into small pieces. She is just going to use this pestle and mortar. Okay, so this is nothing but a pestle. Now you have to grind it. For this grinding purpose, you need a liquid. Okay, so here you should not add any water. You have to add an acid here. Okay, so which acid you are adding? This is nothing but trichloroacetic acid. It is very important. Trichloroacetic acid. You just grind it. Now it become a very thick paste. That we call it as a slurry. Now you have to strain it into another container. Now take one more container here. Now take one more container. So on this container, you just place a cheesecloth or a cotton cloth, any strainer, okay, so that it has to get filtered here, no? Let's imagine I have put some cotton cloth here, okay. Now you just pour the container which are so present in this mortar. So after this, what happened? All the material which are so got dissolved in this acid, that is acetic acid, now they got strained here from this cloth and they start collecting down. So now I can give a name to this as filtrate. But many things which are heavier, which couldn't able to got dissolved here in the soil, now they just got collected here only on the surface of cotton cloth or a cheesecloth. Now I should give a name to this also. So this one is called retentate. Why retentate? It got retained on the surface. Why filtrate? It got filtered, just like filtering a coffee or a tea. So how we are going to got a filters at the bottom? How we are going to get the debris on the surface as a retentate? So in the same way, we are going to do this. Now, these filtrate many molecules, thousands, more than thousands of molecules which have got dissolved here within the acid. Therefore, once again, we could give another name to this also as a acid soluble pool. Okay, so filtrate itself is known as acid soluble pool. So now, if you just keep analyzing, if you subject it for further chemical tests, so that you are going to learn in your higher classes, what type of chemical tests that you are going to perform here inside the acid soluble pool to separate the material. Okay, so those tests are not given in NCERT, so let us not worry about that. But you just know here these material will be subjected for further analysis, chemical process, so that we are going to find many materials. So all those materials which will be having the molecular weight around 8 into 800 Dalton. It means what? These are all micromolecule, right? These are all biomicromolecule which comes under acid soluble pool. Now here, if you just test the material which are so present in the retentate are nothing but acid insoluble pool, which shows the material having more than 1000 Dalton, 10,000 Dalton, then obviously here the materials would be what? Biomacromolecule. And even in that, in this, so many inorganic materials also got dissolved. Okay, so this is how we can able to separate the macromolecule as well as the micromolecule based on the chemical analysis. Understood? Yes. Now, here, okay, let's go back here. So here, if I just keep analyzing what are all the material, then I will come to know that, listen here carefully, this is very important for your NEET exam. Here, I could able to get a protein. Okay, I do agree because protein is formed by joining many monomers like amino acid. Here, I could able to get amino acid. Here, I could able to get nucleotide. I could able to get monosaccharide. Many things, many monomers are going to get dissolved here. But here protein is available, nucleic acid is available, okay, and even carbohydrate is available. But lipid is also formed here only. Now it is a danger sign. Because lipid is not first of all a biomacromolecule. Because it is having the molecular weight 8 into 800 Dalton only. But how could it got separated on the cheesecloth only? So this is what the big question mark. To seek the answer for this, let's understand why it is happening. Just recall cell chapter. In the cell chapter, what happened? What we have learned? We learned the structure of plasma membrane, right? So the plasma membrane is made up of lipid bilayer. So how you could write here? It will be having the head and it will be having a tail. So it was having like this structure, no? This is hydrophilic. This is hydrophilic head and this part is hydrophobic tail. So always the phospholipid, this is phospholipid. Always phospholipid says, you can pour water on my head, but not on my leg. My leg should not touch the water. So this is what the condition. So that's why, how they made a plan, they started aligning themselves in this way. All the head, they were on the surface because here interstitial fluid is there. So like that here cytoplasm is there. To face that, they started showing their head at the bottom. So that they started upside down, forming upside down. So, like such molecule, we could able to get on the plasma membrane is nothing but lipid bilayer. So, already we have understood. Now, just think what happened. When you start grinding the material, that is vegetable or a tissue or any part, obviously the plasma membrane which are so present, they will become into a small pieces like this. Correct. So, when they become a smaller pieces, obviously, their lipid bilayer part also got separated like this only. Correct. Now, easily the water could enter. The water could touch their leg. So, to avoid that, again they started converting themselves into a bubble-like structure. So, something like this, they form. Okay. So, here is the lipid. Here once again the lipid started forming in this way. Can you understand what I am telling? So lipid layer, they started converting into a vesicle or a bubble-like structure. So bubble is a wrong word. Let me consider it as a vesicle only. So we call them as vesicle. Now if you just measure this single lipid, it weighs 18 to 800 Dalton only. 18 to 800 Dalton. Within that we could able to get its molecular weight. But because of this fear, so that if water touches our leg, we should not expose our leg to the water. Because having this fear, they are going to become a vesicle-like. They are going to align like this. So now, when you measure these vesicle, obviously, if you just keep counting, this would be 800 Dalton, another one is 800 Dalton. Just keep multiplied. Then obviously what happens? Its weight will be more than that of the 1000. Right? So that's why they are going to retain on the surface here on the acid insoluble food. Understood? So that's why in the written taint, even though the lipid is not having a molecular weight like a 10,000 Dalton, but still it is present on the surface because they form a vesicle. Okay. So this is what the answer for this question. If such question comes in your exam, you need to answer carefully by reading the question. Understood? Yes. So hope I am very clear up to the chemical analysis part. Now let's read NCERT. Here while reading NCERT, I will tell which line is important for your NEET point and which word is very important. You just make a note in your textbook also. Okay? Yes. There is a wide diversity in living organism in our biosphere. Yes, we do agree. Now a question that arises in our mind is, are living organisms made up of the same chemical? Okay? That is elements and compounds. They are asking, NCERT is asking question, are living organisms made up of the same chemicals, that is elements and compounds, which we are going to found on the, all, whether it is a plant or animal? Yes, the answer is yes. Okay, you have learned in chemistry how elemental analysis will be performed. If you perform such an analysis on a plant tissue or an animal tissue or a microbial paste, that means in this question they are telling, if you take a plant tissue, do they have a same element? Yes, like that of the animal tissue. Plant, animal, microbe, any tissue you take or any cell you take, do they have a same composition? Of course, yes. Even though the structures are different, some bacterial cell will be having a mesosome, all those structure, but still they have this component only. That is what the question they are asking. Now we obtain the list of elements like a carbon, hydrogen, oxygen and several others. and their respective content per unit mass of a living tissue. That means their content will be varies depending on the cell mass. If the same analysis is performed on a piece of earth crust, that is nothing but a non-living, as an example of non-living matter, we obtain similar list. That is what we talked in the first slide itself. Both living and non-living have the same component. What are the differences between the two lists? I said no, non-living is having only inorganic, living is having both inorganic as well as organic. That is the difference. In the absolute terms, no such differences could be made out. All the elements present in a sample of earth crust are also present in a sample of living tissue. That means they are telling all the elements are same to same. However, the closer examination revealed that relative abundance of carbon, important, hydrogen with respect to the other elements in the higher in living organism than in the earth crust. It means you are going to test the elements in the living organism as well as in the non-living things. Now, when you do such chemical analysis, in the living organism, hydrogen concentration will be high, carbon concentration will be high, but here in non-living, their concentration will be less. That is what the difference they are telling. But in the summary part, if you have a textbook with you, I didn't put the summary part here, but at the last, I am going to read the summary also. Now, if you have a textbook along with you, just open your textbook. Just look at the summary part. In the summary part, along with hydrogen and carbon, they have also mentioned oxygen. So, this is important. So, which element concentration will be higher in living compared to non-living? Then you have to mention these three elements: carbon, hydrogen and oxygen. So, this is important. Okay. Yes. So, they are asking, just check the table 9.1. Let's check it. Look at here. This is table 9.1. Here also, you could able to get a question in your NEET. What such question? Here the element list they have mentioned. Here the percentage of weight of Earth's crust human body they have mentioned. Look at here, oxygen. So, oxygen is 46 in the Earth crust. Whereas in the human body, it is 65. That's what that sentence. It is very important. Which material is more abundant? Oxygen. It is 65%. Okay. And even look at the carbon. Carbon is here 0.03 in earth crust, whereas in our body 18.5. And hydrogen 0.14. Here 0.5. So these three are more compared to earth crust in our body. Okay. So again, it could be a NEET question. And which one is very, very little? Very little is nitrogen. And sulfur, sodium, calcium, magnesium. So all these are compared to our body, earth crust will be having more. Okay, so these data, even if you won't remember, no problem, but these 3 points are very important. And the last one, once again, it could be a NEET question. Silicon, it is having 27.7 in earth crust, whereas in our body, it is negligible. Okay, again, it is a NEET question. So remember this table. Next. How to analyze chemical composition? They are asking the chemical analysis part. We can continue asking the same way. What type of organic compound are found in the living organisms? How does one go about finding the answer? To get an answer, one has to perform the chemical analysis. That is what we did, pestle and mortar grinding experiment. We can take any living tissue, a vegetable or a piece of liver, etc. and grind it in a trichloroacetic acid. See, this formula is also important. Cl3CCOOH. I wrote CCl3COOH. Both are same. Don't get confused, ma'am. You wrote CCl3COOH, but NCERT is telling Cl3COOH. Anything is okay. Okay. Using mortar and pestle, we obtain a thick slurry. If we were strain this through a cheesecloth or a cotton cloth, we would obtain two fractions. One is called filtrate, or more technically the acid soluble pool. Okay, and the second is the retentate or acid insoluble fraction. The scientists have found the thousands of organic compounds in the acid soluble pool. So hope you have understood this paragraph. Then in higher classes, you will learn about how to analyze a living tissue sample and identify the particular organic compound. That means in an acid soluble pool, so many things are there. No, oh, this is amino acid. How are you going to analyze? Simply just by looking at the filtrate, we can't say, oh, here is amino acid, here is a monosaccharide, here is a lipid. Lipid is not there, first of all, sorry. Okay, nucleotide, how you are going to do that? Of course, you have to subject it for chemical analysis. So by looking at the molecular structure, by looking at the molecular arrangement, then you can come to the conclusion that, oh, this would be the monosaccharide, this would be the amino acid. So for those things to understand, you need to go for higher classes. That is what the meaning of this paragraph. Let me read this also, but question won't come from this line. It will suffice to say here that one extract the compounds, then subject the extract to various separation technique till one has to separate the compounds from all other compounds. That means so many compounds are there inside, so we need to separate everyone. In other words, one isolates and purify a compound. Analytical techniques, when applied to the compound, give an idea of the molecular formula and probable structure of the compound. So all these things will not be asked in your exam. So they are just mentioning, after analyzing the structure, obviously you are going to know the molecular structure, formula, everything of that particular compound. This is important. All the carbon compound that we get from the living tissue can be called biomolecule. That means biomicromolecule itself can be called as biomolecule. Okay, which we got in the acid soluble pool. Here NCERT says just all the carbon containing compounds that we get from the living tissue. But I mentioned all the carbon containing organic molecule. Ma'am, which one is right? Should we write the same definitions or what you wrote? Both are same. Here NCERT has not mentioned organic word, but organic word should be there. But suppose if in a statement based question, if such statement comes, then don't write false. Just go along with NCERT, just write true only. But for a better convenience, for more accuracy, I use the term organic. Okay? Yes. However, living organisms have also got inorganic elements. Okay? And compounds in them. I said no, in the cytoplasm, we have even inorganic molecule also. How do we know this? Yes, here is one more experiment. How you come to know that living organism, I mean living tissue, will be having even inorganic molecule also. For that, you can do one more test. Take any piece, take any tissue, either it is a vegetable or a plant, or even you can take any liver part, anything you can take. So this is a wet mass. As it is, wet mass means it will be having water content inside it. Now you just dry it. You just dry it. Now it is something like this. It become dry. Now it is a dry weight. Okay. Now with the help of a burner, you just burn it. So once you burn, what you are going to get? You are going to get an ash. So when you just burnt this, we are going to get ash, right? Now you subject this ash for chemical analysis. What you are going to get? You are going to get inorganic molecule. Phosphorus, nitrogen, oxygen, everything, because all carbon dioxide will be get evaporated. Okay, during burning. So left behind materials are nothing but only those nitrogen, phosphorus, sulphur, those things only. That is what the meaning here. Let's read. A slightly different but a destructive experiment has to be done here. That means to check the inorganic element present in a living tissue. So what we have to do, one weighs a small amount of a living tissue, say a leaf or a liver, and this is called wet weight. As I said, wet weight or wet mass, both are same, and dry it. All the water evaporates. The remaining material gives a dry weight. Correct. Now if the tissue is fully burnt, all the carbon compounds are oxidized to gaseous form, that is carbon dioxide and water vapor got evaporated. What is remaining is nothing but a ash. This ash containing inorganic elements like a calcium, magnesium, all those things. Here inorganic materials list they have given. Sodium is there, potassium is there, calcium, magnesium, water, other compounds, everything is present in this ash. Okay. So these inorganic compounds like phosphate, all these things are also seen in acid soluble fraction. It means they are trying to tell that in our living tissue all these things are present. That's it. Let us not elaborate much. Therefore, elemental analysis gives the elemental composition of living tissue in the form of hydrogen, oxygen, chlorine, carbon, etc. While analysis of compound gives an idea a kind of organic and inorganic constituent present in the living tissue. That means just now we have seen organic molecule as well as the inorganic molecule. Okay. So in the chemistry point of view, we will call this term as aldehyde, ketones and aromatic compounds. But in biological point of view, we shall classify them into amino acid, nucleotide, fatty acid, bases, etc. Okay. And here one more table is there. Okay. This is actually table 9.4. It is not just after the 9.2. But I need to connect it to this topic. No, that's why I put this table here. From this table also, the chances of asking question will be high. What they could ask, which component is having more mass in our body, cellular mass, which component? That is nothing but water. Water is 70 to 90% present inside our body. Okay. I mean, in the cell. Then protein part is 10 to 15%. This is in a first rank. Apart from water. Water it's the topmost place. And if you just leave the water, then if you just keep counting the percentage of rest of the things, then the protein ranks higher. Okay, let me write here protein. Then afterwards, nucleic acid, it is 5 to 7%. This holds a second rank. Let me make N here. And after that, carbohydrate, it is 3. It is third place, C. Then lipid 2, N. So how we are going to remember? This is ion. Ion is just a 1. So let me write ion at last. How you will remember? Pencil. Here don't put E here for pencil. Just pencil. Okay, pencil means protein, nucleic acid. Protein, nucleic acid as well as the carbohydrate and the lipid. Last one is ion. That's it. You can remember in this way. So hope I'm very clear in this part. Now, before entering into the organic molecule part, let's solve few MCQ regarding this. So, like this, I have done for all the chapters. After completion of one topic, we will discuss few MCQ. Then we'll move on to the next topic. So, like this. So that if you have any doubts, we could get solved here only. Okay? Yes. If you have understood the class very well, then easily I can bet that you can answer the question without taking much time. Which substance is most abundant in the cell? Okay, carbohydrate, protein, water, fat. Just now we discussed which one. Yes, your guess is right. It is water. Next, which element is present in negligible in human body? Which one is negligible? Just recall the table. Oxygen, carbon, hydrogen. In fact, they are present in more quantity. Silicon is actually negligible. Correct? Which of the following compound is found in acid-soluble pool during the analysis of living tissue? In acid-soluble pool, all macromolecule were there. So, protein is a macromolecule. It is not present. Lipid, though it is a macromolecule, but because of their nature, so that it should not touch the water, that's why they form a vesicle. They also found on the retentant. Polysaccharide is nothing but a carbohydrate only. Many monosaccharide join to become a polysaccharide. So, they are also found on the acid-insoluble pool. Then, the only left item is monosaccharide. Monosaccharide amino acid, but here monosaccharide word is given, therefore this is our answer. Yes, which of the following is not a macromolecule? Not a macromolecule. Amino acid, micromolecule, it is not a macromolecule. Nucleotide, micromolecule. Monosaccharide, micromolecule. Which is not a macromolecule? No one is macromolecule, therefore all of the above is the answer. All are micromolecule only. For the chemical composition analysis, dash is used. CH3COOH, no. Trichloroacetic acid. Here chloro is there. Here Cl3 is not there. Therefore, this is also wrong. Cl3CCOH. This is our answer. Here Cl3 fine. But one more C is not there. That's why it is wrong. Next question. Acid insoluble pool is also known as retentate only. Slurry retentate. Filtrate, which is present in the acid soluble part. Not all. If the tissue is fully burnt, what we are going to get? Ash. All the carbon compounds are oxidized to gaseous form, carbon dioxide and water, correct? Remaining are known as ash, correct? Ash containing inorganic elements and inorganic compounds, correct? So all statements are correct here, therefore this is our answer. Okay, so hope I am very clear about the first part. Even if you have not understand, just rewind the video because it is a recorded video, it's not a live session. You can watch it again. Okay. Yes. Now let's move on to carbohydrate. Yes, carbohydrates. It is also called sugars, even called as saccharides, nothing but somewhat sweetish in nature. Okay. So even I will just give few examples so that you can understand. The table sugar that we are going to eat, no, that is also carbohydrate. The ragi, the rice, and even the wheat, when you start chewing it, at the end you are going to feel somewhat sweetish in nature. That is also kind of carbohydrate. So carbohydrates are nothing but it is one of a component of our food. But here, having a carbon molecule in this, it comes under the group of organic molecule. So now we need to learn few things about the carbohydrate. In NCERT, just only a paragraph given about carbohydrate, but questions will come from the structure part. So we need to learn the structure. But before entering into that, let us understand few basic things. See, in the word carbohydrate, if we just split this, we are going to get two words, right? One is carbo and another one is hydrate. So here, the word carbo means nothing but carbon. So what does it indicate? Here, these organic molecules, they will be having an atom called carbon in them. So that is what the indication. So like that, when you just look at the term hydrate, hydrate means what? Water only, no? So water is made up of H2O. So what are all the elements present here? H as well as O. That means whenever we talk about a group of organic molecule, carbohydrate, remember one thing, in the carbohydrate, we have these group. Okay? It will be having C, it will be having H, it will be having O. So that is what the first basic thing we need to learn. And here, I have written also, it has mainly the compounds of carbon, hydrogen and oxygen. And here in the word carbohydrate, the two functional group that we could able to see are, one is hydroxyl group and another one is CHO group, OH as well as CHO group. Okay. Now let's enter into the classification of carbohydrate. All these things about the classification is not given in NCERT. But here we are preparing ourselves for NEET exam. Okay. So that's why we need to learn all these things. But trust me, it is very simple. Though NCERT has not given in the format of classification, but the information regarding these things, what I am going to give now, will be given maybe in the form of a structure, maybe in the form of certain words, but the question can be asked from those structure itself. The question can be asked from those terms itself. So that's why, if you know all those things, what is the loss in that? Okay? Yes. Come, let's begin. So carbohydrate, based on their group, some may having very big molecule. That means by joining small group together. Some may having a moderate size. Some may having very simple sugar. Okay, just one monomer. So based on that size, we could able to classify these carbohydrates into three categories. One is monosaccharide, second one is oligosaccharide, the third one is polysaccharide. I will tell you, very simple. Monosaccharide means what? Just few minutes back, I gave example of a wall, constructing a wall, right? So in that wall, what I said, small bricks. Let's consider this is one brick. Okay. So this brick itself, you can consider as a monosaccharide. Okay. So imagine this is one monosaccharide. That means it is the smallest sugar. It is the smallest molecule. So further, you cannot break this. Further, you cannot break this monosaccharide into further simple sugar, because they are the smaller one. They are the monomer. Okay. But here in this sugar itself, the carbon atom number may vary. For example, some molecule will be having 3 carbon atoms and it may extend up to 7 carbon atoms. Okay. For example, if a monosaccharide, simple sugar, if it is having a carbon molecule of 3, it means what? C, like that. If 3 carbon atoms join together, then such sugars are termed as triose sugar. Okay. Suppose if they are having carbon molecule or carbon atom 4, then we call them as tetrose sugar. If it is having carbon molecule 5, that means 5 carbon atoms, then it is called pentose sugar. So in our NCERT, the examples are not given from triose sugar or tetrose sugar, but you need to learn about the pentose sugar. So the best example for this is ribose sugar. So this sugar even follow you in your class 12th also. There you are going to learn a molecular basis of inheritance chapter. There we need to learn about the structure. So here itself we will learn this basic structure. So it is very important. Not only at class 12, even in class 11th also. And after that, if these organic molecules, if it is having 6 carbon atom, obviously it is called what? Hexose sugar. So the example comes under here. For hexose are glucose, fructose and galactose. Okay. So if they having 7 carbon atom, then it is called heptose. So need not to bother about the heptose, need not to bother about the tetrose or triose. Only thing is 5 carbon atom, 6 carbon. Only pentose as well as the hexose. Okay. And the next category is, what is oligosaccharide? As I said, it will be having only one monosaccharide. There is one simple sugar. It is a monomer. Okay. So now, if these monomer join like this is one monomer, there is one monosaccharide. I will take one more monosaccharide. I will take one more. Like this, if I keep joining all these monomer with the help of certain bond. Okay. Just like how we are going to construct a wall by placing a cement in between the bricks, in the same way, here we are going to construct a bond. Okay. So this bond is called what? Glycosidic bond. We are going to learn shortly about the bond formation, but you just know it is a glycosidic bond. So such sugar, if they have, what I am telling, listen to me carefully. If the sugar, if they have 2 monosaccharide too, okay, 2 to 9 monosaccharides or 2 to 9 monomers join together, then we call them as oligosaccharide. Okay, so when you just grind a liver or when you just grind any vegetable piece or any molecule, so when you just start filtering it, as we have learned just now, these oligosaccharide as well as the monosaccharide, that will be comes down in the acid soluble pool because their size is very small. Okay, so here if I am telling 2 monosaccharides are joining, one monosaccharide, another monosaccharide, both are joining like this, then I can give a name to this as disaccharide. Disaccharide, like that. If 3 monosaccharides join, then it is called trisaccharide, tetrasaccharide, pentasaccharide. So like that. But in our NCERT, they have not mentioned anything except this disaccharide. So that's why you just know if they have more than 2 as well as less than 9, then they comes under the group of oligosaccharide. Okay, somewhat medium sized. So here for these molecules, for disaccharide, we could get 3 examples. The first one is maltose. We need to learn the structure of maltose. So what is the structure of maltose? If you take one glucose and join one more glucose to this, then it becomes a structure called maltose. Like that, there is one more disaccharide I can take, lactose. So to this also, just take one glucose. To this glucose, add one galactose. See, galactose is also monosaccharide, right? So that's why I'm adding galactose here. So how you can remember? See, lactose, galactose. So in the word itself, it is indicating that I am a part of lactose. Okay. And the third example we can take as sucrose. So here, glucose is a common. In every 3 molecules, glucose is common agent. And along with that, we are going to see one more sugar called fructose. Okay. So these 3 examples can be comes under the group of oligosaccharide. So here we need to learn the structure of pentose sugar, that is ribose sugar, as well as hexose. In the hexose, we just need to learn the glucose structure. And after that, we need to learn maltose only in disaccharide. And after that, let's enter into the polygosaccharide category. What is polygosaccharide? Very simple. You can analyze, you can guess what I am able to tell here. Okay. So here, I am trying to tell about the polysaccharide is nothing but if the molecule having more than 10 monomers, monomers in the sense here monosaccharide. So their number, their range can be varied. It could be 10, it could be 100, it could be 1000, but it should be more than 10. Okay. So like this, they are joining. They just keep joining like this. So obviously, being so heavy, how you can expect in acid soluble pool? Obviously, comes under retentate only, no? So let me write here, these polysaccharides are a part of acid insoluble pool. That means they are a part of retentate. So hope you are understanding. Whereas monosaccharide as well as oligosaccharide, they comes under acid soluble pool. Okay, so these two things we have understood. Now in polysaccharide, we need to learn for example also. Let's learn in the next slide. Hope you are making notes along with me. Polysaccharide, more than 10 monomers, more than 10 monosaccharide. So here once again, we can classify it into two main categories. One is homopolysaccharide, another one is heteropolysaccharide. Very simple. What is the meaning of this? Imagine I have a glucose here. Again, I have brought one more glucose. Let me write it as glucose. Now, I am going to join all this glucose with the help of glycosidic bond. That means what? All are showing the same monomer. One type of monomer, right? Then such polymer is known as homopolymer or homopolysaccharide because we are talking about saccharide. Okay? So, this is one category. But in certain polymer or in certain carbohydrate, what happens? Imagine this is one kind of monosaccharide. So, what I will do, immediately, I will take one more. So, all are different. So, let me consider this as a glucose, this as fructose. Again glucose, fructose. Are you going to find the same kind of monomer here or same monosaccharide? No. So, all are different in the sense they are different, not of one type. When they join together by a glycosidic bond, then such polysaccharide, we call it as what? Heteropolysaccharide. It doesn't mean it should be bonded with only glucose and fructose. It could be any monosaccharide. Okay. So, their nature should be different. It is not of the same kind. Just for making you to understand, I am taking this example. Okay. Now, let me take few examples to make you understand. Now, let's come to homopolysaccharide. Here, though it is a homopolysaccharide, but still, based on their function, again we are going to classify it into two categories. One is storage homopolysaccharide or homopolymer. Another one is structural homopolysaccharide. Here name itself is telling storage. There are certain polysaccharide, they act as a storage molecule. For example, you know about the starch, correct? So, in the case of plants, the food is going to store in the form of a starch. Do you know one thing? If I just draw a plant here, this is a plant, right? So, plant is going to prepare the food by photosynthesis. No, these are basics, photosynthesis. So, in this photosynthesis, it is going to prepare glucose. So, glucose has formed. But plant says, I cannot hold glucose here only. I need to transport further. I need to transport it into different parts. Okay. So, it start transporting. But for the transportation purpose, it is not transport in the form of a glucose. It will change itself into sucrose. It change into sucrose for the purpose of transportation. So, how these things happen? That will learn in plant physiology. Okay. But it will transport in the form of sucrose. But finally, imagine here it is going to store in the form of a vegetable. Let's imagine here all these molecules are going to store here. But how they are going to store? They convert themselves into one more structure called starch. So therefore, starch is a stored food. Where in the case of plants, all the vegetable, all the pulses, cereals, what we are going to use, what we are going to eat, nothing but the starch only. Okay, so let me write here. So starch is the storage molecule in the case of plants. So like that, even in case of animals also, when you start having excess of food, what our body will do? So after utilizing the amount it required for metabolism, then the rest of the things is start stored into where? In the liver. So in the liver, it start storing in the form of a molecule called glycogen. Okay, it starts storing glucose. Number of glucose, it will be started adding like this in the form of glycosidic bond. So that finally it become a bulk molecule. So that is nothing but a glycogen. So glycogen is a stored molecule in the case of animals. So like that, one more example we can take, that is inulin. It is not insulin. It is inulin. Don't get confused. Inulin is a stored food in the case of a plant called dahlia. In the root of dahlia plant, the inulin will be stored. So these three examples you need to remember. Okay. And after that, when it comes to structural polysaccharide, so definitely you heard the name about cellulose, right? So you take any plant cell. Obviously, the plant cell wall is made up of what? Cellulose. So it is forming a structure in a plant cell. So therefore, it could be a structural polysaccharide. Let me write here, present in cell wall. Present in cell wall of plants. Okay. So one more example I can take. Chitin. So chitin, it is an exoskeleton in arthropods. And even it acts as a cell wall of fungus. Yes. So these are structural polysaccharide. Here also, for the purpose of learning, so you need to remember the names. That is fine. But when it comes to learn the structure, then we need to learn the structure of a glycogen. Only this one. Not cellulose, chitin, inulin, starch, nothing. But only few points we need to remember about the starch as well as the cellulose. But apart from that, if you learn this much, that is more than enough. And one or two line I may add while explaining, but this is more than enough. Okay. Then when you come to heteropolysaccharide, as I said, it is made up of different monomer. Here, same monomer. So here, let me take one example. So you know about peptidoglycan. So peptidoglycan is a cell wall of bacteria, correct? And so if you just analyze the bacterial cell wall, its name is peptidoglycan, right? But it is made up of different monomer. So here, one monomer name is NAG, NAG. And another monomer comes, its name is NAM, NAM. So again, they are going to join by a bond, that is glycosidic bond. Again, NAG, again, NAM. So like this, they are going to make NAG, NAM, NAG, NAM. So like such monomer, they are joined together and form a polymer called peptidoglycan. So here NAG means what? NAG means N-acetyl glucosamine. So how these structures are forming, what is acetyl glucosamine? No need to worry. So don't worry about all this structure, you just know the name. It is nothing but N-acetyl glucosamine, that's it. So like that, the full form of NAM is N-acetyl muramic acid. Muramic acid. That's it. Okay. So just please make a note. It is very, very important. Now let's move on to the structure. So this part is over. Let's move on to structure of ribose sugar. So just now I said it comes under the group of what? It comes under the group of monosaccharide. So this is first thing we need to remember. Particularly in monosaccharide, it is a pentose sugar. Why we call it as a pentose sugar? Because it is having 5 carbon atom in this structure. Okay. So let's see how we need to construct this. It is very simple structure. It is given in our NCERT. Let's look at here. Carefully, I will construct it easy. Easily you can learn it. Okay. So let me construct a ring-like structure here. In the center, we need to place oxygen because we know that carbohydrate is nothing but CHO only. Only these molecules are present. So now, we need to construct a ring-like structure in this way. Just practice it along with me. You can learn it easily. Now here in these edges, it indicates that the carbon atom is present. Imagine this is the first carbon atom. This is second carbon atom. This is the third carbon atom. This is the fourth carbon atom. But I said here 5 carbon atom is there. Where is the fifth one? Here it is. These carbon molecules is, look at here, I am here on the top. Here is fifth carbon atom. But fifth carbon atom is not simply present. It is present in the form of CH2. OH even. OH. So here the functional group of carbohydrate is OH. correct? So we need to add OH, either OH or EH. So if one end is having OH, the other end, that is the opposite end, must have H. So here OH is there, therefore I should write H here. Okay, so now in the same way, for every carbon atom, just put a line like this. Okay, now it is having more affinity for OH. Let's learn like this. This ribose sugar, it is having more affinity towards the OH group. So therefore, it is just placing everywhere OH at the bottom side. So it is decorating itself with the OH. Let's imagine here is one OH, here is one OH, here is also one OH, only one H at the last part. Okay, so everywhere it has been decorated with OH. Let me write in this way. So this is one beautiful ribose sugar. It says I am very fond of OH. So let me place OH under my leg, so that everywhere we are just placing a OH group. So only at its hand, it has just hold a H group. That's it. So obviously the opposite side must be having H here, H here, H here. So this is what the structure. This is what the structure. Okay. Don't write these I's and all. Understood now? Over. If you practice it twice or thrice, it will come. Okay. Now, after learning this, we need to learn even the structure of deoxyribose sugar. So why we need to learn this ribose sugar and deoxyribose sugar? I said these two sugar will follow you even in your class 12, right? So let me show here. We know about nucleic acid. Again, we are going to talk about this shortly, but let me tell you here also. Nucleic acid is nothing but DNA or RNA. Okay. So DNA is a polymer. It's a polymer. Here also it consists many monomers like this. Many monomers. All these monomers are joined with a kind of bond. It is not a glycosidic bond. It is something else. It is something else. I will tell you, it is a phosphodiester bond. But monomer must be present here. These monomers are nothing but nucleotide. So nucleotide is not just one simple nucleotide. It is also complex. It is having three structures along with it. So what are those three structures? Look at here. It is having one beautiful sugar. So this is the sugar. So this sugar, okay, this sugar is making a friendship with this nitrogen base. And the same sugar, it is making even a friendship with phosphate group. Phosphate group. Okay. So that's why if we are saying that one nucleotide is there, means it must have these three molecules together. Nitrogen base, sugar and phosphate group. Here, the hero, that is the sugar, what you are going to see, it is looking like a girl. But it's okay whether it's a girl or a boy. Right? So this sugar, if it is a case of RNA, this sugar is nothing but a ribose sugar. It is a ribose sugar. Just now we have learned the structure, how ribose sugar will look. Okay. Suppose if it is a DNA, phosphate group will be there as it is, nitrogen base will be there as it is, but this sugar is not a ribose sugar. In the case of DNA, this sugar is deoxyribose sugar. Understood? Very simple structure. It is exactly ribose sugar. Only the thing is just one oxygen atom has got deleted. That's it. I will tell you. I will tell you. So hope you are understanding what I am trying to tell. Yes, let's come back to the structure of deoxyribose sugar. What it is? Once again, it is also a pentose sugar. No change in this. It is also a monosaccharide. No change. Obviously, it is also having 5 carbon atoms. Okay. So now, how this structure will look? Very simple. Oxygen atom is there. Just like in the case of ribose sugar, just make 2 arms in this way. Just place carbon 1, carbon 2, carbon 3, carbon 4. Where will you write the 5th carbon atom? Obviously, yes, here. 5th carbon atom, which has been having CH2OH. Correct? So, what I said earlier, just put a line like this. Correct? And the ribose sugar, it is having more affinity towards the OH group. So, that's why everywhere we just wrote OH, 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 and only one H is having in its hand. Okay. Suppose just imagine a girl who is having a basket. And here on her 2 leg, she is having OH. And even on her, in her hand, she is carrying one OH. But just not to make a discrimination for H, only one H she is carrying in another hand. Okay. So this was ribose sugar. But now in the deoxyribose sugar, what you are doing, we are just deleting one oxygen. So here D means what? D. D means delete. What you are going to delete? Oxy. Oxy means oxygen. Just delete one oxygen, man. Then it becomes deoxy. So from where you are going to delete one oxygen? From here. Just remove one oxygen from here. So H will be as it is. H will be as it is. So that's why in the second carbon position, one oxygen atom has got deleted. Then the structure becomes deoxyribose sugar. Now in this girl, if she is a deoxyribose, then you have to remove one oxygen from her leg. That's it. Over. Deoxyribose sugar structure. So pentose sugar structure we completed. Now we need to learn the hexose sugar structure, particularly the glucose. Let's learn that also. That is also very easy. Very simple. So glucose, it is having 6 carbon atom. So it is a hexose sugar. Right? Obviously this is also monosaccharide only. So we are going to draw the structure again. Just put a ring structure, but this ring is quite different. It is somewhat like this. Hexa means 6, no? We need to place 6 carbon atom. So that's why, how we are going to place the carbon atom? Here the first carbon atom we are going to place. Here this is the second position, second carbon atom. Here it is the third position. Here this is the fourth position. Here will be the fifth position. Then where is the 6th carbon atom will you place? Obviously just like ribose sugar, just place it on its top. Here is 6th one. So here CH2, once again OH group will be there always. And here it is mandatory that oxygen atom is always here. Okay. Now just start putting lines. Everywhere you just start putting the lines. Now this hexose, that is a glucose, in her both hand she is carrying what? OH molecule. Okay. So if you just know where to write OH, then obviously the opposite side will be what? H only. So no need of writing H. And here on her leg, okay, in her leg, she is having one H group here. So write OH here also. So if it is OH, opposite side will be H. Here you just place OH. Over, the structure is over. So wherever you are having the opposite of OH, just place H. Just place H. Just place H. Then we call it as a alpha glucose. Why it is called alpha glucose, ma'am? Just based on the position of OH in the first carbon atom. If this first carbon atom, if it is having OH here, then it is alpha glucose. But instead of OH down, if the OH is present in the top side, then obviously this would be only H. Then it is called beta. Then this will be considered as beta glucose. But here, no need to learn the structure of beta glucose. Just to know the structure of alpha glucose, because this one will come to help you in constructing glycogen also. Okay. Just you know this structure. I am rubbing it again. Just write OH and H. Very simple. Over. Okay. So 2 OH are, let me draw the girl again. So here in her both hands she is carrying OH. Both hands she is carrying OH. But there in the case of ribose, only one hand was having OH, other hand was having H. Right? But our both leg were having OH. But here, only one leg is having OH. There in the case of deoxyribose, this was H. And this was OH. But here, it is reverse. It is H. Rest of the things you can understand. If it is CH2OH, obviously the opposite side will be H. Okay. So this is what the structure of glucose. So glucose can be written as C6. If you count the carbon atom, we are going to count, we could get 6 carbon atom. 1, 2, 3, 4, 5 and 6 1. H and O. So these are the 3 atoms, right? How many H are there? See, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. This would be the 12. So C6, H12, O6. Oxygen is also 6. Okay? 1 O, 2 O, 3, 4, 5, 6. C6, H12, O6. Whereas the case of ribose, where it went? Yes, ribose sugar, C5, H10, O5. C5, H10, O5. That is double. 1, 2, 3, 4, 5. H10, how come? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Oxygen 5. 1, 2, 3, 4, 5. Suppose if it is deoxyribose, just delete one oxygen, man. That's it. C5, H10, O4. Simple. So, ribose sugar structure we have understood. Deoxyribose sugar we have understood. Hexose sugar, that is glucose structure also we have understood. Now let us construct one more molecule, that is maltose. Maltose is a disaccharide. Correct? Let me write this first. It is a disaccharide. I know you are facing it quite difficult. It is just a beginning. That's why you are feeling like this. If you practice it, definitely it will come. It is a disaccharide. First thing is, it is made up of 2 glucose. Remember, one glucose joined with glycosidic bond with another glucose, so that it form a disaccharide called maltose. And here, this maltose structural formula, I will tell you at the end. Okay, what is the structure it is having? Not structure, molecular formula. So for that purpose, we need to construct what? 2 glucose molecule, right? So you just start constructing in your copybook. Let me construct here. So this is one glucose, right? So this is one more glucose. Okay. So let me mark it. Okay. Before that, let me write here oxygen. Yes. Okay. Yes. Let's start writing it. Here on the top, CH2OH. Where will you write OH? Here in the first position also OH. Second also OH. Third is H. Here also OH. Because both the hand is having OH. So like that. Here also you just start putting CH2OH. Place here also OH. Here also OH. Here H and here OH. So opposite thing I won't write because it is understood that it is H. Okay. Now, this is one glucose molecule. This is another glucose molecule. 2 glucose molecules are there. Now the question is, how we are going to make a bond between them? Very simple. So these 2 glucose molecules, imagine this is one glucose. This is another glucose. Both come together. Let's make a friendship. So you are also glucose, I am also glucose. I will give one H. Okay, one glucose says, I will give one H from my first carbon atom. And another glucose is, I will give one OH from the 4th carbon atom. So that when both react together, one H, another OH, what is the product? H2O, 2H plus 1O, that is H2O. So H2O is nothing but water. So when you just remove water from these 2 glucose molecules combining, then obviously they become a bond. So how it happened? Look at here. This is OH, this is OH. Now, you just remove one OH from here. Just take one H from here. Then obviously what happened? Obviously what happened here? So we are going to get O in the center and this will become a bond. How come it happened? This is because we have removed a water from this. Water we have removed. Water has been removed so that they made a bond between them. So this bond is nothing but glycosidic bond. Glycosidic bond. And in the beginning itself, I have introduced a term called alpha while writing glucose structure, because here at the bottom they have OH group. Therefore, this bond is called alpha. Listen to me carefully what I am telling. This is called alpha and it has made a bond between first carbon atom as well as the glucose of another glucose of fourth carbon atom. So therefore I can write alpha 1,4 glycosidic bond. Did you understand? Alpha 1,4 glycosidic bond. So you have to remember the bond name. Now here if the question comes, what is the molecular formula? What is the molecular formula of this maltose? Then if you get many options, in that there is an option called C12H24O12. So what will you do? Many students, earlier this question came in NEET exam. So many students, what they did, they just put a right mark to this option H24O12 because they thought that this is a combination of these two glucose molecules. Glucose is C6H12O6. Add one more glucose C6H12O6. Then obviously 6 means 12. 12 means 24. 6 12. So this is what the answer, no ma'am. So what is wrong in that? Remember my students. Remember here. We have removed water for making these glucose molecules for joining. Okay, when they join together by removing water, then only it become a maltose. So that's why whenever and wherever, if you are writing an answer as a molecular formula for maltose, remember you have to remove 2 hydrogen and 1 oxygen. Then what would be the answer? So carbon remain as it is, C12, because you have not removed any carbon here. And in the H case, you have removed 2 hydrogen molecules in the form of a water. So therefore, just delete 2 hydrogen from here so that it will become H22. And you have removed even 1 oxygen also. So oxygen instead of 12, it will become 11. So therefore, the molecular formula of maltose is C12, H22 and O11. Remember, it's important. Write up note. Okay, just write down in your copybook. So this is what the structure of maltose. Yes. So monosaccharide structure we have learnt, disaccharide structure we have learnt. Now the glycogen. So glycogen already have told it is a polymer of glucose. So you take one glucose, make a bond. Take one more glucose, make a bond. Take one more glucose. Like that, if you just keep adding glucose, obviously you are going to get a long chain of glucose. Correct? So this long chain of glucose itself, my dear, we call it as what? Glycogen. In our liver, the glucose is keep storing like this by glycosidic bond and it make a structure called, or it make a food called, reserve food called glycogen. But this is not simply a straight chain. So if I just show here in this way, somehow it looks like a straight chain. But once again, somewhere else it is going to show a branching like this. In this branching, again the glucose molecules are going to be add up like this. Again, one more branching. Here also the glucose is going to add. So like that, we are going to see many branches so that the glucose molecule is going to join in this way. Now we need to learn how we need to construct a bond. How this glycogen will look. Though it seems to be quite complicated, but trust me, if you know how to make a bond between 2 glucose, that is in maltose, easily you can do in the same way. Okay. Only thing is you need to be very cautious in branching portion. Okay. I will tell you, I will teach you, don't worry. So this is a branch structure. Let's learn this structure now. Okay. We don't have space. Let me show here only the glycogen structure. So what I said, it is a polymer of glucose. Correct? It is a homopolymer. That means same molecule. So let me draw glucose structure first. You just write down in your copybook. So this is one glucose. One more glucose. Write down along with me. So like this, if I just keep writing, I can write plenty of glucose molecule. Okay. Here, for making a bond with glucose, I just need only first carbon as well as the fourth carbon atom. So that's why I am writing only this one. First and fourth. First, fourth. First, fourth. First and fourth. That's it. So everywhere, what I am going to get? Here OH, here also OH. OH. Everywhere, just write OH. Rest of the thing we know already. Okay. So at the time of bond making, what happens? First carbon atom of one glucose, fourth carbon of one glucose. Okay. So they started donating one OH and oxygen. I mean one OH and one hydrogen. Right. So as a result, what we are going to get? We are going to get a bond like this. So here also everywhere we are going to get the same. Okay, we are going to get the same thing. So what I can write here? So this is alpha 1 4 1 4 because it is first and fourth. Again, this is first and fourth, first and fourth, first and fourth. So everywhere we are taking OH and H from 1 and 4th carbon only. So therefore alpha 1 4 glycosidic bond this is. So straight chain we have constructed, but it is a branched. Okay, let's imagine there is one more glucose chain is coming in this way. Let's assume here it is coming in this way. Okay, now let's assume this is the first and this is the fourth, right? And between these two, the bond has constructed 1 4 glycosidic bond. Here also first as well as fourth, a bond has constructed. But here is the problem. Here this first carbon atom is ready to donate its group. Okay. But where is the fourth carbon atom? This fourth carbon atom already got engaged with this one, right? So that's why this glucose says, I don't have any molecule to donate you for water molecule. That is to make a bond, to release the water molecule. So therefore, just look at here. Look at here in the top. I have a 6th position. In the 6th position, I have CH2OH. I have CH2OH. So in your first position, you have OH again. Okay. Now you can make a bond between these two. So I will give one OH, you can give one H from your side. So as a result, let me rub this part. As a result, what happened? Here we could able to get a CH2 in this way, and OH has been donated from this molecule and H has been donated from this carbon molecule. So therefore, here in the branch chain structure, we are going to see a bond between alpha 1 6 glycosidic bond. Alpha 1 6 glycosidic bond. Understood? So like this we could construct. This is what the structure of glycogen, very simple structure. So that's it. If you learn the glycogen structure, all structure has been completed. Only thing is rest of the information we need to just remember. That's all. Okay. Look at here. In the case of cellulose, what are the information already we know? The same thing we need to repeat. That's all. So cellulose, it is a structural polysaccharide. This is one thing. Okay, before going to cellulose, I will tell you starch. Already we have understood starch is a storage polysaccharide. Okay. So starch is also a polymer of glucose. So this is another point we need to learn. So polymer of glucose means here also glucose like this. They are going to make a bond like this. Okay. But here in the starch, one speciality is it appear in two forms. What are those? One is amylose and another one is amylopectin. Very simple structure. Amylose says, I am very simple, ma'am. I am very simple. I am just having a glucose like this. I am a very simple straight chain. Just construct me with the glycosidic bond. Over. My structure is over. And amylopectin says, I am very difficult. I am very complicated. I am not as simple like you. Even though you make a bond in this way, but I will show branches. Look at here. Again, I am going to show branches. Somewhat just like a glycogen. Okay. Then it got angry. Amylose got angry. My name is also small compared to amylopectin. My structure is also simple compared to amylopectin. So why should be I like a straight chain? So I am going to twist upon myself. Okay. So that I can be like this. I will form in this way. This is called helical structure. Secondary helical structure it is. What is secondary structure? That we'll learn in the protein part. But this is a helical structure. So here what happens? In this helix, it is going to hold iodine molecule in this. So iodine molecules are going to present in this helix. Because of this purpose only, when you just make a starch test, so you might have done in your lower classes, when you just put an iodine solution on a leaf or any starch, it turns into blue color, right? So how come it happened? Why it has turned into blue color by adding iodine solution? This is because it is going to have an iodine molecule in their helix. Okay, so this indicates the starch is present in this food. Understood? So simple structure, no need to learn how these helical has formed, how these amylopectin is forming. Just you know, it is a polymer of glucose. One is amylose, another one is amylopectin. That's it. Now, when you come back to cellulose is a structural polysaccharide. Okay, it is not like a starch. And here also, it is a polymer of glucose only. So all these are glucose. Glucose. That's it. But here, no iodine present. Here, no iodine present, no blue color. That's it. That's about the cellulose. Okay. And the next is inulin. Inulin is a homopolymer. Just remember, it is a polymer of fructose. So when you start getting a polymer in this way, till now we have learned the polymer of glucose only. But inulin is the only product which is having the fructose. So it started making the bond, that is a glycosidic bond, and make a inulin structure. How the structure has formed, which are the bond formation, which are the carbon atom invert, nothing to worry. Don't worry about the structure. You just know it is a homopolymer of fructose. That's it. Okay. It is present in root of Dahlia plant. And the next thing is about the chitin. So chitin already have told that chitin is a homopolymer. In earlier textbook, before the revised textbook, in few textbook, they have mentioned it as a heteropolymer. But later it has been corrected, it has been modified, and finally we got a clear answer that it is a homopolymer. Just make a note of this also, don't make confused. Don't confuse it as a heteropolymer, it is a homopolymer. Okay. So it is a repetition of NAG, N-acetylglucosamine. N-acetylglucosamine, NAG. They are joined by, again, glycosidic bond. Where you are going to find this chitin? Of course, in the fungal cell wall as well as exoskeleton of arthropod. That is, we are learning from this biological classification part itself, and even in animal kingdom also. Okay. So over everything we have completed. Now this is a NCERT image. You can see the glycogen structure here they have shown. Look at here. This is the straight chain. And here they have shown the branches. They are showing the branches. Can you see? And here, the straight chain structure they have shown in this way. This is 1,4 here glycosidic bond. 1,4 glycosidic bond they have managed. So this is nothing but alpha 1,4. This is alpha 1,4 glycosidic linkage. And now here the branch portion has been enlarged. So here the straight chain alpha 1,4. And when you just look at here, this is the first carbon atom. And here the 6th carbon atom has been utilized. So therefore here 1,6 glycosidic linkage bond has been showing. That's it. Very simple structure. If such question comes to identify in the exam, then you can write this is the 1,6 and this is the 1,4. It is a glycogen structure. Okay. Now let's read NCERT part. Let us understand if we have left anything. Yes. Polysaccharides. The acid insoluble pellet also has polysaccharide, that is nothing but the carbohydrate, as another class of macromolecule. So already I told in the beginning itself. Polysaccharides are the long chain of sugar. They are threads. See, they are the threads, literally cotton thread. Polysaccharides are like a cotton thread containing different monosaccharide as building block. That means it could be homopolymer, it could be heteropolymer. For example, cellulose is a polymeric polysaccharide consisting of only one type of monosaccharide. It means cellulose is a homopolymer. So which monomer is that? Glucose. Cellulose, again the word has mentioned homopolymer. Now the next one is starch. Starch is a variant of this but present as a storehouse of energy in plant tissue. Animals have another variant called glycogen. Again it is the stored polymer. Inulin is a polymer of fructose. It is important line. Okay, many students make confusion in this one. In a polysaccharide chain, say for example glycogen, see once again this is important. Look at here. The right end is called reducing end. See, what they are trying to tell is, imagine this is the glycogen. Okay, so here so many glucose molecules are present. Suppose if I consider this as a right hand and this as a left hand. Okay, so they are telling the right hand is called reducing end and the left hand is called non-reducing. Now, don't bother about what is reducing, what is non-reducing, that you are going to learn in chemistry. Okay, you just know R for right, R for reducing. You can remember. Then obviously, if it is not reducing, then obviously other side would be non-reducing. Okay. Yes. In a polysaccharide chain, for example, glycogen, the right end is called reducing end and the left end is called non-reducing end. Important. It has branches as shown in the form of a cartoon. That means just now have shown the image now in the figure 9.2. Starch forms a helical secondary structure. Important. In fact, starch can hold iodine molecules in the helical portion. That means these are the helical. They could hold the iodine in them. The starch iodine is blue color. Cellulose does not contain complex helixes and hence cannot hold iodine. That's it. Over. Then, plant cell wall are made up of cellulose. Paper made from plant pulp and cotton fiber is nothing but cellulosic because it's a cell wall only. There are more complex polysaccharides in nature. They have building blocks, amino sugars and chemically modified sugar. For example, glucosamine, N-acetyl glucosamine, etc. Exoskeleton of arthropods, for example, have a complex polysaccharide called chitin. These complex polysaccharides are mostly homopolymer. Okay, it has been clearly mentioned. Now let's solve few MCQ. Let's check our knowledge, what we have learned till now. Okay, which of the following is a polysaccharide? Tell me. Which of the following is a polysaccharide? Sucrose is a disaccharide. Lactose, disaccharide. Glycogen is a polysaccharide. It is a glucose molecule joined together and form a polymer. Glucose, it is a monosaccharide. Next question. Which of the following disaccharide will give 2 molecules of glucose? That means glucose plus glucose is equal to what? Maltose. So obviously the answer is maltose. What is sucrose? Glucose plus fructose. It's not answer. Lactose, glucose plus galactose. None of these is not an answer. Next one. Amylose and amylopectin chains occur in starch. Okay, not glycogen, not cellulose, not chitin. So if you have understood clearly, you can answer before me itself. Cellulose and starch is a homopolymer of. So till now we have learnt homopolymers only of glucose. Only one is, what is that? Homopolymer of fructose. That is nothing but inulin. And after that, the complex sugar, N-acetyl glucosamine, all those things, that is in chitin part. That's it. So no more complexity. Just learn these basic things. You can solve any MCQ. So cellulose and starch are a homopolymer of this glucose only. Not fructose, galactose and none. Next question. Which of the following statement is incorrect? Starch is a polysaccharide, homopolymer. Correct. Inulin is a polymer of fructose. Correct. In a polysaccharide chain, left end is reducing. This is wrong. Left end should be non-reducing. While right end should be non-reducing is given. It should be reducing. Therefore, this is a wrong answer and this is our answer even. Because we need to give an incorrect answer. Starch form helical secondary structure. This is also right. So, option C is incorrect. Paper made from plant pulp and cotton fibers are starch, cellulose, complex polysaccharide, both B and C. So it is mainly cellulose or it could be a complex polysaccharide. Starch is stored food molecule. Okay, so therefore this is our answer. Okay, that's it. So hope I am very clear about the structures of these sugars. There is nothing but a carbohydrate. Now let's move on to lipid part. Okay, the next topic is lipids. Already we have learnt lipid, though it is a micromolecule, because of its nature of vesicle formation, it comes in an acid insoluble form. That is in a retentate, right? So let me write that name. It is a part of, though it is a biomicromolecule. Okay, what I am telling? It is a part of acid insoluble pool. That is a written date, though it is a biomicromolecule. So what is the weight? Having 80 into 800 Dalton. Why? Already we have understood the reason for this. It is going to show a vesicle-like structure when it's get broken into small pieces when you start grinding it. Correct? So now in this part of lipid, actually in our NCERT only just one paragraph is given. So before starting this, let me tell you one more important thing. See, if you have a textbook along with you and if you are watching this video, I know definitely you are getting confused. Ma'am, actually you are not going along with the textbook. I mean, in a series, what it is given in NCERT. Why? This is because I need to connect the dots. Biomolecule is a difficult chapter for many students. Why? Because here the concepts are abruptly given. All informations are given, but it is somehow abruptly. For example, amino acids as well as the lipid, carbohydrate, so their bond formation, everything has been given in a beginning. And at the end, they started introducing about the carbohydrate, about the nucleic acid, at last about the protein and enzymes. So many students, they are unable to connect the dots with these concepts. So that's why if you follow what I am going to teach, in this way if you follow the NCERT, then you will understand each and every line and every word of NCERT textbook. So finally, what is our aim? We need to understand each and every line, okay, of our NCERT. And at the same time, we need to solve all the MCQ. It may be a difficult part, it could be a applied level questions, whatever, we should be able to solve all such MCQ. In that way, we need to understand the concept. So that is what our main aim, and we are doing that also. So you just follow the video, and after every topic, anyhow, I am reading NCERT only. So you just open that part of NCERT textbook and you can follow it. So if you have a doubt, suppose ma'am have left any part here while doing. So if you have such doubt, you can tick also. You can make a tick mark. So the part that I have finished, and later you can, finally you can analyze all the part that I have covered in this chapter. Okay? Yes. What are lipids actually? Lipids, we know that it hate water, and lipids are present in the acid insoluble pool, though it is a micromolecule. Now within the lipid, we can make a classification. What are those? Three classifications are: one is simple lipid, the second one is conjugated lipid, the third one is derived lipid. Okay, so on which basis we have classified? Based on the nature. But in NCERT, the classification has not given. Simply they have given a name like glycerol is a simple lipid, fatty acid is a simple lipid. But if the question comes, you will get confused. That's why I made a partition like this. Okay, so even in your copybook also, you just write as it is, how I am going to write it on the board. Okay, yes, simple lipid means the name itself is telling that they are very simple in nature. So you cannot further break down into further simplest form. Because if you break the simple lipid in a further simplest form, first of all, it won't act as a lipid only. Okay, so that's why we can say this is the ultimate simple form of a lipid. That is nothing but simple lipid. So to this lipid, if you add some other part or some other derivative, it will become a conjugated lipid. Okay, so like that we can make a classification. First, let us focus on this simple lipid. In a simple lipid, again we could able to get three parts. That means three groups. One is, we need to learn about the glycerol. Glycerol is a simple lipid. That is one thing. Here, we need to learn just a structure. That's it. And after that, we need to know that fatty acid is also a simple lipid. What is fatty acid? How it looks? That also we'll learn. And the third category within simple lipid is glycerides. What are glycerides? Actually, the funny part is, glycerides are nothing but it is a combination of these two. It means, glyceride means glycerol plus fatty acid. That's it. When this glycerol combined with this fatty acid with the help of a bond that we call it as an ester bond, then we make a structure called glyceride. So let us learn all those things one by one. Very simple. Let us talk about glycerol first. Glycerol structure is given even in our NCERT book also. It is very important. We need to learn it. Okay. So glycerol is called trihydroxypropane. Tri means what? 3. Okay. So here it says I have 3 carbon atoms. Just put 3 carbon atoms. Just connect it. Okay. And everywhere, you just add OH. If it is having OH, obviously other side would be H. So we need to balance the valency, right? So therefore, write H here also. 4 valency we have to satisfy. Then OH here, H here, OH here, H here. That's it. This is nothing but, okay, one more H. So this is nothing but what? It is a glycerol structure. It is a kind of alcohol. So it is also called trihydroxypropane. Why trihydroxy? Because 3 hydroxyl group is present. Okay. So in our NCERT, they have just given in this way. CH2OH. CH2OH. Again CH2OH. You can write in this way also or in this way also. No problem. Because here also see CHH. So H2. Obviously OH is there. Here also have written OH. So again CH2. Why 2? Because this is balancing one more valency, right? So therefore CH2OH. So here also CH2OH. Okay. So this is what the simple structure. Okay. So glycerol part is over. Then the second one is fatty acid. What are fatty acid? I will tell you. It's a long chain of carbon. Okay. See, let me draw carbon chain like this. Let me draw carbon chain like this. To this carbon chain, of course, there will be H will be there everywhere. So to this carbon chain, if you add carboxyl group means COOH is a carboxyl group. Then it will become a fatty acid. That's it. It is a simple lipid. So here there is how much carbon chain we have to write, man. How much carbon atom we have to write, man. So you can write just 1 carbon atom along with 3 hydrogen atom, that is CH3. So this will become a methyl group. So this is the structure of methyl. You are very much aware about chemistry than me. I know it. So these things are very silly for you guys. And if you can put C2 as well as 5 hydrogen molecule, I will show you here. 1 carbon. To this 1, 2, 3. Okay. So here you just add COOH. So this is also fatty acid. Suppose if you write 2 carbon atom, then what happened? 1 carbon atom, 2 carbon atom, obviously 1 hydrogen, 2 hydrogen, 3 hydrogen, 4 hydrogen, 5 hydrogen. To this, you can add carboxyl group, that is COOH. This is also a fatty acid. So like that, you can add from 1 to 19 fatty acids. But here we cannot give a name for everything and we cannot keep writing carbon like this. No. So that's why what we are going to do. So to this carbon chain, we are just going to give a name called R. So R is nothing but anything. How many carbon atoms you want to put in a particular acid, you can add. So here, a simple structure or a general formula we can write for this fatty acid is RCOOH. So R can be anything. R can be CH3 group. This R can be C2H5 group. This R can be, it's a multiple of C2H2 group. So like that it can be keep multiplying like this. Okay, not C2H2, CH2. Multiple of CH2 like this. Understood? So here in our NCERT, 2 fatty acid examples are given. We need to learn that also. What are those fatty acids? Hope you can understand. So one example about the palmitic acid. Write down in your copy. So this is very important in NEET point of view. So palmitic acid is having 16 carbon atom including carboxyl group. So in that case, how we are going to write? You cannot keep writing 16 carbon atoms, right, in front of, that means before COOH. So that's why we made a plan. What plan we did? Very simple. Just put CH3. Okay. Just put CH2 and COOH. So this is carboxyl group. It must be there. Ma'am, you can add just R. No. Yes, R is a anything. It is a general thing. We have just given a letter as R. But here, particularly they have mentioned 16 carbon atom for this palmitic acid. So that's why CH3 if you write, and after that in the middle, if you write CH2 and just put a bracket and write 14 to this. What it indicates? It is a multiple of CH2. 14 times. It indicates that you are writing 14 times CH2. Then obviously it will satisfy 16 carbon atom, right? So here 1, here 14, it becomes 15, and here 1, 16. So this is a simple structure of palmitic acid. So like that, one more example is given. Arachnidic acid. So this arachnidic acid is having 20 carbon atom. So the same structure you can write, but here the multiple of not 14. So you have to change the number. How much you are going to write? Write along with me. You just write CH3 as it is, write CH2 as it is, and COOH. Now, to this CH2, you have to add what? 18. Because 18 plus 1, 19 plus 1, 20. So total 20 carbon atoms has become satisfied. Okay. So this is what the structure of arachnidic acid. So I will show you everything. It is actually given in our NCERT. Wait, I will show here only. Yes, look at here. This is palmitic acid. So open your textbook. If you are unable to see this, here it is given in NCERT textbook only. CH3, CH2, multiple of 14, and COOH. It is palmitic acid. And arachnidic acid is not given, but it is understood. Even if in your exam it comes, you should be able to answer it. It is nothing but multiple of CH2 18 times. And rest of the things as it is. And glycerol structure, can you see? CH2OH, CHOH, CH2OH. Okay, here CHOH. I wrote CH2OH. Okay. Yes, here it is just CHOH. Done. Right? So, we have understood fatty acid also. Now the third one is, before going to glyceride, within fatty acid, we are going to get two categories. One is saturated fatty acid, another one is unsaturated fatty acid. Saturated fatty acid means what? You just keep writing the carbon atoms like this. Okay? So, between the carbon atom, only single bond is there. No double bond at all. Then such fatty acids are called what? Saturated. They do not have any double bond between the carbon atoms. Whereas in the case of unsaturated fatty acid, of course, carbon atoms are there. It can be a single bond or it can be a double bond. So, double bond can repeat at any time. Okay? So, it can repeat multiple times or it can be even just one. But double bond is there between the carbon atom. So, such fatty acids are called unsaturated fatty acids with one or more double bond. Here, without double bond. Again, it is also important. Based on presence and absence of double bond, we can classify into saturated and unsaturated fatty acid. So, glycerol part is over. Fatty acid part is over. Then the third one within the simple lipid is what? Glyceride. So, glyceride is nothing but it is a glycerol plus fatty acid become a glyceride. Okay. So, here what they are telling, to this glycerol group means already we have understood the structure. C. So, this is the structure of glycerol, right? So, to this glycerol structure, we need to add fatty acid. Fatty acid means COOH, correct? But here exactly which fatty acid name? It is not mentioned. So, therefore, what we need to do is just place R here. Just place R. Let me take other color. Just place R. In front of this R, just write COOH. Now, these two molecules came near, but they have to react. So, for the reaction purpose, one water molecule has to remove, just like while joining to glucose molecule, how a water molecule has removed, that is in a dehydration reaction. So, that glycosidic bond has formed now in a carbohydrate. In the same way, here also one water molecule will get removed, so that here a bond has been formed. But this bond is not a glycosidic bond. Here, this bond we are going to call as a ester bond. So, how we are going to remove the water molecule? Look at here carefully. So, here from the glycerol part, we will just take only one H. And from the fatty acid part, we will take OH. So, if you remove H and here one H, it becomes 2H. and one water molecule, one oxygen, so that water molecule has got removed from this place. So as a result of this, here in this place, we could able to see a bond. So how it will look after a bond formation, that we'll see. So if two fatty acid react with the glycerol, then we call it as a single fatty acid react with the glycerol, we call it as a monoglyceride. If two fatty acid reacts with the glycerol, it is known as diglyceride. If three fatty acids react with the glycerol, it is called triglyceride. I will show you. I will write here. So monoglyceride means what? First, let me write the glycerol structure here. So obviously in front of this, we need to write OH. Okay. So now it is reacting with what? It is reacting with fatty acid. So R, C, O, H. So instead of writing this, I can even write in this way. Let me rub this. I will show you. The same RCOOH, I can write in this way also. R as it is. Then C, double bond O, and here OH. Understood? Now, just recall what we did in the previous slide. Just remove H as well as OH from this place, so that water molecule will be released out. Okay, it will be get removed. Let me rub this part. Just write down in your copy, or else you will get confused. Definitely you are going to get confused. So this is the most difficult part in biomolecules. You can understand the concept. While writing the reactions or while writing the structure, many students make a mistake. That's why they hate this chapter. But if you know the logic, if you know how these things are happening, then easily you can remember and you can write also. Okay. So first you write down as it is. Then in the next part, you just remove this water molecule. Then you write how a bond has been formed. Then you will understand while writing, oh, here OH was there, here H was there. So that after removal of water molecule only, the ester bond has formed. Okay. So now let me remove this here. So here one OH was there, no? Here one O was there. So let me write the same O like this. Now this is the structure of monoglyceride. Only one fatty acid has involved here with the glycerol. Okay. So like that, I can even write diglyceride. So write down again. Just make practice along with me. Okay. Now we need to add two fatty acid, right? So again, you can use the same formula. R, C, double bond, OH. Again, R, C, double bond, OH. But here, these two fatty acids are different, right? So that's why we can mention it as R1 as well as R2. Right? Now, how we are going to make a bond? In the same way. How we did here, in the same way, we need to apply our knowledge here also for making the ester bond. Let me remove this part. So what happened? Just write O here. That's all. Now it becomes diglyceride. So here the bond, what you are going to see is ester bond. Okay. So like that. Just put 3 fatty acids with the glycerol, then it becomes triglyceride. Write down in your copy along with me only. So this is glycerol structure. Now let me add fatty acid here. R1, C double bond O, OH. Again R2, C double bond O, OH. Why I put double bond here? Because to balance the valency, COOH. Again R3, 3 fatty acids we are placing. Okay. So now, if water molecule has been removed from each fatty acid while reacting with the glycerol, then obviously 3 ester bond will form here. 3 ester bond formation occur. So like this. So this is triglyceride. All fats, all oils that we are going to see in the market, they are nothing but the triglyceride. Write down in your copy. It is also very important. All fats and oils are triglyceride. So the best example for oil is gingerly oil. Okay, and all fats, the ghee, butter, everything, they comes under the triglyceride. Okay, yes. Hope you have understood about the structure. Now in the triglyceride, we are going to see fat and oil. Fat is having higher melting point, whereas oil is having lower melting point. So that's why even the oil remains oil only in the winter season also, because of its lower melting point. Again, these 2 points are important for your NEET point of view. Everything is important for NEET point of view, but few things are very, very important. So here the example is gingerly oil. Understood? So simple lipid part is over. We have learned the glycerol structure, we have learned the fatty acid, we even learned about the glycerides. Now the second part of the lipid is nothing but conjugated lipid. Conjugated lipid means what? Imagine this is a lipid. To this lipid, I am going to add one molecule. Till now, we are just talking about a lipid. If you are a lipid, okay. Are you a glycerol? Okay, come this category. Are you a fatty acid? Go in this category. Are you a glyceride? Come in this category. But you guys are pure lipid till now. But now in the conjugated lipid, what happens? To this lipid, I am going to add a non-lipid part. So this is actually non-lipid part. I am going to join with this. Then these two molecules, when they join together, lipid and non-lipid part, it become a conjugated lipid. That's it. Okay. Conjugated lipid means what? Lipid plus non-lipid part. I will give you example. You can understand it better. Imagine I have a lipid here. So to this lipid, I had protein. I add protein to this. Then such lipids are called what? Lipoprotein. So it is a conjugated lipid because along with the lipid, protein molecule has got added. So like that, imagine to this lipid, if I had carbohydrate, then the structure can be called as glycolipid. Glyco means carbohydrate. So carbohydrate group added to the lipid called glycolipid. So like that, to this lipid if I add phosphate, can you name? Yes, you are right. It is called phospholipid. So phospholipid structure we need to learn in this chapter because it is given in NCERT, that's why. So phospholipid, the best example for phospholipid given in NCERT is nothing but lecithin. Lecithin. Just recall cell chapter. In cell chapter, we have learned that a cell membrane. Cell membrane. Even in this chapter also I told, no? This is the phospholipid. So this phospholipid is nothing but a lecithin. So this lipid structure we are learning now. Phospholipid structure. Okay? Very simple structure. Very simple. You need to follow 3 steps here. 3 steps. Listen to me carefully. It is given in NCERT. You can mug up this equation, that is structure as it is. But sometimes if you get confused, you can apply this knowledge. What I am trying to tell, please do follow this. Okay? So to construct the phospholipid, we need to follow 3 steps. The first step is, what is the first step? Take one glycerol. Take one glycerol. And to this glycerol, add 2 fatty acid. Then it becomes what? Diglyceride. So this step easily you can write. Okay, let me show that. Take glycerol. So this is the structure of glycerol. First let me write the glycerol structure. Okay, to this add 2 fatty acid means what? Like this. R, C double bond O, OH, C double bond O, R, OH, like this only, no? So this is R1, this would be R2, correct? Now, we need to add means we need to make a bond here. So how we are going to make a bond? Just remove one H from here, OH from here, OH, H. Okay, here, just to make a bond, I am removing this O also, but I will write in the middle. So now, it made a bond like this. That is the ester bond. This is the first step. Now it is easy. You can learn this. Now what you have to do in the second step? In the second step, same thing. Glycerol plus 2 fatty acid. To this, you should add one more thing. What you need to add here? Phosphoric acid. Phosphoric acid. So, take glycerol to fatty acid as it is, already you have done. To this add phosphoric acid. Phosphoric acid structure is H3PO4. Correct? H3PO4. So, let me show you. Here you need to add this to the third carbon atom. So, H3PO4, I can write like this also, no? S3PO4. If I just split it, if I just arrange it in a different way, I can place P in the center. Correct? And place O in all the four corners. And 3 OH is there. Just place OH everywhere. And here to balance the valency, just put double bond here. So I can write like this also. Correct? So let me write here in this way. Here P, OH, double bond O. Here also OH and here also OH. But it has to make a bond here, right? Okay, let me put a bond. So once again, you have to do the same thing. What the thing is? Take OH from here, H from here, so that it has to make a ester bond. So let me rub this part. Look at here carefully. Look at here carefully. So here it was as it is. Now it made a bond here. Correct? So 2 steps over. I added phosphoric acid to the third carbon atom. Now to this phosphoric acid. To this phosphoric acid, you should add choline group. So that is the third step. So what is the third step? Glycerol plus 2 fatty acid. 2 fatty acid plus H3PO4 phosphoric acid. Now the fourth molecule here you need to add is choline group. So this choline group structure is something like this. Very simple. Just look at here. You can understand. CH3, CH2. Okay. To this write N. CH3, CH2. To this you should write N. Now this N is having 3 CH3 molecule. CH3, CH3, CH3. Practice it twice or thrice. It will come. CH3, CH2. It will be joined with N. So that to this N, 3 methyl group has been added. Okay. Now one of the hydrogen molecule from CH3 will react with this phosphoric acid. Okay. Shall I write here? Let me write the choline molecule here. So CH3, CH2, N, CH3, CH3, CH3. Okay, this is separate. Now, this H, one of the H from CH3, they are going to react with this. So, as a result, O will come in the center and here it becomes CH2 again. So, CH2 will be as it is. 3 CH3 at the bottom. So, this is the structure of what? Phospholipid. Phospholipid. Okay. Shall we see the NCERT diagram? How it has been given? Yes. Look at here. Open your textbook. See CH2, O, CO, R1 as it is. Here also, see, here in the second carbon atom, they have made in this side. Okay, so here there is no space, that's why they have written it at the left. So don't bother about this. Either it is a right or left, it should be in the same way. So here also CO, R2. And now, in the third place, look at here, CH2 is there. To this O, P, double bond O, that is phosphoric acid has been added. To this choline group, CH2, CH2N, CH3, CH3, CH3. So that's it. So this is the structure of phospholipid. Okay, now the last one is derived lipid. Let's see that. So derived lipid, I won't tell much information because the structure already given in NCERT. Just learn this cholesterol structure. That's it. Okay. Yes, this part over. Derived lipid means it is not a glyceride. Yes, we do agree. But it have a water repelling property. Example, cholesterol. It hate water even. It also repel water. See, there are so many hormones present in our body. They are actually the modification of cholesterol only. Like testosterone. It's a steroidal hormone. It's a modification of cholesterol structure. And even in a female body, that is the estrogen, progesterone, so many hormones are there. So here, the cholesterol structure, you need to learn from the NCERT because it is simply like a box. Look at here. Like this they are given, no? Let me construct this box. So one hexagonal box like this. Now one more. Now above this one more box. Okay. So here just put one line along with this. Here also add one OH group. So after this, just 5, 1, 2, see 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Not 6. And after that from here you just take 1, 2, 3, 4. So just put a box and something like this. Okay, so this is the structure of cholesterol. It is very, very important. Ma'am, it is not coming. If you practice it, definitely it will come. There is no such tricks to make you understand. Because it is a simple structure, only the thing is practice required. Okay, do practice, it will come. So hereby the lipid part is over. Now let's solve few MCQ, and before that let's read NCERT part also. Lipids are generally water insoluble. Already we know that. There could be simple fatty acid. That means in a simple lipid, fatty acid came here. A fatty acid has a carboxyl group attached to an R group. That means general structure they are telling. The R group could be methyl group, that is CH3. It could be ethyl group, C2H5, or higher of CH2. That means multiple of CH2. That means 1 to 19 carbons can come under this category. For example, palmitic acid has a 16 carbon, including carboxyl group. Arachidonic acid, 20 carbon atoms, including the carboxyl group. Okay, so the fatty acid. So hereby they are explaining the structure, and the next thing is about the saturated and unsaturated fatty acid. So what they are telling, the saturated fatty acid without double bond, unsaturated fatty acids with one or more double bond between the carbon atoms. Then, so here they are talking about the fatty acid, and the next part is they are talking about the glycerol. So this is also simple lipid. So another simple lipid is glycerol, which is a trihydroxypropane. So many lipid have both, so only one line they have given information about this glycerol. And the next thing is they are talking about the glyceride. So lipids have both glycerol, let me take other color, both glycerol and fatty acid. Here they are, fatty acids are esterified with the glycerol. That means they are talking about the glyceride. So they can be monoglyceride, diglyceride and triglyceride. Okay, so all these are nothing but simple lipids only. These are also called fats and oils based on the melting point. I told no. So oil have lower melting point. Example, gingerly oil. Remember example. And hence they remain as oil in winter also. Can you identify the fat from the market? So we can take many things. Some lipids have phosphorus. So here they are talking about the phospholipid. That is nothing but conjugated lipid. So they have a phosphorus and a phosphorylated organic compound, important word in them. So these are phospholipids. They are found in the cell membrane, again important. So lecithin is an example. So just now I told, some tissues, especially the neural tissue, have lipids with more complex structure. So this can once again a statement based question. It can be framed like that. Okay. So structures already have explained. Now let's solve few MCQ. It's very simple. You can easily tell the answer. Lipids are generally dash insoluble. Generally, they are water insoluble. Correct? So, of course, they can soluble in fat. Lipid itself is a fat, not all. Next question. All questions are directly picked from the NCERT line. Okay? So, lipids could be dash fatty acid. It could be simple fatty acid. Or has a carboxyl group attached to an R group. Correct? So, it could be a simple fatty acid. Carboxyl group is attached. So here all this option cannot be the right one. So if you just look at here, you can see lipids could be a carboxyl fatty acid. We won't say carboxyl fatty acid. A lipid Could be a fatty acid. We won't repeat the fatty acid. So lipid could be a carboxyl. Again, there is no such lipid, carboxyl fatty acid lipid. So therefore, it should be the option 4. Next question. The R group attached to the carboxyl group in a lipid could be. It could be methyl, of course, yes. It could be ethyl group, of course, yes. Higher number of CH2, just now we read it. So therefore, all of the above is the answer. Next one. Palmitic acid has dash number of carbon including carboxyl. Palm. Usually I remember palm means, this is palm only, no? So palm is having 5 fingers. So usually I remember 15 for palmitic acid. Along with carboxyl means plus 1. So obviously it will become 16. So here the answer is 16. Next question. Arachidonic acid has, if it is not 16, then obviously another one is 20 only. So therefore the 20. Ma'am, suppose if it some other fatty acid name is given, it won't come. Because whatever the information given in NCERT, the questions will be framed from that line only, from that part only. It may present in the image part, it may present in the summary part, it may present even in your manual part. But the questions will be picked directly from the NCERT. So in many chapters, while doing concept, while explaining the concept, I told where such thing has been appeared. Because, for example, I will give, in a human reproduction, while reading, we will come to know that the testis has an outer covering. So that outer covering is called tunica albuginea. So that word is not given in NCERT, tunica albuginea. But if the question comes in your NEET exam, you will get panic that, oh, it is not given in NCERT, it is out of the syllabus. But my dear student, it is there in our syllabus. But where is that? It is not in our textbook, but it is in manual. So that's why while reading textbook, while reading manual, while reading summary, while analyzing the diagram, we should be extra careful. We should know how the questions will be framed. So if you follow all the videos, what I did in my channel, you will get a clear idea how the questions will be framed and what are the important lines here. Okay, just an example I took. So here also, which is given in NCERT only, the questions will be picked from that example itself. Okay. Next one. Fatty acid could be dash without double bond. If there is no, if there is a double bond, we call it as unsaturated. Where is unsaturated? Here it is. Dash without double bond. If there is no double bond, only simply carbon-carbon like this, it is called saturated. So here it is saturated. So obviously this is our answer. But here first unsaturated given. Here also saturated given. But here both the cases showing unsaturated. So therefore option B is the answer. Next one. How many of the following is an esterified glycerol? So monoglyceride is having ester bond. Diglyceride is having ester bond. Triglyceride is having ester bond. But muramic acid, it is an acid. Here we are not going to say ester bond. Even lignin, suberin, that's just a material. They do not show ester bond here. So therefore 3 are showing esterification. The oil have lower melting point. So is it show all fats? No. Triglyceride, in the triglyceride fat also come, oils also come. But fat become so harder in the winter season. If you just observe the ghee, it will become harder, right? So that's why it will not come under this oil group. So gingerly oil, we can take the exam. Next one, a phospholipid have phosphorus group. Yes, it is even having a phosphorylated group. That means both the options are right. Okay, that's it. Hope I am very clear about the lipid part. So the structure is also very crystal clear. I think if you practice it, definitely it will come. Both carbohydrate part structure as well as the lipid part structure. Now, one more important thing, the most important part, that is amino acid. Okay, let's learn about this. Okay, now let's talk about amino acids. Once again, these are also belongs to what? Acid soluble category. Because their molecular weight is between 80 into 800 daltons. Correct? Of course, this is also one of the organic compound. Why it is organic compound? Of course, it is also having what? carbon atom in it. So, all carbon containing compounds are nothing but organic compound. So, this amino acid is also having carbon in it. Okay. So, now, if I just consider this as an amino acid, let's assume this is one amino acid. Okay. Let me write A. A means amino acid. Then, this is a smaller subunit. That means it is a subunit. I can consider it as a monomer. Okay, so this is one monomer. Monomer means nothing but subunit. Now, I will take one more amino acid. Again, this is a monomer. I will take one more amino acid. Again, it is a monomer. One more amino acid. Again, it is a monomer. Okay. So, like that, I can take plenty of amino acid. So, all these single amino acids are nothing but a monomer. Now, if I want to join these small amino acid, let me make it as amino acid. Name can be of anything. Let us just simply consider in general as amino acid. Okay. Now, if I join together like this, look at here, I am joining like this with the help of a bond. So, this bond is called peptide bond. It is very important for your NEET point of view. Now, it becomes what? By having many monomers together, now this entire structure is considered as a polymer, right? So, this polymer is nothing but, it's a polypeptide. It's a polypeptide chain. Why polypeptide chain, my dears? Because it is having many peptide bonds in it. These peptide bond has formed by combining different amino acids. Okay, so this polypeptide chain or a polypeptide is nothing but a protein. Now, if I take this protein molecule, that is nothing but a polypeptide chain, if I just crush it in an acid soluble, does it melt there? No, because each single amino acid will be having 18 to 800 dalton. So like that, when they join together, obviously their weight will be more than 1000 dalton. Then obviously this portion, that is nothing but a polypeptide chain, being a macromolecule, now it is a part of retentate, correct? It is a retentate. But these are micromolecules. Understood? So amino acid means what? It is just a monomer of a protein. So what I want to tell is, amino acids are just a monomer of protein. These are the building blocks of protein. Just like how we are going to take a brick for a construction of a wall. Every time I am taking the same example to make you understand. Okay. So like that, this is also smaller building blocks. Okay. Now these amino acids, as I said, these are monomer of protein. There will be 20 amino acids that make protein. Though in our body there are a lot of amino acids are there, but for making a peptide chain, imagine I am making a peptide chain like this. These are the amino acids, the circular part what I am drawing here. So here I could found only 20 amino acids, only 20 amino acids. So these 20 amino acids, they may bound in a different sequence, but their number may vary, their sequence may vary, but to get a protein, only these 20 amino acids I am going to get. For example, listen here. Let me take an example of protein 1. It could be of any protein. Let's consider this is protein number 1. Now, let me show like this. This is amino acid A, one more amino acid B, amino acid C, amino acid D. Like that, if I make the sequence of amino acid, which is around 30,000. That means in this polypeptide chain, I could able to get 30,000 amino acids, which are joined by a polypeptide chain. But they are nothing but these 1 to 20 amino acids only. 1 to 20 amino acids. So let me change this number. Instead of A, B, C, D, 1 second, I will make you understand. Let me consider 1, 2, 3, 4, 5, 6 like that. Let me consider this as protein A. Okay. So let me make a 6, 7, 8 like that. Suppose if I take another example, imagine there is one more protein called B. So in this protein, let's assume there are around 10,000 amino acids are there. So in that also, the same amino acids I am able to get only 1 to 20, but their sequence can be varied. Imagine here 1, 2, 3, 4, 5, 6, 7, 8, like that they have arranged. But here it could be 1, 2, 3, 3, 3, 4. Are you getting? Are you getting? So let me take one more example. Imagine there is a protein called C. It might be having amino acid around 50,000. 50,000. Then also, I am going to get these amino acid which are between 1 to 20 only. But again their sequence, that means their arrangement pattern will be different. They have been connected by this peptide bond. Now here immediately 8 may come, 6 may come, 7 may come, 1, 2, 3, like that they may get arranged. So I am just giving a number 1, 2, 3, 4 like that. So amino acid name will be varies. So there are different types of amino acid. If I just make a list of these 20 amino acid, I can make a list like a valine, glycine, serine, tryptophan, tyrosine. So these are the amino acid name. But here I can't write full name of these amino acid. No, just to make you understand, I am writing this number. So hope I am very clear. Hope I am very clear. Okay. So what did you learn till now? Amino acids are nothing but a monomer of a protein. So these amino acids are 20. These 20 amino acids, their number may vary. But for making a protein or a polypeptide, so these 20 amino acids only present, but their number may vary. For example, if amino acid tyrosine, not tyrosine, let me take one more amino acid, for example, glycine. So the same glycine may repeat for several times. Same alanine may repeat for several times. I am telling the name of amino acid. But they will be lie within 1 to 20. Understood? So now we have understood the meaning of these two sentences. Monomer of proteins as well as 20 amino acid make all the proteins. And proteins are always heteropolymer. In the case of carbohydrate, we learned that the polymer could be homopolymer like cellulose, starch. These are made up of same monomer like glucose, glucose, glucose. If it is inulin, fructose, fructose, fructose. So like that, if it is a monomer, they should consist similar. If it is a polymer, they should contain same kind of monomer. But here it is not like that. Here, if I say this is one amino acid, then this is totally different amino acid. This is different amino acid. This is different. Okay? But means same type of amino acid do not form a polypeptide chain. If I take any polypeptide chain, obviously there will be different amino acid will be present. Hope you are getting my point. It is very simple topic. Only the thing is I need your complete attention. First of all, this chapter itself is very easy. I don't know why students are going to consider it as a difficult chapter. But trust me, if you listen it carefully, if you concentrate properly, you will understand each and every line. Okay? So, one thing we have to fix that amino acids are different in a polypeptide chain. So, that's why it will be considered as heteropolymer. Just fix it in your mind. It is important line. It never be a homopolymer because we can't see a same amino acid in a polypeptide chain. Okay. So, these 3 points meaning we have understood. Now, okay, before going to alpha carbon, I will tell you the meaning of this. Then, what is the structure of amino acid, ma'am? If I want to write an amino acid, you said it is a carbon containing compound. Yes, correct. But what is the structure? So, if I want to write a carbohydrate, I can write it is a CHO. So, their number may vary. So, like that, if I have to write a structure of amino acid, what is the structure? So, that's why NCERT has mentioned a line. What is that? Amino acids are substituted methane. So, we know the structure of methane, right? So, methane is having what? It is having carbon in the center, correct? And here, the 4 valency has been satisfied by placing hydrogen on either side, correct? So, the 4 hydrogen molecule will be there on either side of the carbon. So, methane is nothing but CH4. This is what the structural formula as well as the molecular formula. Now, here NCERT is telling, if you know the structure of methane, no, easily you can write the structure of amino acid also. Because it is just a substitute of methane. Okay, substitute means what? Take this hydrogen, replace it by something else. Take this hydrogen, replace it something by else. Take this hydrogen, replace. Take this hydrogen, just replace. Then you are going to get a amino acid. So what substitute it has? Let's see. Here carbon is as it is. Look at here carefully. This is the basic structure. It is a common structure for all amino acid. Okay. Now, here, let me write these valencies first. Here hydrogen is there, no? Just remove this hydrogen from the methane and just replace COOH. What is COOH? COOH is nothing but carboxylic acid. It is a carboxyl group. It is nothing but it is an acid. So, acid group is present on one side. Okay. Now here, just remove this hydrogen. In this place, you just place NH2. NH2 is nothing but amino group. It is an amino group. This is actually basic in nature. So, 2 place we have substituted. Now here hydrogen, it will be remain as it is. You take any amino acid, this hydrogen will remain as it is. Now in this place, that is in the hydrogen which is present at the bottom, it can be replaced by any molecule. It can be replaced by methyl group. Or suppose if I do not want this methyl, it can be replaced by, okay, C2H5. If I do not want this also, it can be replaced by CH2 group. That means any group can replace here, depending on the kind of amino acid. Okay, so that's why what we are going to write here, simply because it's a general formula, it's a common structure, therefore we are just going to write it as R. So R is variable, it can be of any group. Okay, so that's why we said that always amino acid is a substitute of what? Methane. Okay, so here remember, whenever if you want to consider this structure as an amino acid, then always remember, to this carbon only, the carboxylic group as well as the amino group must attach. So this carbon, we consider it as what? It's a alpha carbon. Why it is alpha carbon? Because to this carbon only, carboxyl group as well as amino group will be attached. But here, ma'am, you said 4 hydrogen has been substituted, but here we have substituted only 3 hydrogen atom, no? Where is the 4th one? But NCERT has given like that, we need to follow NCERT. So 4 hydrogen atom has been substituted, you just stick on to that line only. Don't write in your exam that only 3 hydrogen in the methane has got substituted. Just don't do such nonsense thing. Just follow NCERT. Okay. Now let us move on to this alpha carbon. So here I am just going to write 2 amino acid structure. Actually, one is amino acid, another one is something else. Now you need to identify which is actually amino acid. Okay, let me write here. Simply I am writing. This is not a particular structure, simply I am writing just to make you understand. Now tell me, let us consider this as amino acid number 1. This is amino acid number 2. Which one will you consider as amino acid? Here I can't ask you the question because it is not an offline class, and even it is not even a regular class so that you can tell the answer in a chat. So I will only tell the answer. Okay? Okay, let me tell you. Hope you have guessed the right answer, because as already I told, no, it is a recorded class. Therefore, I can't see your answer also. I mean, what you are telling. Okay. So, if your answer that it is not an amino acid, then it is right. This is not an amino acid. If your answer is this one as an amino acid, then obviously this is the right. This is our amino acid. Why? Because whenever we talk about an amino acid, always remember to the alpha carbon. What is alpha carbon? A carbon which must have carboxyl group as well as amino group on either end. Okay? A carbon should have both carboxyl as well as the amino group attached to the same carbon atom. Then only it will be considered as amino acid. So here it is having carboxyl group as well as amino group. So let us not bother about this R group. It can be of any number. It can be of any structure, any formula. But here, one hydrogen is there. So that's why we have written it as R. So let us not bother about R. But this is a amino acid. Okay. But here when you just look at this structure, if you consider this as a carbon, alpha carbon, then COOH group is there. But on either side, there is one more carbon is there. That should not come here. to this alpha carbon only the NH2 has to attach. But here this has not done. Okay, so that's why, but rest of the thing is there. Rest of the thing is there. Here hydrogen is there, here R group is there, everything is okay. But your alpha carbon is not having both amino as well as the carboxylic group, no, right? So that's why it is not an amino acid. Hope I'm very clear about this concept. Okay, now let's move on to one more topic within amino acid. That is nothing but neutral amino acid. What are neutral amino acid? So if an amino acid, if it is having same number of NH2 as well as the COOH group, then it will be considered as a neutral amino acid. The example is alanine, valine, glycine. So here I will draw the structure of alanine. So alanine structure I will draw. It is very important because it is given in our NCERT. Let me put a star mark here. Okay. What is the alanine structure? C as it is. Write down COOH. Obviously NH2 because it must be present in an amino acid. Then H on the top, and here just write methyl group. So this is methyl group. When methyl group is there, then this will be considered as what? It is a alanine structure. It is important. So valine structure is not given in NCERT. So let us not bother even about that. But glycine structure is given. Let us see that also. It is still more easier compared to this. COOH as it is. NH2 as it is. H is there. Now in the R place, we need to write something else, no? But here no need of bothering. Just write H. Very, very simple structure. So this is the structure of glycine. Write down in your copy. Both these structures are important because it is given in NCERT. Okay. Now the question is, okay ma'am, we have understood. This is neutral amino acid because here the same number of, here one carboxylic group is there, one amino group is there. So it is a neutral. So here also one carboxylic group is there, one amino group is there. Okay, fine. This is also neutral. Valence structure is not given. Even if it is present also, it must be having one amino group as well as a one carboxylic group. Now the question is why it is considered as neutral? Because it will do some magic. It will show a characteristic structure called Zwitterion structure. What is the meaning of Zwitterion? Before reading this, I will make you understand the meaning of Zwitterion. So let's imagine this is a tub. I have taken a tub. Okay. In this tub, just put some neutral medium. Okay. Neutral medium in the sense, it is neither a acidic medium nor even a basic medium. It is just like, it is a neutral water. Okay. It is a neutral medium. Now, in this neutral medium, you just put your neutral amino acid. What are neutral amino acid? That is what? Your alanine, valine, glycine. You put any amino acid. Okay. But here, just for reference, I will just write a common structure. Okay. I will write here as C, COOH. I will just write it as R. Either it can be your glycine or it could be your alanine. And NH2. And as usual, your H will be there. So here what we did? Here we have put neutral medium. Correct? So now, how they are going to behave in this neutral medium? Because you have added here neutral amino acid. Okay. Now here what happened? Whenever they get a chance, always H want to get removed from this side. H is like a boy. Okay. He is a boyfriend to this girl NH2. Let's imagine she is a girl. She is a girlfriend of this H. Okay. So whenever he get a time, whenever he get a chance, he just want to go and combine with her. Okay. Just you want to go and make a enjoy with her. So that's why he is just looking at the opportunity. He got such opportunity. So immediately what he did, he just escaped from this place. So if I just remove this H, this is nothing but hydrogen ion. No. If I remove this, shall I remove here? Hope you are writing in your copy. I will remove here. I am removing. I removed. Okay. Now what happened? Immediately, this COOH, after losing H, okay, immediately it will get a negative charge. It got what? A negative charge it got. Then, this NH2 is ready to accept this H+. She is just waiting for H+. Now she got accepted. So immediately what happened when it accepts this H+, To this NH2, one H if you add, then it will become what? This will become NH3. Okay, that is nothing but this NH3 shows the plus here. Are you getting my point? Are you getting my point? I will make you understand. If you take any molecule, for example, if I write a sulfate, okay, so I am writing it as anion. Anion. Even if I write phosphate, I am going to write it as anion because it is having negative charge. This phosphate molecule is having negative charge. This sulfate molecule is having negative charge. So like that, if I write copper, I will write plus because I write it is a cation because it is having positive charge. Like that, even if I write hydrogen also, I will write plus because it is also cation. Okay. Now this molecule which you had put in the neutral medium, no? This, by losing this H plus, become negative, that is anion. By accepting this H plus, become positive, become a cation. That means the same molecule showing both the negative charge as well as the positive charge. Okay. So such ions or such molecules which are present, which are having both the ions, we call it as zwitterion. Okay. So that is possible only by a neutral amino acid when you put into a neutral medium. Understood, no? If you have not understood, please rewind the video and just watch it again. Okay? So that is what the sentence I have given here. Zwitterion means positive as well as the negative charges coexist in a single molecule. They are present in the same molecule. It is the same molecule, right? Both are present. Okay? So this structure, we call it as a zwitterion structure. It is very important for your NEET point of view. Very, very important. Now, this neutral amino acid, it will do one more magic. What is one more magic? It will show one more special feature. That special feature, we call it as an amphoteric nature. So, what is this amphoteric nature? Again, I won't read this. First, I will make you understand the concept. What here? Once again, let's take two container here. Okay. So, now, in this container, just put acidic medium. Just put acidic medium. So there, what we did? We put neutral medium, right? We took neutral medium. There we put neutral amino acid. But here, you just take acidic medium. And in another container, you just take basic medium. Basic medium. Now let's see. Here some funny things is going to happen. Okay. Again, put the neutral amino acid. So neutral amino acid means the same structure we have to write. C, COOH, NH2, H and R, because R could be of anything. Here also write the same structure. Write along with me in your copy. Okay. Now, if I have to consider this as an acidic medium, then what happened? Remember always, acidic medium is considered as acidic because it will be having more hydrogen ion concentration. Okay. So that's why just by looking at this medium, if I see that, oh, do you have more hydrogen ion? Then you are acidic. Okay. Then obviously here also there will be more hydrogen ion is roaming. Just like Romeos, they are roaming here. And if I consider this as a basic ion, it will be having hydroxyl group, that is OH minus OH. OH group will be roaming here. Right. Now come to this case. Here usually what happened? The neutral amino acid, how it was behaving, whenever it was getting a chance, it is just got escaped from this COOH group. So that H group was just moving out from this and it was just combining with this. This is what the thing happening till now, right? But here, there is no such chance occur. Why no such chance? Because this H remain H only. H remain in the COOH group only. Because here this NH2, it was just waiting for boyfriend. Or it is just waiting for a person to get married. Let's assume it's not a boyfriend or a girlfriend. She's just waiting for a person to get married. But before him, if somebody else who is near to her, who is very near to her, just got a proposal. She got a proposal from this guy and immediately if she get married, then automatically NH2 becomes what? It becomes NH3. Okay, I will take one more color. I am just writing NH3 here. NH3. then she behaved like basic only. So what is the basic nature? What is the basic nature? By accepting one hydrogen ion, the amino group behaved like a base. Okay, so she did like this. She accept hydrogen ion, she behaved like a base. Okay, but not from this OH. Usually, when there was no any hydrogen ion in our surrounding, usually this H was going and combining here. But here, there are a lot of Romeos are roaming here. So she got a proposal from this guy and she got married immediately. So why she need to wait for this H plus ion? So he remained as it is. When he just moved from this place, then we could say that, oh, this is acidic. He behaved like an acidic. Acidic behavior means what? Removal of H plus or formation of hydrogen ion. But here no hydrogen ion is forming from this place. So you are no more acidic, man. You are no more acidic. But she is behaving like basic. Are you getting my point? So that's why if you put a neutral amino acid in an acidic medium, it behaves like what? It behaves like base, not like an acid. So that is one thing we need to remember. So like that, if you just put a neutral amino acid, say neutral amino acid, but in a basic medium, then what happened? Here, he removed because he got a chance, okay, nobody is there to compete with me. I will just come out this place. So nobody can stop me. So he just coming out, he become minus. So this COO got minus charge because it lost H plus here. H plus has just moved away. So now he want to go somewhere where near to this NH2, but immediately these girls who are just roaming here, no, coming from some other place, they just accepted this H plus. They are just telling, why you are just going towards her? We are here. Okay, you just get married with her. I mean, you just get married with me so that we will join to become H2O. H2O means here one H, another H become 2H and one oxygen become H2O. That means here by losing hydrogen ion, it started behaving like a base. Sorry, it started behaving like an acid. But here, it didn't accept any H plus, no. So that's why now it is not behaving like a base. Are you getting my point? If you accept H plus, simple. Remember it in a simple terms. If you accept H plus, you become acid. I mean, you become base. If you just lose H plus, you become acid. But here she accepted H plus. Now it behaved like a base. Here it just lost one H plus. That means hydrogen ion. So it behaved like an acid. But in which medium it is doing so? In basic medium. So that's why neutral amino acid, when you put it in a basic medium, they behave like acid. They behave like acid. Understood? So this special property which exhibited by neutral amino acid, we call it as what? Amphoteric in nature. Ampho means both. So we used to call amphibian, right? So which live in both water as well as land. So like that here, these amino acids are called amphoteric nature of amino acid. So in acidic medium, amino acid behaves like a base. In basic medium, amino acid behave like an acid. So hope I am very clear about this concept also. Okay. Now let's move on to the next one. Now let's talk about. So neutral amino acid we learned. Now we need to learn basic amino acid and acidic amino acid. What exactly basic amino acid? Very simple. Let's learn it in a simple way. Okay, I will write here only. C, COOH, NH2, H. So R can be of anything, no? Now I will write CH2 here, CH2 here, CH2 here. But along with this, I will write NH2 also here. Now count NH2 number. So 1 NH2, 2 NH2, but only one acid group. So if any amino acid, I am just writing a hypothetical example. It is not a specific example of any amino acid. Just an hypothetical example, okay? So you take any amino acid. In this amino acid, if you find more amino group, okay? So here I am getting more amino group than carboxylic group. Then such amino acids are considered as what? You are basic amino acid because you have a more base, more NH2 group. That's it. Only this much if you learn, that is enough. when amino acid has more NH2 groups as compared to COOH group, then it is a basic amino acid. So only one example you have to remember, that is lysine. So lysine structure is not given in NCERT. Let us not bother about that. Okay. Neutral amino acid over. Basic amino acid over, we learned. Now what is next? Acidic amino acid. What is acidic amino acid? You can guess the answer. More COOH group compared to NH2 group. Okay. So here we need to remember two examples. One is glutamic acid, another one is aspartic acid. Glutamic acid structure is given. Glutamic acid structure you need to remember. I will draw here. Very simple again. Draw COOH and NH2 as usual H. So no change in this. Okay. Now here write CH2 and another CH2. To this add COOH. To this add COOH. So now this is the structure of acidic amino acid. Why acidic? Look at here, man. One more along with this. So this is one carboxyl, this is second carboxyl, but only one amino group. Only one amino group. So this is the structure of glutamic acid. This is glutamic acid structure. Okay. So here I am just writing the number. Don't write 2 below COOH. So this is 1, this is 2 COOH. That is what I am telling. Even it can have more COOH, but the structure of glutamic acid is this one only. That's why I have drawn. Aspartic acid structure is not given, so let us not bother. But you have to remember the example. Okay. So all three amino acid structures, differences we have understood. Now if I summarize all three together, how I can write? Can you write along with me? So let me write a neutral here. Let me write acidic here, as well as basic this side. So neutral amino acid, let me take an example of a valine, because anyhow valine structure we need to learn. Okay, not valine, alanine. So this is neutral amino acid, one NH2, one COOH. Okay, and let me write glutamic acid as an example. Here in the name itself, acid is given, glutamic acid. So here what is the structure? Write C, COOH, NH2, H. So here one more COOH is there. Okay, so one COOH, two COOH. Therefore it is acidic in nature. That means acidic amino acid. In basic amino acid, structure is not given. Simply let us write an hypothetical example. COOH, H, NH2. Anything you can write. CH3, NH2. Anything you can write. So here, two amino group is there. That means more than carboxylic group. Amino group are present. So that's why it is basic amino acid. Now I am very clear about the differences between acidic, basic and neutral amino acid. Neutral behave like an acid in basic medium, behave like an base in acidic medium. And if you put it in a neutral medium, it shows a zwitterion structure. Okay. So both cation and anion structure present in the same molecule. That's it. So what we have learned till now, the same thing. Okay. So now let's move on to one more important thing, that is essential and non-essential amino acid. What is the meaning of this? Look at here. In our body, there are plenty of amino acids are there. Whatever we are today, my dear students, this is mainly because of protein. So my eye color look like this. This is because of a protein. That is nothing but a pigment called melanin. My skin color is like this. Again, because of a protein called a pigment melanin. So I used to digest the thing when I eat. This is because of the enzyme. The enzymes are protein. Okay. My hair is like this because of keratin protein. My skin is like this because of collagen protein. That means, if I want to summarize in a simple words, whatever we are today, majority of the things which are present in our body, which has been conquered, which has been dominated by proteins only. Okay. So now, to make these proteins, of course, we require amino acid. But if you just consider all those amino acids, we can make a two group. What are those? One is essential amino acid, another one is non-essential amino acid. First of all, if you just keep getting lot of sweets, do you give a respect to that? Definitely not. Anyhow, every day I used to have a sweet. Just keep it aside. I don't need. If I want anyhow, I can get it. Suppose if you get the same sweet, once in a while, you are going to give a lot of respect. Correct? In the same way, in our body also, there are so many amino acids that will be present plenty. So need not to depend to take those amino acids in the form of a food. So that's why we just neglect it. We consider them as non-essential. Why non-essential? Because body is going to produce inside it. Body is going to produce. So therefore they are non-essential. But these are very much required because body is not going to produce. So that's why we need to take through the diet. Need to take through the diet. So therefore we are giving a huge respect to this. You are very essential, man. You are very, very essential. So just come to my body through diet. So we are giving a lot of importance. Let us consider them as essential. Okay. So if it is not required through diet because by our body itself is going to produce, you are non-essential. Why we need to take through the food? Okay. Just remember in this way. There are so many examples we can list for this essential as well as the non-essential. But in your NEET exam, it won't come because it is not in our NCERT. They have just mentioned the two categories, essential and non-essential. But even for your curiosity also, I have brought this. You can write in your copy. So essential amino acids are histidine, isoleucine, leucine and lysine. Just remember if you want, or else you just leave it, no problem. Non-essential amino acid means our body itself is going to prepare methionine, threonine, valine, tryptophan, arginine, phenylalanine. Okay, so these are non-essential because our body is going to produce. Right. Now almost we are at the end of the amino acid part. Only 3 structures we need to learn. One is amino acid which is having sulfur. Another category is amino acid which is having alcohol group. Another category of amino acid is there which is having a ring-like structure along with them. Okay. So let's learn one by one. One is sulfur containing amino acid. What is there in this? Cysteine and methionine. Just remember these two names. So here to this amino acid, let me write amino acid structure. Write down amino acid C, COOH, H, NH2. So this is amino acid structure. Correct? Okay. Now write down CH2. To this amino acid, if you add sulfur group, that means SH if you add, then it will become sulfur containing amino acid. This is the structure of cysteine. Remember this structure. Just add SH group to this R group, that is to the CH2 group. Okay. And another example, methionine, you have to remember. So like that, if an amino acid, if it is having OH group in the R chain, then we consider it as an alcoholic group. Example, serine. So how we are going to learn this? It is also once again very simple. Write down basic amino acid structure. Basic means general, common structure of amino acid. COOH, NH2, H. Instead of R, write to CH2. To this R group, you just add alcohol group. Alcohol means OH. Now this will be the structure of serine. Practice it. Twice or thrice it will come. What is there in this? All are easier structure. Then the third category is aromatic amino acid. So whenever we use the term aromatic, the ring-like structure will be there. Okay, R chain has aromatic group. So examples important once again, tyrosine, tryptophan, phenylalanine. So we need to learn these tyrosine, phenylalanine in our class 12th also. Okay, so our body is having tyrosine. Our bodies convert this phenylalanine into tyrosine. Tyrosine is most important amino acid for the proper functioning of our adrenal gland, the thyroid gland, even the melanin production in our body. For everything, this tyrosine is like a precursor. If tyrosine is not there, then all these things, all this metabolism will be get hindered. So, okay, so that's why tyrosine is very important. It will come to you in your class 12th. If you are a class 12 student, you know about this. It comes in Principles of Inheritance chapter. Okay, so these two once again, it's an aromatic amino acid. So now here, let me draw a structure of tyrosine. Okay, it's important. How we are going to write? C, COH, NH2, H. Simple, very simple. Then write CH2. Now to this, just add a ring-like structure. Okay. Now you just place OH to this. So this entire structure, what I have drawn now, this is the structure of tyrosine. Tyrosine, tryptophan and phenylalanine. So over. So, amino acid part is over. Now, let's read NCERT. What is given in here? What our NCERT says. Let's see. Amino acids are organic compounds containing amino group and acidic group as a substituent of the same carbon, that is alpha carbon. So, I told about the meaning. Hence, they are called alpha amino acid because on the same carbon, both groups are present. They are the substituted methane. So, we have understood the meaning this. There are 4 substituent. Remember, it is not a 3. NCERT is telling it is 4. We need to follow it 4. Substituent groups occupying the 4 valencies position. These are hydrogen, carboxyl group, amino group and a variable group. Variable group in the sense they are talking about the R. Designated as R group. Here it is given. Based on the nature of R group, there are mainly amino acids. However, those which occur in a protein are only 20. 20 types are there. Okay. The R group in these proteinaceous amino acids could be, see, R group can be hydrogen. The amino acids are called glycine. So, in the glycine, we have hydrogen. Methyl group, that is alanine. Hydroxyl methyl, that is serine. So, just now I draw the structure. 3 of the 20 are shown in the figure 9.1. I have not put this, but if you just open the NCERT textbook 9.1, the same structure what I have drawn no, till now, so it is there. The chemical and physical properties of amino acids are essentially amino, carboxyl and R functional groups. Okay. Based on the nature of amino and carboxyl group, there are acidic, example glutamic acid, basic, lysine and neutral, valine amino acids. So, example you need to remember. Similarly, there are aromatic amino acids, that is tyrosine, phenylalanine and tryptophan. Remember the example. A particular property of amino acid is ionizable nature. That means they are talking about the Zwitter ion property. NH2 as well as the COH groups. Hence, in solution of different pH in the sense either it is acidic or a basic, the structure of amino acid changes. It can behave like an acid or even it can behave like a base. Okay, look at here. So, this is the Zwitter ion property where they are behaving like this in a neutral medium. So, when you put this amino acid in a neutral medium, so it just lose one H plus ion and added here so that it become NH3. So, this is the Zwitter ion form. It is in a neutral medium. And here, what they are telling? COH is remain as it is. But here, it become NH3. How it become NH3? Because you are adding this amino acid into acidic medium. Acidic medium. Already there are so many Romeos are there. Why it need to accept H plus here? Therefore, it remain as it is. It is not behave like an acid. So, therefore, it is behave like a base in acidic medium. Here, this is a basic medium. Basic medium. Therefore, it behave like an acid. Because there are OH is there. OH is there. Therefore, this H plus has removed from here. Just now, I have told everything. Okay. So, if such question comes in your NEET, you should be able to understand it. So, now let's solve few MCQ regarding this part. Okay. Alpha amino acids are organic compounds containing. See, alpha amino acid means what they are telling. Amino group and acidic group substituted on different carbon? No. Keto group, first of all, we are not talking about keto group. Therefore, these options are wrong. So, let us check option C. Amino group and acidic group substituted on the same carbon. So, this is our answer. Therefore, it is called alpha carbon or alpha amino acid. How many substituted groups are present in alpha amino acid? How many substituted groups? Alpha amino acid, just take an example of a methane. 4 substituted groups. So, this is our answer, not 3. The R group in a proteinaceous amino acid could be, it could be hydrogen, like a glycine. It could be methyl group, like alanine. It could be hydroxyl methyl. Okay, so anything, any of the above. This is our answer. You can put anything in the R group. A hydrogen substituted carbon containing amino acid. Hydrogen substituted means they are telling COOH is here, H is here, NH2 is here. Now in the R group, we have been substituted this H. Means it is the structure of glycine. Glycine structure. If it is alanine, it should be CH3. Which of the following group of amino acid is aromatic? Aromatic means tryptophan, tyrosine, tryptophan, phenylalanine. So here tyrosine is there, phenylalanine is there. Let us go with this option. And here tyrosine is there, tryptophan is there, but glutamic acid is given. Glutamic acid is a acidic amino acid. It's not aromatic. Again, glutamic acid is there. It is also wrong option. None of the above is not an answer. Option A is the answer. Next one. Which of the following is a neutral nature? Neutral amino acid. Valine, serine, alanine, R. Which one? Alanine. Alanine is a neutral one. Correct? Yes. Next. Which of the following determines the particular property of amino acid in ionizable nature and structure of amino acid? Based on which ionizable category we are going to do this? NH2 as well as the COOH. So this is the answer. Not just by only COOH, not just by only NH2, not even none of this. So okay, I am very clear about this concept now. I said the amino acid joined to become by a polypeptide chain, peptide bond, it become a polypeptide chain. That become a protein. Now let's learn few things regarding protein. Okay, now let's talk about proteins. So one thing is very clear that proteins are nothing but it is a polymer of amino acid. Correct? So polymer means many monomers join together and form a polymer here. So here these are the monomers. What are these things? Of course it is amino acid only. They are joined with the help of a peptide bond. So here I can mention these are a peptide bond. Okay. So this is one peptide bond. This is another peptide bond. So like that every amino acids are interconnected with the help of a peptide bond, so that it form a long chain. So this long chain itself we call it as a polypeptide chain. So obviously their weight would be definitely more than 1000 dalton, because single amino acid itself weigh between 18 to 800 dalton, means obviously their weight will be more, no? So that's why it is one of the macromolecule, biomacromolecule. And one thing you have to keep it in your mind that, so these amino acids are not of the same type. All amino acids are different. Maybe their sequence might be different, and even the same amino acid might be repeated for several times. For example, if I say this is a lysine, here also I may get a lysine, here also I may get a lysine, but it doesn't mean everywhere I am going to get a lysine only. Because in between, it may be by some other amino acid. Say for example, this would be the leucine, this would be tyrosine. So like that, different amino acids are joining here, and such a chain we call it as a heteropolymer, where different amino acids are present. So these are the basic things that we are understood in a protein part. Though we have learned amino acid, but these basic things we got it. Now the question is, okay ma'am, you said the two amino acids can be interconnected by a bond called peptide bond. Okay, we do agree for that. But how this peptide bond formation occurs? So if you know the structure of amino acid, easily we can construct this peptide bond. Okay, so let's see how this peptide bond formation occur. So for that, write two amino acid structure. You take any amino acid. Either it is a neutral amino acid or acidic amino acid or a basic amino acid, anything you take. But if they have to connect together, there must be a reaction occur. What is that reaction? Dehydration reaction. That is nothing but a removal of water molecule. So in every cases, wherever the bond formation is taking place, either it is a ester bond, or it is a glycosidic bond, or it is a peptide bond, okay? Always dehydration reaction occurs. Water molecules do get removed from those molecules, okay? Let's see. Let's construct a peptide bond between two amino acids. Write down in your copy. Write down two amino acids. So this is one amino acid which is having COOH here. H, NH2. So let me consider anything. Okay, so that's why let me consider it as a R1. Now let me take one more. So two amino acids are there, right? So this is amino acid number 1. This is amino acid number 2. Now, for the removal of water molecule, so what we need? We need one OH. We need a OH group from one amino acid and H from another amino acid. Here 2 H are there. So that's why for our convenience, I can write in different way also. Just look at here carefully. First, before rubbing please do write down these 2 amino acid in your copybook. Okay. Yes, I will rub it here now. See, I will rub this OH so that the OH has removed here. And here also I will rub this part. I will write. Wait for a second. See, I can write NH also like this way. Because it is nothing but NH2 only. But the writing format is different. That's all. So like that here also, this O, I will rub and I will write here on the top so that the valency has to get balanced. So here OH is there. Now it is perfect. Correct? So now you take OH from this side and H from this side. So what happened? So here water molecule has got removed. Correct? So this process we call it as a dehydration reaction. What is dehydration? Removal of water molecule. So as a result, if I rub this part and if I construct a bond, I am rubbing it. Look at here carefully. Now, here I can write this bond. This bond is nothing but what? It is a peptide bond. Peptide bond. Okay. Very simple structure. Now these 2 amino acids are nothing but dipeptide. Okay. So many students may confuse. They get confused and they write, if the word dipeptide is there, they think that there are 2 peptide bonds. But actually dipeptide means only 1 peptide bond between 2 amino acids. Okay. So very simple structure. So like that, I can construct this peptide bond, or I can make this peptide bond between 2 amino acids like for 50,000 amino acids, for 10,000 amino acids, for 1 lakh amino acids. So it is depending upon the protein. So how much the length of the protein, if it is very high, obviously the peptide bond also very high because it must take place between 2 amino acids, right? So let me construct a polypeptide chain. Okay, shall I move? Okay, polypeptide chain. I will tell you about these N-terminal and C-terminal later. First, let us learn how the polypeptide chain will be constructed. So if you know the peptide bond formation process, then obviously you can do it for several amino acids. Okay, write down in your copy along with me. When Neela ma'am is here, why do you fear? Okay. Yes. So I could construct only 4 amino acids here. So like that you can make it for 50,000. Just now I told no, 50,000, 1 lakh, 10,000. So the number of amino acids, there is no limit for that. Okay. So now let's construct peptide bond. What I said, for our convenience, let us write H in different way. So I will rub this part now. I will rub this part also. Okay. I can write N, H and H here. So like that, I can write this double bond O and OH here. So what happens? Here, I can remove this OH as well as the H. So as a result, the water molecule has got removed and here one peptide bond formation occur. Okay. So like that here also, if I remove this OH, if I remove this H, that means one H has got deleted. So therefore, so here also I can make a peptide bond, right? So here also the same way, I can write, so I will write O here and H, here OH. So that if I remove OH and H from this side, OH and H from this side, obviously the resultant would be the peptide bond. Understood? So this is a long chain of amino acid. So this long chain is nothing but polypeptide chain. Polypeptide chain. That means many peptide bond has formed. Here I don't know which amino acid we have took. So that's why in a general structure, in general way, I have represented the common structure. There is R1, R2, R3, R4 like that. Okay. Now listen to me carefully. So what I said, like this, you can construct a peptide bond between several amino acids. But if you start making a protein chain, that is a polypeptide chain, by taking amino acid, obviously you must start from first amino acid, right? Though their number may get varies up to 50,000, but it has to begin from the first amino acid. Correct. So now let us consider this as a first amino acid. Then obviously, if you look at here carefully, if you observe here carefully, so in this first amino acid, the NH2 is the terminal end. Correct. So that's why I can mention here this amino acid as N-terminal amino acid. N-terminal amino acid. Because this amino acid is having the terminal portion as amino group. Okay. And this N which is present in the left side, this is the left side. So that's why the left side, the amino acid which is having N-terminal is also called N-terminal end. N-terminal end. So like that I can start constructing peptide bond between several amino acid, and finally, though their number may get varies, it could be 10,000, but it has to stop at one end, right? It could be 1 lakh, but finally it has to get stop at one particular amino acid. So if you observe the last amino acid, so let's consider this would be the last one. So in this last amino acid, which end is the terminal portion? Of course, the COOH. Okay, so the C terminal. So that's why the last amino acid is considered as C-terminal amino acid. So this is the first amino acid and this is the last amino acid. So that's why this end itself we considered as what? C-terminal end. C-terminal end which is present on the right side. This is very important, which is present on left side. This is N-terminal, right side, C-terminal. Okay. So now the same information I have written here. The left is N-terminal and the right end is C-terminal. Now we have understood the construction of amino acid. But this amino acid is not like a stiff rod. Okay. Just like a pen, it is not like this. So that I cannot bend, I cannot make it flexible. This is actually a flexible chain. Suppose if you consider these are the amino acid. If it has been constructed by a bond like this, peptide bond, then it is a flexible thread. This amino acid chain or a polypeptide chain is a flexible thread. It is not like a stiff rod. This line is given in our NCERT. That's why I'm telling. So because of this nature, of this flexibility, it could be able to show many different variety of forms. So some may imagine you have a thread with you. So you can stretch this thread in this way also. If you want this thread to get folded in different way, you can fold it also. If you want the same thread to wave it like a woolen ball, you can do this also. Okay. So in any form you can do because the thread is flexible. In the same way, this polypeptide chain being flexible in nature, it is going to coil upon itself and started showing different structure. So based upon its flexibility in the sense, based upon the coiling nature, based upon the structure which is so going to form, we can classify the structure into 4 basic levels. Okay. One is primary structure, another one is secondary structure, tertiary structure and quaternary structure. Okay. So first let us learn about the primary structure. Primary structure of a protein. It is very simple here. See, this is one amino acid. Let us consider this as a methionine. Let me take one more amino acid. It is somewhere here. Let me consider this as a leucine. So like that there is one more. Let me consider it as a lysine. One more. Which shall I take? Phenylalanine. Tryptophan. Tyrosine. Any amino acid may come in this chain, but finally they have to join with the help of this peptide bond. They are joining with the help of this peptide bond. So such a single structure, just a single flexible thread-like structure, what you are going to see here, no, this is nothing but a primary structure of a protein. So in this structure, we could able to see the sequence of amino acid. That means in each sequence they are arranging, oh, first is methionine, after leucine came, after lysine came. So like that you can understand what is the sequence of this amino acid, and at the same time, the number of amino acid also. Suppose if it has begun from the first amino acid, now the last amino acid number might be 100, then I can say, oh, in this protein chain, It is not actually a protein, it is just a polypeptide chain, okay? It is just a polypeptide chain. It do not show any biological activity in our body, right? But I can understand how many amino acids are there. So this is just a simple structure of a protein called primary structure. Now, this amino acid chain, there is a polypeptide chain, sometimes they started coiling upon itself, just like a spiral staircase. Then it started forming one more structure. That structure we call it as a secondary structure. Okay, I will show you here. What is secondary structure? Listen to me carefully. Imagine I have this thread which has been constructed by a polypeptide chain like this. So, what is the name I have given to this? This is nothing but a primary structure. Okay. Now what happened? Now what happened? Listen to me carefully. So, it started coiling like this. See, it started coiling like a spiral staircase. Even in spiral staircase also, it is something like this only, no? Something like this only. Understood what I am telling? So, such a structure is formed once again by these amino acid only. So, here all the amino acids are present. So, it has been connected by this peptide bond itself. Are you getting my point? But this is not like a straight chain. It is somewhat like a helical structure. Therefore, we have given a name to this as alpha helix. Alpha helix. This is a secondary structure. So, a protein can do a secondary structure like this. So, such protein we could able to see in formation of a structure. For example, in our nails, we are going to see a keratin protein. So, keratin protein is something like this only in a helical form. And in our hair, once again, the keratin protein is there. That is also in this way only. Shall I write the example? Example part is not given in our NCERT. But if you have interest, you can write keratin protein, hair and nails. Okay. Now, sometimes the chain, the same chain, polypeptide chain, what you are able to see, which has been connected by a peptide bond, right? Now they are going to arrange in this way, just like pleats of a saree. So how we are going to fold the saree? That is, we are going to make a pleats, no? So like that, they are going to make a structure. So here, these are the amino acids, which has been connected by peptide bond only. But it is not like a simple thread. It has been pleated. So that's why we have given a name this as beta pleated structure. Beta pleated structure. So where we are going to see this map? Have you heard the fibroin protein? So in the silk, there is a protein called fibroin. So this fibroin protein of a silk is going to be arranged in this way. So this is beta pleated structure. It is also secondary structure. And this is alpha helix structure. This is also secondary structure. So in a protein chain, we could able to see both alpha helix as well as the beta pleated. But in some cases, the secondary structure which has been shown only by alpha helix, and some secondary structure which has been conquered only by beta pleated. So it may get vary depending on the kind of protein. But if we are talking about a secondary structure, you should know that either it can be present in the form of alpha helix or in a beta pleated structure. Understood? But they do not do any biological activity. Biological activity in the sense, if they have to do certain breaking process like enzymes, if they have to do certain hormonal action, that is what hormones only. So such action will not be carried out by secondary structure. We call them as biological activity, but it's not done by this. Just they could able to give a proper shape or a structure to a parts of a body, like a hair, nails, what I give the example, no? The same thing. So hope I'm very clear about this secondary structure. Now the question is, okay ma'am, each amino acids are going to be attached by these peptide bond. But if they are twisting upon themselves, obviously there must be some other force, right? For example, look at here. If I say here, certain force are present. Because of this, they could able to twist upon themselves. So this force should able to hold this helix in a proper way. No? Here also. So who is going to do this? Apart from peptide bond, there are certain other forces also act on these structures. Okay, what are those forces? Hope you are writing in your copy. I will move on to the next slide. Those forces are hydrogen bonding, disulfide linkage, van der Waals forces and electrostatic forces. If I say that these are the amino acid, this is the primary structure. This primary structure is stabilized by peptide bond. If I say the primary structure once again get called and become a secondary structure, so additional forces required to make it stabilize. So those additional structures are hydrogen bonding, disulfide linkage, van der Waals forces and electrostatic forces. Okay. And one more important thing you have to remember. These helix, it could be left-handed helix or it could be right-handed helix. Left-handed helix means something like this. This is left-handed helix. Imagine you are standing here and if you want to climb a staircase in this way only, you are going to hold the rod. You are going to hold the rod in this way. You are going to step up, right? So here what the steps are present. So you are going to climb the stairs in this way. Okay, so this is called a left-handed helix. Left-handed helix. And if the helix are present in this way means, imagine you are standing here. Now we are going to move in this right direction. Okay, this is called right-handed helix. Right-handed helix. Remember one important point here. No protein occur in a left-handed helix form. No protein occur in this way. Only they are present in right-handed helix form. Understood? So this is one more important thing you have to remember. Now let's move on to the tertiary structure. What is tertiary structure? Let me write once again the primary structure. So this is primary structure which has been connected by this peptide bond. Now they started coiling upon itself. Either it is in a helical form. So these are the amino acids connected by peptide bond and other forces. So this is secondary structure. This is primary. This is secondary structure. Now what happened? Listen to me carefully. It is further interesting. Now these helix further started coiling like a woolen ball. Something like this. Something like this. Something like this. So here it has been intertwined, right? Here are the amino acids. Everywhere the amino acids are present. Let's imagine these green color things are nothing but the peptide bond which has been present between these two amino acids to make them to form a chain. That is a polypeptide chain. So this polypeptide chain only they are interconnecting in this way. Okay. So this is nothing but the tertiary structure. So these tertiary structure are responsible for biological activity. So in our body, if any protein is carrying biological activity, then obviously it will be in the form of a tertiary structure, remember. See, majority of our hormones, you are going to study in human physiology, there is a separate chapter about the chemical coordination and integration. There we are going to talk about the hormones. So some hormones, they are proteinaceous in nature. Okay. There are some hormones, they are having a lipid nature. We call them as a steroid hormone. Okay. But majority of the hormones are proteinaceous. So they are going to carry certain functions in our body. So they are in this way only. That is in the tertiary structure. So like that, in our body, the enzymes are present. So almost all enzymes are proteins, except one enzyme that is nothing but RNA, which act as a protein that I am going to tell shortly about in the nucleic acid. So except few things, majority of the hormones, majority of the enzymes are nothing but proteins only. They are proteinaceous. Okay. So imagine this is one enzyme. So this enzyme, it has to break the molecule. Imagine you have ate something, you had some carbohydrate, you had some lipid in your body. Okay, lipid, carbohydrate, I don't mention. You will get confused. Imagine you had some breakfast, okay? So that breakfast has to get digested. How it is possible? That is by these enzymes. Imagine this is enzyme only. In this enzyme, there is a particular place. This place is known as active site. In this active site, the substrate molecule will come and bind here. So we'll study even about the enzymatic action. So the particular substrate is going to combine in this active site. So as a result, it will break down into a small molecule that is nothing but a product. So that is also biological activity. So that is done by this tertiary structure only. So who is going to stabilize this? Because somebody should be there to hold this structure in this proper shape, right? Once again, the same forces. Hydrogen bonding, disulfide linkage, van der Waals, electrostatic forces. These forces only. Now let's talk about the quaternary structure. It is very simple. So what we have learned till now, this is primary structure, correct? So this is secondary structure, right? And this would be the tertiary structure. Now construct many tertiary structure. So this is one tertiary structure. This is another tertiary structure. This is one more. This is one more. Okay. So I have constructed 4 tertiary structures. All these tertiary structures, once again, they are interconnected like this. That is by same bond only. Disulfide linkage, hydrogen bonding, van der Waals forces, electrostatic forces. These forces itself make them to construct in this way. So this itself we call it as a quaternary structure. So the best example we can take for quaternary structure is nothing but hemoglobin. Hemoglobin, which is so present in our body, it is a best example for quaternary structure. Here in this hemoglobin, the 2 chains are similar, right? So these 2 chains are called alpha chain. And these 2 chains are similar, right? They are called beta chain. Beta chain. So you should know this because once again in sickle cell anemia, we are going to study about this alpha chain and beta chain. Okay. So here the 4 tertiary structures are joined together and form a quaternary structure. Okay. So hope I am very clear about the structure. This is primary, this is secondary, this is tertiary, and this would be the quaternary. Very simple. Over. Class over. I mean protein part is over. Now let's read NCERT part. So protein part I am reading. Amino acid part we have done. Now we have entered into the protein part. That's why I said just please open your textbook because I am doing in between. Or else we cannot make the dot to get connect. Okay. So this chapter itself is like that. If you connect the dots, then we will understand each and every line of NCERT. And finally, after completion of the syllabus, after completion of the chapter, if you read NCERT, you will understand each and every point. Okay. So now I am reading the protein part. Okay. Just open the protein part of your NCERT and just follow it. So proteins are polypeptide that we have understood. They are the linear chains of amino acids linked by peptide bond as shown in the figure. That means already we have understood that. Each protein is a polymer of amino acid as there are 20 types of amino acid. So examples are given: alanine, cysteine, proline, tryptophan, lysine, etc. A protein is a heteropolymer. Remember, it is not a homopolymer. So it is a statement based question. Okay. A homopolymer has only one type of monomer repeating n number of times. But such things do not happen in protein case because amino acids will be different. This information about the amino acid content is important as a ladder in your nutrition lessons. That means we'll talk about essential and non-essential amino acids. You will learn that certain amino acids are essential for our health and they have to be supplied through diet. Hence, the dietary proteins are the sources of essential amino acid. Already we have learned that in amino acid part itself. There are amino acids can be essential or non-essential. The latter in the sense they are talking about the non-essential one. are those which our body can make. That means we are not giving respect to that, which is actually making in our body free of cost. Okay, that's why we said you are non-essential. While we get essential amino acid through the diet, there is nothing but through food. Proteins carry out many functions. See, till here we have learned in amino acid part only. Now here few functions are given. Let us understand about that. What they are telling, proteins are carry out many functions in living organisms. So some transport nutrients across the cell membrane. It means what? Look at here. Look at here. Imagine this is a cell. Imagine this is the cell. So this cell is very upset. Why it is very upset? Because if this cell want to get ATP, it required glucose, and this combined with oxygen, it has to get converted into ATP only. So who is going to give glucose for this? So glucose is there in your bloodstream. You had carbohydrate, you had lot of roti, chapati, everything, rice, dal, everything you had, but which is present only in your blood vessel. Imagine it is there in your blood vessel. This is the blood vessel where glucose are moving. But glucose molecule are telling, we are ready to go inside the cell. This is cell. But the gate is not open. How we can go? So by that time, our hero comes. So you will, once again, you will understand about this also in chemical coordination and integration chapter. This hero is nothing but insulin. So insulin says, don't worry my friend. If you are present in more quantity in blood vessel, then it's a big problem for me, because my job is to reduce your level in the blood. Okay, so that's why insulin is very important, to reduce the glucose level in the blood. So what it will do, it will say, cell, you do not have gate. No, I will make a way. So it will just open the gate here. Open the gate in the sense, a kind of channel will be created by this insulin so that the glucose can easily enter inside the cell. So this channel or this protein is nothing but GLUT4. It is called GLUT4. It's a kind of protein. This channel itself is a protein. Its name is GLUT4. It is very important. It is very important for your NEET point of view. GLUT4 means glucose transporter. Glucose transporter 4. So the option may comes GLUT1, GLUT2, GLUT3, GLUT4. So you should know it is GLUT4. So through cell membrane, we could make the nutrients to pass. This is one function. And the second function, some fight infectious organism. That means, what is the meaning of that? Let me show here only. Okay, I will show here. Imagine this is a blood vessel, once again. So in the blood vessel, let's assume a bacteria or a virus has entered. This is a pathogen. So to destroy this pathogen, we need macrophage. So imagine here the macrophages are there. Actually, which will be present here only. Macrophages are there. So these are roaming, they are multiplying, they are just moving here and there in the blood. So by that time, certain proteinaceous molecule will come. So these are the proteinaceous molecule. They will come, they arrest it like this. They combine to this pathogen. So they just call the macrophages. This is macrophage, no? Macrophage is nothing but, it is a kind of immune cell only. So these are nothing but antibodies. So antibodies which are so present in our body that will enter into the bloodstream, they will catch this pathogen and they will call this macrophage. Hey macrophage, you just come here. We have hold this pathogen, you can now eat. Now easily this culprit will enter into the mouth of this macrophage. So this is how our proteins are helping in our body. So these antibodies are nothing but proteins only. For proper immune system, once again we require protein. Where it went? Yes. Okay. So it fights the infectious organism. Some are hormones. I said some proteins are act like hormones and some are enzymes. Just now I gave the example about the enzymes also. That is nothing but a tertiary structure. So here they have talking about these things in the table 9.5. Where is the table? Here is the table 9.5. So collagen, it is an intercellular ground substance. So collagen is a kind of fiber which is present in a connective tissue. Ground substance means if it is a tissue, these are the cells and the rest of the thing which is so present, they are nothing but the ground substance. Here these fibers are present. Beneath the skin, the connective tissue is there. If our skin is so elastic and they are very flexible and we look young because the collagen fibers. Okay. So this collagen is also a protein. So intercellular ground substance. And trypsin, it's an enzyme. And insulin, it is an hormone. Antibody, which fight against infectious agents, they are also protein. And receptors, receptor means, imagine this is the cell. On the surface of the cell, certain receptors are present to receive the signal, so that the chemicals will come and combine here on this receptor. So these receptors are also proteins only. Suppose if you are able to sense the smell, and we could able to sense the taste, okay, touch, everything, that is by these receptors only. And certain hormone will come and sit here on the cell, so that proper function will occur in our body. That is also by the receptor itself. Okay, so these receptors are also protein. So just within 1 or 2 minutes, you cannot understand these concept of what is the meaning of receptors, where they are present. If you want to know the meaning, if you want to become a master in biology, then you should understand each and every line of NCERT textbook, whole textbook. NCERT textbook, both of class 11th and class 12th together, if you combine, and that will be considered as one chapter, because every chapter, every line, every concept are interconnected. For example, here I am telling the receptors are there, the receptors for hormones, receptors for neurotransmitter. So what are these hormones? What are these neurotransmitter? For that to understand, you must have a knowledge about the hormones. You must have knowledge about that chapter, chemical coordination and integration. So that's why I said these chapters are interconnected, interlinked. So if you have a knowledge of all these basic things, then trust me, definitely you will become a master, and there is no doubt in scoring out of out in NEET exam. Okay? Yes. But even if you don't have knowledge, you can understand the basic things what I am explaining here. Then GLUT4, just now I told, it enables the glucose transport into the cell. Okay, so 9.5 table we understood. Collagen is the most, this is very important line. Collagen is the most abundant protein in animal world. Okay, in animal world, collagen is most important. And cellulose is most abundant in plant world. But cellulose is not a protein. Cellulose is a carbohydrate. Here, collagen is a protein. And ribulose, biphosphate, carboxylase, oxygenase, that is nothing but ribisco. You are going to study about this ribisco in photosynthesis chapter. I am going to tell you in detail about that. It is having some interesting and most important thing we need to learn about ribisco. They will learn. Okay, is the most abundant protein in the whole biosphere. Did you understand? If you start looking at who is having the most abundant protein, I mean, which is the most abundant protein in the whole biosphere, either it is a plant case or animal case, then we could able to see ribisco. Okay, but if you just look at only the plant, it is cellulose. I mean, cellulose is the most abundant, but it is a carbohydrate. But if you look at only the animal, then the collagen. Remember these 2 points, collagen as well as the ribisco. Ribisco is in the whole biosphere, collagen is only in the animal world. So we have understood all those things. Now let's study about the structure of protein. Structure of proteins. Proteins are mentioned earlier as heteropolymer, containing string of amino acid, already I have told. Structure of molecule means the different things in different contexts. See, if you have to explain the structure of a molecule in different way, I can explain in different contexts are present that shows a different manner to explain the structure. For example, if I ask an inorganic chemist, some person is there, inorganic chemist, I am going to ask him, can you mention the structure of NaCl? NaCl is an inorganic molecule. How we can explain the structure? He just say, look at the molecular formula. Na is there, Cl is there. So it is just his cup of tea. Only he could able to explain the molecular formula, not a structure. Suppose if I ask an organic chemist, if I call a person who is studying about the organic chemistry, and if I ask him to explain the structure of some benzene, if I ask him to explain the structure of ribose sugar, then obviously he can say, oh, the ribose sugar is something like this. It is having the oxygen here. This is having these type of carbon. So he can explain the structural formula, but it is just a two-dimensional structure. So like that, if I call a physicist physicist, he can explain the structure in 3 dimensions. So look at this pen. He can explain this in 3 dimensions. So in a 2D view, you can see that like this. So in 3D view, if you just turn, we could able to see all the structure of this pen. That is a 3-dimensional view. Now, being a biologist, we can explain the structure of a protein. That is in 4 levels. That is primary level, secondary level, tertiary level, quaternary level. So that is what they are telling. In different context, it varies. Whether it is a 2-dimensional, 3-dimensional, 4 levels like this. So that is what the meaning it is given. But from this question won't be framed, don't worry. But still let us understand. In inorganic chemistry, the structure invariably refers to the molecular formula. For example, NaCl, MgCl2, etc. Organic chemists always write 2-dimensional view of a molecule while representing the structure of a molecule like benzene, naphthalene, etc. Physicists, 3-dimensional views of a molecular structure, while biologists describe the protein structure in 4 levels. Okay. The sequence of amino acid, that is the positional information. Look at here. Positional information they are giving, which is the first amino acid and which is the second and so on. So that is nothing but a primary structure. We have understood the sequence and even number of amino acid. A protein is imagined as a line. The left end, look at here, the left end represented by the first amino acid, right end represented by the last amino acid. The first amino acid is also called N-terminal amino acid, I told, which is present in the left side. The last amino acid that is present on the right side, is called C-terminal amino acid. A protein thread does not exist throughout as an extended rigid rod. See, NCERT itself is telling protein is like a thread, it is not like a rod. The thread is folded in the form of a helix that is similar to a revolving staircase. Of course, only some, see, look at here. When you start reading this paragraph, you will get confused. NCERT has given in such a way that the students definitely will get confused in the secondary and tertiary structure. So what I said actually, the chain, polypeptide chain, it can be twisted in this way like a alpha helix. Okay. So actually the helix has given in this way, but somehow it is like a spiral staircase. Or the same thread in the sense, the same polypeptide chain even can be in the form of a beta plated structure, something like a plates. Okay. So here they have given in this way, This is what the beta plated sheet. So this through diagram we can understand, but while reading NCERT, we will get confused. Look at here, I will tell you. Of course, only some portion of the protein thread are arranged in the form of a helix. That means they are talking about the alpha helix here. In proteins, only right-handed helices are observed. So this also I told, it never be left-handed. See further, other regions of the protein thread are folded into other forms in what is called secondary structure. It means they are telling, while reading, what the students will assume, mostly the secondary structure is only a beta plated. It is not a alpha helix. But actually both comes under this secondary structure only. Okay. So the meaning of this line is, the same thread can show both alpha helix as well as the beta plated. It means to say, look at here. Suppose this is a primary structure. Okay. So it may twisted like a spiral staircase so that it can form an alpha helix. Within the same thread, we could able to see even a beta plated sheet also. Okay. So it may be present on a different proteins or it may present within a same protein. Alpha helix and beta plated sheet both can be sometimes seems to be occur on a same thread. That is what the meaning here. Okay. Hope I am clear about this. In addition, the long protein chain also folded upon itself. That means they are talking here about the tertiary structure, like a woolen ball giving rise to a tertiary structure. Okay. So this gives us a 3-dimensional view of a protein. Tertiary structure is absolutely necessary for many biological activities of a protein. That is important. So this structure we have understood. See, tertiary structure it is here, hydrogen bond, disulfide bond. Remaining two are also there, van der Waals forces, electrostatic forces, but it has not mentioned. And this is the quaternary structure. Here, these are the 2 chains, alpha chain, and these are the 2 chains, that is beta chains, but they have combined together and form a quaternary structure. Okay. This one, the slide repeated. Some proteins are assembly of more than one polypeptide. Are subunits the manner in which these individuals folded polypeptides or subunits are arranged with respect to each other. That means, example, a linear string of a sphere. Spheres arranged one upon each other in the form of cube or a plate, etc. is the architecture of a protein, otherwise called quaternary structure. That means their arrangement they are explaining. Adult human hemoglobin, it is the best example. It is very important. 4 subunits it consists. 2 of these are identical to each other. Hence, the 2 subunits are alpha type and 2 subunits are beta type. Together constitute the human hemoglobin. So, the structure we have seen. Sometimes the question may comes to identify. So, which type of structure it is? You should know it is a quaternary. If this image come and if the question has been framed about this, then you should write it's a tertiary structure. Then obviously these two you can understand. Okay. So, the protein part is also over. Now, let's solve few questions. A protein, if a heteropolymer means what? Different type of amino acid. It contains only one type of amino acid, wrong. It contains different types of amino acid. This is our answer. It cannot be both. Sometimes the questionnaire, examiner gives such options so that you should get confused. But don't get confused. Always stick on to this line that it is made up of different type of amino acids. Next, what are the functions of protein? Just now we have explained. Carry out many functions in a living organism, correct? Transporter of nutrient, it means they are talking about GLUT4. Then fight infectious, that is also right. Then regulates in the form of hormones, enzymes. So all the 4 statements which are so given about the proteins are correct. So it is not only 2, only 3. So it is 4. If it is given all of this, then we could go to this option. But here it is none. Next one. The most abundant enzyme in the animal world. Which one is the most abundant? Collagen. So collagen should be the answer. While in the whole biosphere, it is Rubisco. So, here it is PEP case given. Ma'am, what is the meaning of PEP case? That also you are going to learn in plant physiology. So, I will tell you, I will explain easily and beautifully. You will understand about this PEP case, Rubisco, everything in photosynthesis chapter. Okay. But here our answer should be collagen and Rubisco. So, therefore, option C is the answer. Not B, not even D. Next one. In a protein, the left end represent. Left end means here, the first amino acid they are talking about. So here first amino acid, okay, but it is C given. Therefore, it is wrong. Last amino acid given, wrong. First amino acid, but it should be N-terminal. So this is our answer, not last amino acid. So I know you are answering along with me because I know you have understood the topic very well. Next, which of the following statement is untrue? Untrue in the sense they are asking which one is false here. A protein thread is folded in the form of a helix. It is true only. Only some portion of the protein thread are arranged in the form of a helix. Correct? In proteins, only left-handed helices are observed. See, how come it is possible? In a protein, we see both left-handed as well as the right-handed. This would be the right hand. This would be the left hand. Both the hand we can observe. So, this is wrong here. So, this statement is wrong. This is not true. And the wrong statement itself is our answer. Therefore, this is our answer. Not both B and D. I mean B and C. Next one. The long protein chain is also folded upon itself like a hollow woolen ball is known as what? Tertiary structure. Where is? Yes, here is the tertiary structure. Next. Dash subunits of alpha type and dash of beta type together constitute human hemoglobin. 2 subunit. Yes, 2 given. But once again, here also 2. Here also 2 should come. So, therefore, this is our answer. 2:2. Not 2:4, 4:2 or 4:4. Next, peptide bond is formed by. So, how peptide bond formed? Just by removing one water molecule. The process we call it as what? Dehydration. So, here elimination of water molecule, that is rehydration given. Wrong. Addition of water molecule given, wrong. Here also addition of water given, wrong. Elimination of water molecule, that is dehydration. So, this is our correct answer. So, all are easy. Oh, done. Now, we need to talk about one more important topic. So, this topic actually follow you till you finish your NEET exam. because it is there even in class 12th also. So it is just a basic here. Okay. So once again in class 12, that is in molecular basis of inheritance, a big chapter that is. In that also you are going to study about this DNA. In detail you are going to study that. But here the basic things. What are the monomers? What are polymers? How these monomers are connecting? So those things you are studying here in this part. Okay. So that part once again repeated there also. So here whatever the information given in our NCERT, I am just teaching that much only. And of course, I am going to give little bit more information regarding DNA, though it is not given in class 11th, but that would be helpful to you in class 12th also. But don't worry. If you start watching molecular basis of inheritance chapter in my channel, there also you are going to get the information from the basic itself about the DNA. Okay. Now let's talk about nucleic acid. So what do you mean by nucleic acid? Nucleic acid means it is an acidic material. If I say a cell, if I say this is a cell, either it is a plant cell or animal cell or a viral cell or a bacterial cell, anything you can consider. So let's imagine this is the nucleus. Inside the nucleus, the DNA will be there. So DNA will be there. So this DNA carry all the genetic information. What it carry? It carry genetic information. Genetic information means what? Your height, your color, your face structure, your nose structure, your chin shape, your hands, your fingers, everything that has been passed from our parents. Okay. So the characters which are so present in your parents, that will be inherited to the offsprings, through which, that is nothing but through a structure called DNA. That means DNA is considered as a master molecule. It is having all the information of a cell. So how much enzyme has to produce in our body, how much hormone has to produce in our body, when it has to produce in our body, where it has to produce. So everything has been controlled by this master molecule, that is nothing but our DNA. Okay. So as it is carrying the genetic information, that is from parents to offspring, no, that's why we call it as a genetic material. Okay. So this DNA itself considered as genetic material. But in our body, apart from this DNA, even another nucleic acid is also there. For example, here we could able to get, that is nothing but RNA. So what is this RNA? In the case of eukaryotic cell, in most of the organism, let me write here, in most of the organism, DNA is the genetic material. DNA is the genetic material. What is the full form of DNA? Deoxyribonucleic acid. Few minutes back we talked about this. Deoxyribonucleic acid. RNA means just a ribonucleic acid. Right? So information will be carried from parents to offspring through DNA. That's why it is a genetic material. Then what about RNA? Is it not a genetic material? Of course, it is also genetic material only in some viruses, not in all organisms. It acts as only in few viruses. Except those few viruses, the rest of the organism that you are going to see in our surrounding, that is from bacteria to human being, everybody is having what? DNA as their genetic material. Okay. Then in our case in the sense, in human case, in plant case, in animal case, what this RNA will do if it is not carrying the genetic information? Very simple. I will tell you. Imagine this is the DNA. Imagine this is the DNA, something like this. Something like this. We don't know which information is there here. Okay. So, but that information, imagine here is the information in the form of certain nucleotides. I will tell you. Even if you are not understanding, I will tell you. Just know that here is some information. Might be it is having the information about how much height you have to grow. What is your height? So that has to be determined by this part of the DNA. Let's assume like this. Okay. So this part of the DNA, which is so having this information, it has to get translate into protein. Translate into protein means it should be in the form of a hormone or in the form of an enzyme, whatever. The information has to get copied. Okay. So that information will be collected from the RNA. Imagine here is the RNA. Imagine this guy is RNA here. So he is going to carry this information. So this RNA will be something like this. So he is the RNA. He carried all the information. Oh, DNA, do you have information like this? Okay, you just send it to me. I will take this information. So to whom I am going to give? I am going to give to certain subunits. I am going to give it to a machinery so that that information should be get carried in the form of a protein. So that these information which are so carried from this RNA, that will be translated and finally that will be formed in the form of a polypeptide chain. That is nothing but a protein. So ultimately we got this protein. So our body consists, majority of the organic material present in our body is protein. See, almost 50 to 60% our body is having water. That is actually inorganic material. But apart from that, I told about that organic material chart, pencil, you can remember in this way. So here P has the first rank, P in the sense protein. So protein is the most abundant organic material present in our body. So that's what we read few minutes back. Majority of the enzymes are proteins, hormones are proteins, and so many structural part is nothing but a protein. So this protein will be formed only through this RNA. So that's why RNA, even though it is present in our body, it just acts like a messenger. Okay, it's not actually the genetic material. So who is genetic material then? The DNA is the genetic material. Understood? But either it is the DNA or RNA, whoever it is, definitely they should also be having a monomer. Just like once again recall the construction of a wall, how we are going to place a brick one above the other through a cement by plastering a cement. In the same way, we have to keep a nucleotide one after the other to form this chain. Okay, that means I will write here only DNA is a polynucleotide chain. DNA is a polynucleotide chain. Even RNA is also polynucleotide chain. Where shall I write? I will rub this part. Or else let it be, no problem. I will write here only. RNA is also polynucleotide chain. So whenever we are using the term polynucleotide in the sense it is nothing but a polymer. So obviously it must have a monomer, right? It must have a monomer. So who is monomer here? The nucleotides are the monomer. Nucleotides are the monomer. Just like how amino acids are a monomer for a protein. Small amino acids join together by a peptide bond. They become a polymer called protein. Small saccharide units, that is a monosaccharide, they combine together and form a polysaccharide like a cellulose, starch, glycogen. So like that, small nucleotide join together and form a polymer, that is polynucleotide chain. So this polynucleotide chain itself we call it as a nucleic acid. Why nucleic acid? It is having an acidic material in it. Very simple, easiest topic it is. You will understand. Okay. Now, if I say this is one polynucleotide, this is one nucleotide and this is another nucleotide. So like that, I can make several nucleotides. Then obviously these nucleotides are going to get joined like this by a bond. Which bond that is, we will see. But these nucleotides here, I can consider as a monomer. Okay. So this entire unit, what we are saying, this entire structure is a polynucleotide. It's a polynucleotide. Being heavy, where it must come? It should be there in acid insoluble pool. It's a part of acid insoluble pool in the sense it is retentate, found in the retentate. Whereas these nucleotides, that is monomer, they comes in acid soluble. Now, the funny thing is, everywhere till now what we have studied about the monosaccharide or amino acid, they were just one single structure. So you cannot break it further, right? If I take amino acid, you cannot break it further into smaller pieces. If you break it, it will lose the property of amino acid. But here, this nucleotide, even though it is a monomer, but within that monomer, it is having three components. Look at here, for example, I will show you. Imagine somebody is here. Somebody is here. somebody is here in this part. So this is one portion. So like that, somebody is here also. So this is another portion. And one more person is here. He is another person. That means 3 components together. This is one component, this is second, this is third. 3 components together present within one nucleotide and form one single subunit called nucleotide. Let's see what are those 3 3 components. Okay. Components of nucleotide. So let me write 3 components. So the first component here is nothing but already you know about this ribose sugar. So this ribose sugar acts just like a ribose only in the case of RNA. Okay, I'll do one thing. I will rub this part. I will write as sugar only, not ribose sugar. So the first component here is sugar. So this sugar is nothing but a pentose sugar. Already we have learned the structure of drawing this pentose sugar, right? So if it is a case of RNA, this sugar is ribose sugar. If it is a case of DNA, then this sugar is what? Deoxyribose sugar. So we have understood the difference between deoxy as well as the ribose sugar. Okay, so this is one component. And another component is nitrogen base. So this nitrogen base is considered as heterocyclic ring. Why it is heterocyclic ring? I will tell you. Because in a ring-like structure, imagine this is a ring of nitrogen base. Here, in the corner, it will be having carbon-carbon. But it is even, in some places, it is having nitrogen along with the carbon. It is having nitrogen. Therefore, apart from carbon, even in the ring, we could able to see the nitrogen. Therefore, it is called hetero. Hetero means different. Cyclic means ring-like. Okay, in a ring-like structure, different compounds are there, not only carbon. Therefore, it is heterocyclic ring. Now, this heterocyclic ring, that is nitrogen base, are of 2 types. One is purine. Another category is called pyrimidines. So total there are 5 nitrogen bases. So those 5 nitrogen bases can be classified into a purine group as well as in a pyrimidine group. 5 members are there. So we started grouping them. Oh, you go to the purine group and you also go to the purine. And 3 members you just go to pyrimidine group. Okay. So who are those people? We are sending them into purine and pyrimidine. Let's see. So there is a nitrogen base called adenine. It has been represented just as A, placed under purine. So like that, even guanine, which has been placed under purine only, represented as G. Whereas in the pyrimidine, cytosine and thymine comes, which has been represented by C and T. Okay, so these are the 4 nitrogen bases. But there is one called uracil, which has been represented by U. Okay, so listen to me carefully. If we are talking about RNA, adenine is there, guanine is there, cytosine is there, but thymine is not there. Instead of that, uracil will come. Where? In the case of RNA. So which is present only in RNA. Along with these three. But if you are talking about DNA, then in the place of uracil, this thymine will come. So thymine and uracil are the exchanging part of nitrogen base. But these 3 structures, they remain as it is. You should know this. It is very important. Okay. Now, how we are going to represent this purine? Let me show here. I don't have enough space here to write. Purine. And let me write pyrimidine. I'm just writing the general structure. Pyrimidine, though the name is very big, but it says, I'm very down to earth. My name is big, but I don't behave like having a head weight. Okay, I'm very simple structure. See, I'm just a small 6 ring-like structure. That's it. So you can you can give a number like this from the bottom, because I'm down to earth. No, just give a number from bottom itself. 1, 2, 3, 4, 5, 6. Here is the nitrogen in the first group. Okay, in the first place. And to this nitrogen, hydrogen molecule is attaching. You should know this. I will tell you. It will come to your help. Okay. But purine, it says, you have named me with a very small digits. Not digits, with a small letters. So my name is small. It's not like a purine. So that's why I should have a big structure compared to pyrimidine. So that's why it is having 2 rings here. What are those? One is 6 number ring like this. And another one is pentameric. This is hexameric. And another one is pentameric. 1, 2, 3, 4, 5. See, 1, 2, 3, 4, 5, 6 here. 1, 2, 3, 4, 5, 6. Here 1, 2, 3, 4, 5. That is pentameric. So this is having head weight, I said. Correct? So that's why let's name it from the top itself. 1, 2, 3, 4, 5, 6, 7, 8, 9. Here at the 9th position, nitrogen is there. And to this, hydrogen has been attached. Okay. So this purine, it could be your adenine. Or it could be your guanine. So like that here, for pyrimidine, the same structure, I can represent it for cytosine, or thymine, or uracil. So small little bit changes that we could be able to find in different nitrogen bases, but it is not given in NCERT, not even in class 12th also. Let us not bother about this. Okay. And the third structure is nothing but a phosphate. So this phosphate itself known as phosphoric acid, which is having negative charge on it. Therefore, DNA is a negatively charged particle. Both the DNA and RNA are negatively charged particle. But here we are giving main importance on DNA only. Therefore, DNA is considered as negatively charged because phosphate is there. Okay. And this is nucleic acid. Why nucleic acid? Because the phosphoric acid is present in this nucleotide. That's it. Okay. Now let's see how we are going to combine these 3 together to form one nucleotide. When you start knowing how one nucleotide has formed, then you can understand how these nucleotides are going to connect each other to different nucleotides to form a polynucleotide. So it is bit confusing, but actually not that much confusion you will have. But only thing is, I need your complete attention. Just focus here, you will learn. So this much we have learned, this much we have learned. Now, I will tell you the main difference between nucleotide and nucleoside. Here, these 2 terms are given in our NCERT. What is the difference between these 2? Let's see. Imagine here is the sugar. She is the sugar. So she is the sugar. First, she was having a friendship with nitrogen base. Then these 2 combination, nitrogen base as well as the sugar, these 2 combination we consider as nucleoside. When a sugar is combined with a nitrogen base can be of anything. It can be a purine, it could be a pyrimidine. But if a nitrogen base is having a bond with, if a sugar is having a bond with nitrogen base, then such group is nothing but nucleoside. Here this bond is called N-glycosidic bond. I will tell you how this bond will form. N-glycosidic bond. Okay. Then what is nucleotide? Look at here. Same sugar only. Sugar can be deoxyribose sugar or ribose sugar, but sugar. Okay. Now, earlier she was having bond with this nitrogen base, right? Through N-glycosidic bond. Later, she wanted to make a friendship with another guy also. So who is that fellow? He is nothing but the phosphate. So this bond is called ester bond. This is ester bond. Are you getting my point? Phosphoester bond. Not diester, it is phosphoester. Phosphoester bond. This is N-glycosidic bond. Now these 3 combination. Sugar is the same sugar. Earlier she was having a friendship with nitrogen base, but now she is even having a bond with phosphate. Now the combination of these 3 all together is known as what? Nucleotide. These are called nucleotide. So based on the name of the nitrogen bases, we started giving name to this nucleoside as adenosine. If this nitrogen base is adenine, then it is called adenosine. If this nitrogen base is guanine, then such nucleoside are called guanosine. If it is cytosine, it is called cytidine. If it is thymine, it is called thymidine. If it is uracil, uridine. So like that, the name will be varies based on the type of nitrogen base present here. But let us know how this bond formation occur between the sugar as well as the nitrogen base. So you know the structure of ribose sugar and even the deoxyribose sugar. Okay. So let me show here. So just recall. Here I am writing beta, not alpha, because I need to show the bond, okay? So this is first carbon atom, second, third, fourth. In the fifth place, CH2, OH is there. Correct? And here OH, 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 OH and H. I wrote like this, right? So this is alpha sugar. But now I need to make a bond with this. Therefore, I will write OH here and H here in this place. H in this place. So which sugar it is? Of course, you are right. This is ribose sugar. This is ribose sugar. Now, if I write everything same to same, OH here, CH2OH, and obviously here OH, H, H. But here in the second position, if I just delete one oxygen and if I write only H, and everywhere will be same to same. Here also H, everything will be same to same. But only the difference is I have removed one O group from here in this place. And here also opposite place is H only. Don't get confused. Then such sugars are called deoxyribose sugar. So this deoxyribose sugar is present in DNA. So this is present in RNA. Correct? But now let's learn the bond formation occur in DNA. So therefore, I will take this deoxyribose sugar only. Right? Okay. Yes, I will show here. Structure of nucleoside. Look at here carefully. Very simple structure. Let's draw deoxyribose sugar. Okay. So here it has to come CH2OH. Opposite is H. Here OH. Here H. Here I should draw OH. I should write OH. Okay. Everywhere is as it is. So here the carbon atoms are present. Now, if you have to take a nitrogen base, either it could be a purine or pyrimidine. Let me show the bond formation by taking both the examples. If it is a pyrimidine, it should have only single ring, right? Let's imagine this is the pyrimidine. This is pyrimidine. Okay. So in this pyrimidine, what I said, in the first position, it will be having nitrogen. So to this nitrogen, the hydrogen will be attached. Correct? Correct. Just now I told. Now, the first carbon of this ribose sugar or deoxyribose sugar that reacts with the nitrogen base which is present in the first position. So as a result, what happened? What happened here? Here the sugar says, I am going to give OH to you and you just give me H so that we both will react and we will remove water molecule. So when water molecule has removed, obviously a bond formation takes place, right? So let me write this. I will rub this water molecule. So as a result, here we could able to see a bond. This bond itself is known as glycosidic bond. Why the name N-glycosidic? Because it is making bond with hydrogen atom which is attaching to this nitrogen. Therefore, it is N-glycosidic bond. N-glycosidic bond. Glyco. Okay. Suppose if the same combination, that is nothing but deoxyribose only, if it has to make a bond with the purine, then how it will take place? Everything is same to same. Look at here. First draw ribose sugar, that means deoxyribose sugar. Right? So this is the first carbon, second carbon, third, fourth, and this is fifth. Now I will take I will take a purine. So purine is having 2 rings, right? So this is one hexameric ring, another one is pentameric ring. So in the here the numbering start 1, 2, 3, 4, 5, 6. Here 7, 8, and this would be the 9. To this 9, nitrogen is attaching. Correct? Not only here, the nitrogen might be present in somewhere else also, but here just to make you understand, I am showing this. Others you will get confused. Okay. So now the 9th position where the nitrogen is attaching to this hydrogen is present. Attaching to this nitrogen, the hydrogen is present. Now the reaction takes place between the OH as well as the H, just like here. So water molecule has got removed. Because of this, the N-glycosidic bond will be formed. Just now we have seen in the same way. So let me write N-glycosidic bond. Simple, very easy. Okay. So the glycosidic bond between the sugar and nitrogen base can form between the purine as even with the pyrimidine. So based on the name of the nitrogen base, we are going to give a name to this as adenosine if it is adenine. So sugar can be anything. If it is just a ribose sugar, it is adenosine. Suppose if it is deoxyribose sugar, then I should call it as deoxyadenosine. Okay. So like that, guanine with sugar, that is ribose sugar, it is guanosine. If it is deoxyribose sugar, then it is deoxyguanosine. Just add the term deoxy, that's it, to our DNA. Cytosine, then it is called cytidine. Don't write cytosine, cytosine only. So wherever you are going to see the term sin or min, that means for all pyrimidines. For all pyrimidine, the last term should end with din. That is cytidine. If it is thymine, it should be thymidine. If it is uracil, it should be uridine. Okay. If it is purine, these two are purines only, no? The purines are always end by sin. Adenosine and guanosine. Okay. So nucleoside structure we have understood. So how it made a bond with nitrogen base. Now these two combinations are there. These two are having a friendship. Now they wanted to make a friendship with phosphate also. So how this bond occurred? Let's see. So when these three combine together, we consider them as a nucleotide. Let's see the structure of nucleotide. Okay. So very simple structure once again. Let me draw sugar. So this is the sugar, right? Okay. The sugar is having OH group here. Here we are writing the structure of deoxyribose sugar. Now let me take anything. Either it's a purine or pyrimidine. Just I will take pyrimidine only. So both have combined. As a result, I saw a bond here. This is the N-glycosidic bond. Now let's take a phosphoric acid. Phosphoric acid means what? In this way shall I write? P, double bond O, here OH, here OH. Just now we have learnt while doing ester bond formation. Okay. Now this phosphate group says or phosphoric acid, I will give one H, you can give one OH. So both will react here. So that water molecule will get eliminated. That is once again dehydration reaction. So as a result, if I remove these two particles, the O should come in the center. So this bond, what you are going to see, this is nothing but ester bond. Ester bond. As it is forming between the phosphate as well as the nitrogen, sugar, it is called phosphoester bond. It is not diester. It is phosphoester bond. This is N-glycosidic bond. Simple. We have constructed a nucleotide itself. Okay. So now, once again, based on the nitrogen base, the phosphate remains same for every nitrogen base. So it could be of anything. It could be adenine, guanine, cytosine, thymine, anything. But sugar and phosphate remain same in case of DNA. But still, the name will get changed. How it will change? Look at here. If the adenine combines with sugar and phosphate, then we call it as adenilic acid. But before addition of phosphate, we called it as what? Adenosine. But now it is adenilic acid because phosphoric acid has been added. No, that's why. So like that, guanine sugar phosphate, it is called guanilic acid. If it is DNA, we consider it as deoxyadenilic acid, deoxyguanilic acid. So like that, cytosine, sugar plus phosphate, deoxycytidinic acid. If it is just RNA, just cytidinic acid. Okay, so thymine, sugar phosphate, obviously deoxythymidinic acid. Here we cannot say just thymidinic acid because thymine is absent in RNA. Okay, so once again the question can be framed. The thymidinic acid is present in RNA, DNA or both, something like that. You should know it is found only in DNA, not in RNA. Whereas the uracil, sugar and phosphate combine to form uridylic acid only. Here I cannot write deoxyuridylic acid because uracil is absent in DNA. Write down, uracil is absent. Or else I will write, in the place of thymine, uracil is present in RNA. Okay. So simple, we have understood the structure, bond formation within the nucleotide, between the components. Now, how these nucleotides are interconnected, because we need to construct a polynucleotide chain. So this polynucleotide chain construction is not given in NCERT, that is in class 11th NCERT, but it is there in class 12th NCERT. So let us learn that also. Anyhow, we have learned this much, then what is there to learn? Just one concept. Okay, let's learn it. Structure of polynucleotide chain. Okay. So actually I will tell you one thing. Okay, first I will show the structure. Then I will tell you about that. So let's write nucleotides here. First, let's write only the sugar. Right? So this is the first position. Here to this position only, who was attaching? Nitrogen base. Okay, so that's why even if you write in this way, then also it is okay. But here you should not write a phosphate in this way. Right? So I will tell you. First I will show the simple structure. See, instead of writing this much, if you write a ribose sugar, and to this ribose sugar, this is the first carbon atom, right? So if you just add a nitrogen base here, which nitrogen base you are representing, you can write that also. If you are adding adenine here, you can just mention A. If you are adding guanine, just mention G, C, T, anything. Okay. So this is nucleoside. Now, phosphate group you need to attach. So if you write phosphate like this is wrong. Because what does it indicate? It says that you are making bond with fourth carbon atom. But actually bond is not forming with fourth one. It is making bond with fifth carbon atom. Correct? So therefore, if you write like this is wrong. So what you have to do? Very simple. You just write a line here. Put a line. And then you write a PO something like this. This is the simple structure of nucleotide. So that is what I am going to show here in the next slide. Okay, to construct a polynucleotide. So just write down. So instead of writing all this line, shall I write like this? That would be easy for us. I will rub entire thing. Okay, now I will write A here. It means adenosine. To this first carbon atom, I will write G here, guanosine. To this first carbon atom, I can write T here, thymidine. Okay. Now, I can write phosphate group to this in this way. Okay, just put a line. Just write P, O, put a line, P and O. Okay, now the question is how we are going to connect these nucleotides together. So, let me take only two separately. So, let's consider. Here, let us not touch this adenine as well as the guanine. Anything you can take, any nitrogen base you can take. Let us not touch it. So, which carbon already has been occupied? The first position has been occupied and even the fifth one also been occupied. Okay. So, now only left one is second one, third one and fourth one. Fourth one you cannot take because it is almost near to this only. Now, imagine this is the third one. So, to this third group, OH group is there. Correct? OH group is that. Now, the phosphate group is something like this. I can write double bond here also. And one of the bond has been make a bond, phosphodiester bond has been made with this 5th carbon atom. Did you understand? See, here you can write double bond on the top or even at the bottom also. So, one of the OH group which was there here has been utilized. This OH and this H has been utilized for removing water and make a phosphoester bond. So, therefore, only this OH and this OH has left. Now, I am interchanging, that's it. O I am writing down and OH I am writing at the top. Okay, now, see, let me write OH here and another OH here. Okay, now, what happened here? This H and this OH will react. This H and this OH will react, and here they will remove one water molecule and again a bond formation occur. So, when a bond formation occurs, how it looks? Here it will be, this should be P, okay? Okay. So, here it will be double bond. Here it would be OH as it is. Now here the OH has been utilized. Only is O group. Correct? Now, I can write in this way also. In this way also I can write. That means it has been made a bond with the 3rd carbon atom of one sugar and once again the 5th one of the phosphate, the 5th carbon of the next sugar. So that one more phosphoester bond has been formed. So this is also phosphoester bond, this is also phosphoester bond. So together we consider as phosphodiester bond. Phosphodiester bond. Hope you are understanding what I am trying to tell. This is phosphodiester bond. Look at here. This is one ester bond. This is one ester bond. Single, this is only one ester bond. But now I made a bond with this also. Look at here. This also. Together phosphodiester bond. So like that you can construct a number of nucleotide. Number of nucleotide you can construct in a polynucleotide chain. So this is one polynucleotide chain. Okay, I will write here once again. Let me take 4 nucleotides. Let me join together. Just do along with me. It will come definitely. First, write down the sugar molecule. Okay, did you write? So here just write A. This should be G. Don't write B, C, D. Don't write A, B, C, D. This is A, G. This is C. This is G. Okay, G repeated. No problem. Even if it gets repeat also no problem. But for our convenience, let us take all different nitrogen bases. And here to the 5th one, we need to make a bond with this phosphoester bond. Correct? So let's make it. So we have constructed. This is N-glycosidic bond. Correct? And this is phosphoester bond. Phosphoester bond. And how we made phosphodiester bond between 2 nucleotides? We took OH from here and H from here. So as a result, we got like this, one more bond. Okay, I will take one other color. So this is another bond. This is also phosphoester bond. But between 2 nucleotides, we could able to get 2 phosphoester bonds. Therefore, it is called phosphodiester bond. Okay, here also one more ester bond, form phosphodiester bond. Here also one more ester bond, phosphodiester bond. So I got a complete one polynucleotide chain. But DNA is not just one chain. Let me show here. DNA is something like a twisted ladder. Something like this. So in this ladder, if you just open this structure, helix, it is something like a helical structure, right? If you just remove it, then it looks something like this. It looks something like this. 2 polynucleotide chains are there. So here one nucleotide, here one nucleotide. All this pink color, what I am writing, these are the nucleotides. Okay. And these are present upside down. It is having a polarity. Polarity in the sense, one chain what you are saying, it is having, let's imagine this is the head portion and this would be its tail part. Okay. And another chain what you are saying, their head will be here. It would be its tail part. That means another chain is upside down. Okay. So that's why to construct here one more chain, we need to make this sugar upside down. So as a result, it looks something like this. Shall I construct sugar in this way? Fine. So now if you just write this ribose sugar, deoxyribose sugar in this way, then here this would be the first carbon atom, this would be the second, this would be the third, this would be the fourth, and this would be the fifth. So in every fifth, we are going to write this phosphoester bond. Okay, so to this nitrogen base will be attached to the first carbon atom. Now how will you construct the phosphodiester bond? In the same way, here it will be the third one. So you just construct in this way. So this is another chain we got. So remember one thing, a scientist called Chargaff, when he was observing the polynucleotide chain, he came to know that wherever adenine is there, the opposite chain is always having thymine. Wherever guanine is there, the opposite chain was having cytosine. So here cytosine, always opposite is guanine, thymine, always adenine. So they are vice versa. Okay, and they are also not present simply like this. Here I have shown like a ladder. Okay, so here it is something like a ladder. So this ladder, when you just twist like this, it comes in this way. That's why DNA is called twisted ladder. Okay, so what does it indicate, twisted ladder? It is because the hydrogen bond present between them. So between A and T, 2 hydrogen bonds are present. Between G and C, 3 hydrogen bonds are present. Again G and C, 3 hydrogen bond. Again A and T, 2 hydrogen bond. So this is the structure. So now when you just look at one chain, look at here one chain. So it has started from one nucleotide, right? So in the first nucleotide, this is the 5th position. So in the 5th position, the phosphate group is present. Therefore, it is called 5' phosphate. So above this, no more nucleotide. So this is the first one. So like that, when you just keep going down, here it has been end in the 3rd position where OH group is there. It is not making bond with anybody else. So therefore, this place is called 3' OH. 3' OH. So that is what I said here. This is having the phase at the top. This is 5' phosphate group. And this one, the tail part, this is 3' OH group. And now, another strand, it is upside down. Correct? Let's go back. Upside down. Why? Here, if you look here, the 3' OH is at the top. So therefore, this is 3' OH. And like that, if you just come down, here this is the 5th position, having the phosphate group. This is called 5' phosphate. Understood? So therefore, the DNA, in DNA, the 2 chains are having anti-parallel polarity. They are having anti-parallel polarity. This is very important. So how these structures has twisted like this? And if you twist in this way, how many nitrogen bases we are going to get, ma'am? So everything that we are going to learn in class 12. Don't worry. And here if you just observe carefully, let's go back again. If you consider this entire structure up to here as the backbone of a ladder, the phosphate as well as the sugar, they are present at the back. Okay, these nitrogen bases are moving inside. Correct? So shall I show in this way? Here, this is the sugar which is having phosphate. Everywhere sugar is there. Let's imagine everywhere sugar is there. This sugar is connecting to this phosphate. This sugar, everywhere phosphate is there, like this. Phosphate is connected by phosphodiester bond. Correct? Here also, in other chain also, sugar is there. Phosphate is connecting. Now, if you just look at the nitrogen bases, they will be present. If it is adenine, here it would be the thymine. If it is thymine, it would be adenine. If it is guanine, it would be cytosine. If it is cytosine, it would be guanine. Are you getting my point? Thymine, adenine. So, here they are projecting inside. So, therefore, sugar and phosphate, they act like a backbone. They are a backbone of polynucleotide chain. So, this much is enough for class 11th. Of course, you are going to learn in detail in class 12th. There is a separate chapter called Molecular Basis of Inheritance. If you are a class 12 student, you know about this. Okay. So, let's solve few MCQ. This much is enough about the nucleic acid. That is DNA. Okay. So every time I keep repeating why biomolecule chapter is difficult, because the information has been shuffled like anything. They have given a bond formation of all the organic molecule in one side and every macromolecule on separate side. Separate, that is proteins, carbohydrate, nucleic acid separately, bond formation separate, monomers separate. So if you are a master, if you could understand everything, then you could connect the link, or else it creates confusion in your mind. So that's why, just to avoid such confusion, I am taking the parts from each topic which has been mentioned in NCERT and I am grouping it as one topic. For example, if it is a nucleic acid, bond formation, monomer, polymer, everything I have done at a time. Then I will move on to the next organic molecule like a protein or a carbohydrate. Like this I am doing. So that once we have finished the chapter, then if you read NCERT, you could be able to understand. Okay. So now let's read this part. Again, it is there in NCERT only. The living organism have a number of carbon compounds which are heterocyclic ring can be found. Some of these are nitrogen bases. Okay. Adenine, guanine, cytosine, uracil and thymine. When found attached to a sugar, they are called nucleoside. Okay. If a phosphate group is also found esterified, see, ester bond, to the sugar, they are called nucleotide. Adenosine, it is a name, adenosine, guanosine, thymidine, uridine, cytidine are nucleoside, important. Okay, like that, adenilic acid, thymidilic acid, guanilic acid, uridilic acid, cytidilic acid are nucleotides. Nucleic acid like DNA and RNA consist of nucleotide only. You can take either it is a DNA or RNA, they are made up of nucleotide itself. DNA and RNA are function as genetic material. So in general, they have given, they are not talking about RNA acts as a messenger. Again, that you are going to learn in class 12. Okay. Yes, nucleic acid. Separately, NCERT has given about nucleic acid. As you notice in the figure 9.1, if you open textbook, till now what I have constructed the heterocyclic ring as well as nucleoside, the same thing has given. The heterocyclic compounds in the nucleic acids are the nitrogen bases named adenine, guanine, uracil, cytosine and thymine. Adenine and guanine are substituted purine, important. While the rest in the sense, cytosine and thymidine are substituted pyrimidine. The skeletal heterocyclic ring called purine and pyrimidine respectively. That means these two are purine, these two are pyrimidine. The sugar found in polynucleotides are either ribose, that is monosaccharide pentose, or deoxyribose. That means why they have shown 2' here. In the second position, they have deleted oxygen. That's why. A nucleic acid containing deoxyribose is called deoxyribonucleic acid, while that which contains ribose is called ribonucleic acid. Simple. What is there to learn here? That's it. NCERT has given information about nucleic acid. That's it. But we have learned a lot. But it helps you a lot even in class 12th also. Let's solve your MCQ. Nucleic acids are, it means only nucleotides. Nucleic acid means they are polynucleotide only. We won't say nucleoside. Because if there is no phosphate, first of all, we won't consider them as nucleotide only. So therefore, all these options are wrong. Next one. A heterocyclic compound in the nucleic acid. Which one is heterocyclic? Nitrogen base. Because name itself is telling I'm having nitrogen along with carbon. Okay. So usually in an organic molecule, we are going to see this carbon and hydrogen. But here, this ring is having nitrogen. Adenine and which one? Adenine and guanine are substituted. Just now we read. These are substituted purine. Where it is? Cytosine is given. Guanine substituted. Guanine and substituted. This is the answer. Not uracil. Guanine is there but unsubstituted given. The sugar found in polynucleotide is either ribose. This ribose is monosaccharide. Correct? Or deoxyribose. That means at second position, we have deleted oxygen. It could be deoxyribose. The same line picked directly from NCERT. Here, reverse has given. Here also, disaccharide given. Wrong. Disaccharide. Wrong. It is a monosaccharide. Ribose sugar. In the nucleic acid, phosphate link. Phosphate. 3 carbon of both sugar. No. One sugar contribute OH from third carbon. Another sugar contribute once again OH from, that is two sugar. One is from 3', another from 5'. So 3 carbon of one sugar and 5 carbon of other sugar of succeeding nucleotide. Then only we could able to get phosphodiester bond, right, in a nucleotide. Okay, this is right. 5 carbon of one sugar and succeeding sugar, wrong. 5 carbon of one sugar and 3 carbon of other group of succeeding nucleotide given, wrong. They are telling the phosphate link. Choose the correct nitrogen base pairing in DNA. So AT correct, here also AT correct, AU is wrong. But here two hydrogen bond should connect. A hydrogen bond should make with each other. Therefore, this is our right answer. Here triple bond has made. Triple bond will come only between guanine and cytosine, not between adenine and thymine. Okay, that's it. Now let's move on to the last part, that is enzymes. Okay, now let's talk about one more important topic, that is nothing but enzymes. So while talking about proteins, one thing it is very clear that enzymes are nothing but the proteins, that we have understood. But NCERT has given one line. What is that? Almost all enzymes are proteins. Almost all enzymes are proteins. Just look at the twist here. Almost all enzymes are proteins means what? It says not all your proteins are enzymes. Almost all proteins are enzymes means there are certain enzymes that are not proteinaceous in nature. So that is what the meaning of this sentence. It is important. Sometimes if the question framed in the form of statement format, then you should know almost all enzymes are proteins. Suppose instead of this, if the question comes or if the statement comes, all enzymes are protein, then that statement is wrong. Okay? Then which are the enzymes which do not belong to the protein category? Okay? So for that, we need to name two enzymes name. Look at here. There are certain enzymes like ribonuclease. Easiest topic, just focus here, you will understand. Ribonuclease as well as ribozyme. So these are the two enzymes. Like that, there are few enzymes which are actually nucleic acid. So nucleic acid means what? DNA, RNA, just now we have studied. So though they are nucleic acid, but they behave like enzyme. Then how can you say that these are protein? So therefore, there are certain exceptions to this line. That's why this statement says that almost all enzymes are protein. Okay, we do agree for that. Now the question is, enzymes, suppose if we consider enzymes are protein, except few exceptions. Okay, let us leave those exceptions. But if I just consider the proteinaceous nature of enzymes only, then which structure the enzyme could be present in the nature? Once again, that also we have understood shortly. We have learnt about this, right? Few minutes back we have learnt. So this is the primary structure. So in primary structure, the enzymes are not available. They do not do any work. And suppose if they twist like this, it is nothing but a secondary structure. In this structure also, the enzymes are not available in nature. Then in which format? Of course, you are right. Let me take some other color. It is in a tertiary structure. Look at here. Tertiary structure. So here, while constructing tertiary structure, if you observe carefully, there are certain pockets like structure has present, while it is twisting upon itself, just like a wool. Okay. So here, few regions are going to show a crevices. Crevices are nothing but a pockets. Okay. But all crevices do not helps to hold the substrate. I will tell you what is the meaning of a substrate. There is only one side that acts. Of course, there are so many pockets are there. So many crevices are there. But there is only one pocket which acts as an active site. So because of this active site only, the substrate molecule can go and combine. Now the question is, what is substrate molecule, ma'am? Let me tell you one thing. Look at here. In our body, what are all the chemical reactions which are so taking place? They are all because of enzymes. So you know the difference between physical process or physical reaction and chemical reaction. Suppose if I have take a water in a tumbler or in a vessel, If I just boil it, it will evaporate. Okay, so that is a physical process. Water which was in a liquid form, it got evaporated, it turns into gaseous form. So like that, if the same water, if I just place it in a freezer, it will condense to become a ice cube. That is nothing but it has changed state of matter. That is just a physical process. Here there is no breakage of bond. If I just look at the water molecule in the ice, the water remains water only. If I just test these water molecule even in the liquid state, it remains water only, and even in the vapor state also it remains water only. So these are the physical process. Suppose if I take some example, say for example H2CO3, carbonic acid. Okay, this is carbonic acid. You are going to learn about this in breathing chapter. There I will teach you, don't worry, but you just know. This is carbonic acid, this is one compound, right? Now imagine this compound has subjected for chemical reaction. So chemical reaction means what? Here the bond which are so present. Hydrogen means the hydrogen are attaching like this. Okay, so to the carbon molecule, So, okay, I won't write anything. Let's imagine something it is present like this, okay? It is somewhat like this. Let's assume. Now here the bond is there, correct? The bond is there. This bond, when it get broken down, then obviously all the molecules which are so present like a carbon, hydrogen, oxygen, everything has got separate, correct? That means the breakage of bond when occurs in a molecule, that is nothing but a chemical reaction. So such chemical reaction usually occur in our body with the help of enzymes. So as a result, here what happens? H2O form and CO2 has formed separately. So when you combine this water as well as carbon dioxide, again we are going to get this carbonic acid. So I'm just giving this as an example. So like such many reactions are happening inside our body. We are going to have food. Right? That is glucose. So glucose is C6H12O6. So it has to break, correct? C6H12O6, all these are combining together with the help of bond only. So we have learned that also while studying the carbohydrate. So these has to get broken down into small pieces. Then only we are going to get energy. That means if I want to summarize everything in a simple way, I can show in this manner. Suppose if there is a material called A, let's assume a material called A, if it is get broken into B and C, I can consider these two are the product. These two things are nothing but the product. How these products are forming? It is forming by a substance. This is nothing but a substrate. Okay, that means if any chemical reaction, if it is undergoing for breakage process, that also takes place with the help of enzyme. Or there are certain reactions which are happening inside our body where materials are going to become joined together. For example, let me take X and Y. X and Y, these two are the substrate. So these two substrates are joining and they form a material called Z. So this is a product I got finally in my body. It is just an example I am telling. There is no such Z product, X product like that. There is a specific name for those products. Just to make you understand, I am just generalizing it, okay? So here the materials are combining. Here the materials were breaking, right? So here also for such reaction, we need enzyme. So that means whatever the reaction, chemical reaction or metabolic reaction. So these are nothing but a metabolic reaction, which are happening inside our body that takes place with the help of enzymes. That means enzymes are so important, correct? So how this substrate molecule is going to get act with the help of enzyme? For that, enzyme required a special site, a special pocket-like structure that is nothing but active site. So here what happened? I will take separate slide to show you here the action of enzyme. Let's imagine this is our enzyme. It's a tertiary structure of enzyme. So here is also crevices, pocket is there. Let's imagine here is also one pocket, but here this is the active site. This is also pocket, but let us consider this is the active site. Okay, so let me mark it as the active site of this enzyme. So to this active site, specific substrate comes and bind. Imagine here is a substrate. Let's consider this as a substrate. Okay, it's fit nicely in this active site. Just like how we are going to put a key, proper key into the lock, in the same way, to the active site, a specific substrate only should come and attach, or else it won't. Suppose if you take some other key to open the lock, how it open? It won't, right? There is a specific lock for specific key. There is a specific key for specific lock. Okay, in the same way, there is a specific active site or an enzyme for a specific substrate. So let me consider this as a substrate molecule. So when substrate comes and combine to this enzyme, then we are going to mention this process with an equation like this. Enzyme combining with the substrate. This is the first step in the reaction. And soon after combining, what happened? It will enter into the second step. In the second step, what happened? The enzyme will binds to the substrate properly with the help of certain bond, certain linkage. Okay. So imagine there are certain specific bonds are present here. Certain specific bonds are present and they are just holding the substrate properly in this way. So we call such a combination as enzyme substrate complex. This is the second step. This is the second step. E stands for enzyme, S stands for substrate. Okay. So let me mention it as E, that is enzyme. So here S is nothing but substrate. Right. Now what happened? After combining this, the enzyme starts chopping it into small pieces in the sense the bond which are so present in the substrate, no? As I have told in this example, there is a bond present inside with all these molecules. That bond will get broken up. Okay. So because of that, the substrate become into two pieces. Okay, either it can be combined by joining the bond or it can break the bond. Let's assume here this enzyme is responsible for breaking the bond. Now as a result, what happened? Let's assume like this. Here it has broken into two pieces. Two pieces, something like this. Right? So that is the third step. Enzyme product complex. This is the third step. But still, the substrate is attaching to this enzyme only. And finally, after the breakage, it will comes out. So this is one substrate molecule, this is another substrate molecule, something like this, let's assume. So finally, we consider them as a product. Are you getting my point? So here is the fourth step. Enzyme plus the product is going to be formed here in the fourth step. In the fourth step. Okay. So this is what the nature of the enzyme. Understood? So here it could be metabolic, it could be catabolic or it could be anabolic. I will tell you later what is the meaning of this catabolic or anabolic. Okay. So hope I'm very clear about the nature of enzyme action. Right. So hope you are writing the points. So till now what we have learned. Let's read in NCERT. Enzymes. Almost all enzymes are proteins. Statement based question. Okay. There are some nucleic acid that behave like enzyme. These are called ribosome and even ribonuclease also comes under this category. One can depict an enzyme by the diagram. Okay, so the diagram I have not shown, but let's read this point first. An enzyme, like any protein, okay, any protein you take, if you consider it as an enzyme, so if it has a primary structure, that is amino acid sequence of the protein, an enzyme, like any protein, has a secondary and tertiary structure. That means what they are trying to tell is, you have seen the protein structure, right? So we have seen the primary structure, secondary structure, tertiary structure, everything. So you won't consider primary structure, secondary structure as a protein. Because first of all, these two structure of a protein do not involve in biological activity. So that's why which one we will consider? Of course, that is a tertiary structure. That is what the meaning of this line. Okay. So when you look at the tertiary structure, look at here, tertiary structure, figure 9.3D, already we have seen that. You will notice that the backbone of protein chain folds upon itself. The chain crisscrosses itself and hence many crevices. or pockets are made that I have told, no? One such pocket is the active site. An active site of an enzyme is crevice or a pocket into which the substrate fit. Thus, enzymes through their active site catalyze the reaction at a high rate. Okay, so enzyme catalysts differ from inorganic catalysts in many ways. So here what they are telling, so you have learned chemistry. Okay, so you know very well about chemistry than me. So in chemistry, we are going to study the inorganic molecule, organic molecule, everything. Here also we are studying organic molecule only. So in inorganic reaction also, if you take calcium as well as the chloride to become a calcium chloride. So in such reaction also, I'm just taking an example, I'm poor in chemistry actually. So while such reactions are going on, if we use a catalyst, imagine we are using a catalyst, then catalyst, what it will do? It will speed up the reaction, right? Though the catalyst will not begin the reaction, but it will speed up the reaction. It won't make any changes, just it make the reaction to get faster. But the advantage of inorganic catalyst is that they can work better in very high temperature. If you increase the temperature, they can do well. But in our body, the enzymes are also acting like a catalyst. But those catalysts, we consider them as what? Biocatalyst. Remember this name also. This line is important. Enzymes are biocatalyst. How inorganic reactions are taking place with the help of a catalyst, like that in our body, there are so many metabolic reactions are taking place with the help of this biocatalyst. This biocatalyst is nothing but the enzyme. Okay, but for this enzyme to act properly, there should be a specific temperature. Suppose in my body in the sense in all human being body, the temperature is 37 degrees centigrade. Of course, enzymes are there. Enzymes are there. Because of the enzymes only, the food molecule will get break into small pieces, the digestion process occur. Correct? We are going to eat the carbohydrate. That carbohydrate has to broken down into small pieces. It will convert it into glucose. Okay, so for those things to occur, the body temperature should be normal only, 37 degree. Suppose if the temperature become too high, if it is become 40 degree, 45 degree, what happen? It will get denature. It will become omelette. Okay, that means you cannot regain back. So that's why whenever we get a high fever, immediately we are going to take the tablet. Why? Because we should not allow our body proteins to get denature. Okay, so if the protein get denatured, we cannot regain it back. So we need to wait once again for more time to our body to synthesize those enzymes or proteins back. Understood? So that's why they are telling many ways, but one major difference needs mention. Inorganic catalysts work efficiently at higher temperature and high pressure, while the enzymes get damaged at high temperature, say above 40 degrees centigrade. And from here, what the informations are given, just make a note in your textbook. This much information, what it is given. This one I am going to tell in factors affecting, there is a topic called factors affecting enzyme activity. So in that topic, there is a part called temperature. So in this part, I will explain these 2 points. Okay, then you will understand better. Now let's move on to the nature of enzyme. Nature of enzyme action. Nature of enzyme action. Till now what we have learned, the same thing has given. Each enzyme, that is E, has a substrate binding site in its molecule so that highly reactive enzyme substrate complex is produced. Okay, this complex is short-lived. That means it is not going to hold the substrate for a long duration. It is a short-lived. Immediately it is going to break the substrate. That is why it is combining. And dissociate in the sense it will break into its product P and unchanged enzyme with intermediate formation of an enzyme product complex. It means what here, when this molecule has get broken down, when it got broken down, so this piece has got removed. Imagine there is no substrate at all. Now this enzyme is ready to receive one more molecule, one more substrate. So like that, it will just speed up the reaction. Here the substrate is getting changed into product. This substrate is changing into product, but enzyme remain the same. Enzyme is not getting affected. So enzyme, if I am an enzyme, I am just making into pieces. I am remaining same only, but only I am changing the substrate into product. So again, I am waiting for one more substrate to come. Again, I will chop it, I will send it back. Again, I will get one more substrate, I will chop it, I will send it back. So like that, enzyme remain calm, enzyme do its action faster, but it won't undergo any changes. So that is what the meaning. Yes. The formation of enzyme substrate complex is essential for the catalysis. So here, E plus S, enzyme plus substrate. ES means enzyme substrate complex, enzyme product complex. Finally, we are going to get enzyme plus product. So here the 4 steps are given. So it may come in your board exam, but here mainly we are focusing on NEET. Okay. So if you are even focusing for board exam also, we can write down for 5 marks, this question may appear. Okay. So what is the first step? The substrate binds to the active site of the enzyme, fitting into the active site. So this already we have understood. The binding of substrate induces enzyme to alter its shape, fitting more tightly around the substrate. It means certain bond has created so that it has to fix properly in the active site of an enzyme. I told you while mentioning certain bond, certain changes, the same point they are given. The active site of the enzyme now in close proximity. It means enzyme substrate complex, which is so formed, that is what they are telling, of the substrate breaks the chemical bond of the substrate and the new enzyme product complex is formed. So this point. And finally, the enzyme releases the product of the reaction and the free enzyme is ready to bind another molecule. Now enzyme is ready. Come on, another molecule, I will break it. That means substrate run through the catalytic cycle once again. That means this process keep going, keep growing. Now the question is, why do we need enzyme? Why do we need enzyme? Suppose if you don't have enzyme, what would happen? I will give simple example. Let me take the same old example of digestion. So when you eat food, when you have a meal, it will store it in your stomach for 4 to 5 hours and finally it will get completely break down, okay, after 4 to 5 hours. That means our food will get broken down completely around 4 to 5 hours. So that is what we can say. For all these things to happen, of course, we require enzyme. The enzymes from the enzyme from salivary juice. There are certain enzymes present in the stomach. There are enzymes present in the pancreatic juice, intestinal juice. All these enzymes are acting on the food. And finally, they are breaking the food into small pieces. So that's why we are getting a proper digestion. Suppose imagine if there is no enzyme at all in our body, what would happen? What do you think that our food digestion occurs? How long it might have take to digest the food? Do you know? It's more than a year. So just think, if you don't have enzyme, then every process, every metabolic reaction which is happening inside the body, everything will get slowed down. So here, one typical example is given in our NCERT. So what they are telling, we know our RBC. This is RBC. So we are going to learn about this in breathing chapter. There is a reaction occur inside the RBC to carry the carbon dioxide towards the alveoli. Because whenever we breathe out, no, we are going to release carbon dioxide. So this carbon dioxide has to come from different tissue, different parts of the body, and finally it will come near the lungs, we will breathe out. Okay. So during this process, certain reaction takes place in the RBC. So one such reaction I am writing here. Right. So here what happened? The same old example. Carbon dioxide, which is combining with water. Right. And it will changes into carbonic acid, H2CO3. This is carbonic acid. So carbonic acid will come when these two molecules has combined. So for this process to happen, for this process to occur, our body require an enzyme called carbonic anhydrase. Carbonic anhydrase. This is the name of the enzyme. Okay. Now, imagine this enzyme is not there. Okay, let me write without enzyme. What happened? Without enzyme. Of course, this reaction definitely would take place, but only 200 molecule of H2CO3 in the sense carbonic acid will form per hour. That means in 1 hour, 200 molecule of H2CO3 is forming if there is no enzyme at all. Suppose if you involve the enzyme with the enzyme, suppose if enzyme has involved, then you may get shock by looking at the number. 600,000 molecule of H2CO3 will be formed not within 1 hour, within a second. Just within a second, we are going to get 600,000 molecule. Just imagine how speed this enzyme will do and what a magic it will do. Okay, it means the enzyme, I will write here, the enzyme speed up reaction 10 million times faster. 10 million times faster. So this is important line. This example is itself is important. These lines are important. All these are NEET questions. Important. Write down in your copy. If you have a textbook along with you, just underline it. Okay. So that much important enzyme is. Now the question is, okay ma'am, we do agree. The enzyme should be there in our body. Now the question is, how it is possible ma'am? How this enzyme is going to do such a faster process? I mean, reaction to be occur so faster. Is there any magic? Is it going to do? Of course, it is going to do certain magic. So how it is doing? So let us understand that process in a simple analogy. Okay. Let's imagine how do enzyme increases the speed, right? Okay, I will show here. Imagine, because it is quite confusing topic. If you understand this example, you can understand better how this process will take place. Look at here. Imagine a boy. So we have given a task to this boy. What a task? Look at man, you have to reach this target pool. Okay. So here is a flag. You have to bring it. So this is the target site. You have to bring this. So what he has to do? He has to run. He has to run. But the problem is he has to face a lot of obstacles. Imagine here is a rock. He has to, so many rocks are there. He has to remove all those rocks. And once again, he move forward. Again, there will be a big gate. So he has to cross this gate. So once again, there might be certain, what will say? Okay. Certain sand particle, granites, all those things, they are present in a large quantity. He has to remove all those things, or else he will get scratched there if he just fallen down. So like that, he will get a lot of obstacles. But final aim is to reach the target. So at anyhow, he has to cross all this barrier and finally he has to reach the target. So for this, he needs some energy, right? He needs energy, of course. So imagine there is some power. Look at here, this is not the way to tell, but what to do, I need to make you understand. No? There is no other way, that's why. Imagine there is some energy, some supreme energy he is going to get. So finally, he becomes so stronger. And he is running, he is just lifting all those things, he is just removing all these obstacles and he finally reaching this target. Okay. So even though he got all the energy, listen to me carefully, even though he got all the energy to do this task, but while doing this task, the energy of course will get waste, right? In the form of a heat, he is going to sweat out. The energy will be released out. How much energy is gaining? He is going to release those energy in the form of a sweat, or that muscular energy will be lost. So finally, he will reach the target. Imagine, for this process to occur, he may take around 1 hour. 1 hour. Did you understand this much? Now, in this place, now a friend come. Imagine his friend will come near to this place. So he is the friend. So he's a happy guy. Let me name him as an enzyme. His name itself is an enzyme. His friend name is enzyme. So what he will do? You don't worry, my friend. I will help you. So you will run. So your work is to run. You just do that work. But what I will do? I will remove all these things. All these obstacles which were so having in this way, I will just remove all these obstacles and so that I will clear your path. So that you will reach the target so earlier. So for that, do you need that much energy? How much you are actually getting in the beginning? So no need. So even though you have a small amount of energy, you can run, you can reach the target. So that is what the function of enzyme. Okay. So what the energy is getting, So we can consider this as a activation energy. Activation energy. So when the enzyme has entered in the life of this guy, say his name is X, then automatically what happened? Activation energy. How much amount he was getting, no? That much amount is no need. So even though he is having little bit activation energy, the task can be completed. So this is what the simple thing we need to understand. The same thing we need to understand through the graph that will come in your exam, NEET exam. Just look at here carefully. How do enzyme increases the speed? So for that we need to plot a graph here. So let me mention it as a y-axis and this as a x-axis. And let me name this as, so in y-axis, let me write it as energy. Actually, this energy is nothing but a potential energy. Let's name it like this. Now, one more information I need to give. What is that? So in nature, listen to me, though it seems to be quite confusing, but if you understand the concept, you will learn it. Imagine there is a substance A. There is a substrate A. So let's assume this substrate is having around 10 calories of energy. 10 calories of energy it is having. Now, when it is undergoing into change as a product, when it is changing into product, then it may lose the energy so that this product, let me consider it as a B as a product. So this may have only around 5 calories. Then where is the remaining 5 calorie? It might have lost in the form of a heat. So that rest of the calorie difference was there, no? It has lost in the form of heat. So such reaction, we call it as exothermic reaction. Exothermic reaction. Exo means out. Therma means heat. So heat is going out. So that's why it is called exothermic reaction. Okay. But in the nature, every reaction is not exothermic. There are certain reactions, say for example, A only, if it is a substrate. Okay. Now it is converting into B as a product. So during this time, what happened? Imagine if it is having 10 calories of energy and at the time of becoming a product, it may around 20. It may around 20. So here what happened? Some amount of energy will be gained in the reaction. It will become in the reaction. So such reactions are called endothermic. Endothermic reaction, and here we are focusing on exothermic reaction. Okay, let us talk about exothermic reaction. Now, let's consider, here is a substrate. Here is a substrate. Let me consider this substrate as S. Now this S, let me write here, this is the S position. This is the position of S. Okay. Now let's assume this S is changing into product. Somewhere here it has to change into product. Okay, here is the product level. So usually what happens? The substrate, look at here carefully. Important. The substrate, whenever it is getting changes into product like this, the energy will be released out in the form of a heat. That means we are talking here the exothermic reaction. Exothermic reaction, right? But just recall, just before starting this graph, what I said, a boy, even though you want to turn into product, imagine this product is nothing but a target site. This substrate is a person, is a boy. Okay, to change into product, he needs certain energy. That energy is called activation energy. So let me show that activation energy. Let me rub this point. This graph it make you confused. Let me rub this line. Okay, just understand this much. But you should know, of course, some amount of energy will be released into heat. But before converting into product, first it will rise. The substrate will gain some extra amount of energy. Okay, let me show the same thing again. This is the substrate. He is getting some extra energy, and finally it will reach into product. Okay, finally it will reach into product. So why it need extra energy? There, while giving the example, I said there are so many obstacles. Here also in the reaction, what happens? There are certain obstacles will come. Imagine this is a substrate. Say for example, this substrate is X and there is, okay, something like this. This is a substrate. There is one more substrate. Imagine it's Y. Okay. So now this X and Y, they are entering into the reaction. So finally they have to combine. So during this process, it is not simply as we are seeing that, oh, this X will come, this Y will come so that it will combine. It won't happen. Okay. So they don't know where is the target site, how to combine. So sometimes it may attach like this. Oh, I forgot. I shouldn't attach like this. I have to combine in this way. Okay, my energy got wasted. And now this substrate will come. It will combine here with this. Again, it got failed. I should not attach like this. So sometimes both will come in this way. So in this way. So they are going to collide each other. So during this process, they will lose lot of energy. And finally, because of so many permutations and combinations, they are going to attach in this way. So that X and Y, they combine and this will nothing but a product. But during the formation of the product, the so many energy which are so combined, no? So during this phase, it started showing so many transitional state. Are you getting my point? Instead of attaching this, it become in this way. This is one state. So again, oh, I lost my energy. Again, this came, it detached. Again, this came, it came attached in this way. So this is another transition state. So again, both will decide, okay, this is not the way to attach. So let us turn like this. Let's attach. Oh, this is also not the way. So like that, while doing such process, so many steps, what you are seeing, no, we call them as transitional phase. So here also, in this process, while getting the activation energy, that means the extra energy what they took, so that energy will get wasted for these substrate to change into different forms till it reaches to the product form. Okay, so these different forms, we call it as a transitional phase or transitional state. So finally what happened? Ultimately it has converted into product. So that is what we need. During this process, of course, the energy will be lost in the form of a heat. Okay, so all these things are happened when there was no enzyme. When there was no enzyme at all. So this much energy was consumed by the substrate. When there is no enzyme, there is without enzyme. Now, in such cases, a hero came here. So who is hero? He is the hero. So this hero will come. He is nothing but, he is the enzyme. He says, why you guys are wasting the energy? I am going to combine you guys. So you are in this way, you are in this way. Just come, let's attach. Okay, if he do like this, then obviously the amount of energy will be minimized. So the amount of energy which was so required for showing different transitional state, that was not required now, because he is helping in order to combine these substrate. These are the substrate. So to finally convert them into product. So how I can represent here? The same graph, if I have to show, then I can show something like this. Finally, they are also converting into product. But here, the activation energy is not required this much. Very little activation energy is required. Why? Because enzyme is there. Of course, ma'am, why it required activation energy, ma'am? Because enzyme is there, no? It can attach. Of course, yes. But the thing is, it should have its own energy, no? At least for combination, at least for converting into product. So that much energy, which is so required, that is also activation energy only, but it is very least. Even it is having, that much is enough. So this arrow, what I am showing, this is nothing but in presence of enzyme. Understood? So this is about activation energy. Okay? So this much energy, what you are seeing, this graph, these things, these arrow mark, everything is nothing but activation energy. Without enzyme, the activation energy is very high. It is very high. In the presence of enzyme, the activation energy is very low. Got to understand? So very important. Definitely it will come. Sometimes the question may comes to mark here just by making it as X. What is this? Whether it is in the absence of enzymes or presence of enzyme. You should know. If the peak is very small, then it is presence of enzyme. If the peak is very big, then it is absence of enzyme. So, or even sometimes the X as well as the Y position may come in order to mark. So, you should know it is a progress of reaction, how the reaction is going in this way. And here, how much energy is consuming in this direction. So, you should know this. Okay. Now, let's read NCERT part. So, hope I am very clear about this concept. If you won't understand, please do rewind the video and just read it once again. Actually, in this paragraph, they are not explaining much about the activation energy. Actually, that is in the other part. But here only they are explaining brief about the active site and all. But let's read, no problem. How do enzymes bring about such high rates of chemical conversions? Okay. To understand this, we should study enzymes a little more. We have learned a lot more. We have already understood the idea of an active site. Yes, we did understand. The chemical or metabolic conversion refers to reaction. That is nothing but metabolic reactions only. The chemical which is converted into product is called substrate. That means the substrate itself is converted into product. Hence, enzyme, that is a proteins with three-dimensional structure including an active site, convert a substrate into product. Symbolically, this can be represented as S changes into P. So, nothing is there. Simple paragraph. It is now understood that the substrate S has to bind with the enzyme at active site, correct? Within a given cleft or a pocket. That means the place where it comes and hold. The substrate has to diffuse towards the active site. That means it will combine. It is just explaining the enzyme substrate complex only. Nature of enzyme action only they are explaining. There is thus an obligatory formation of enzyme substrate complex. Obligatory formation means, imagine this is an enzyme. This enzyme is having like such active site. Now, let's assume here is the substrate which is coming and combining in this way. Suppose if the enzyme is in this way, let's imagine this is what the structure of active site. Then if such enzyme come, substrate come, then it is not going to fit here. Then the substrate should be in this way only. Then only it will fit here. Okay, that is what they are telling. E stands for enzyme. This complex formation is a transient phenomena. Transient phenomena in the sense it will again come back. It will not attach permanently there only. During the state where the substrate is bound to the enzyme active site, a new structure of the substrate called transition state structure is formed. So many transition state it has formed. No, finally it will give. So very soon after the expected bond making is completed. So here they are explaining just enzyme action only. Whether it is a breaking or a making, few minutes back, what I said, the same concept they are explaining. The product is released from the active site. In other words, the structure of the substrate gets transformed into structure of product. The pathway of this transformation must go through so-called transition state structure. So now they are explaining actual part. There could be many more altered structural state. That means just now I gave the example, no? It may, instead of combining like this, it may show in different state. That is called transition state. Between the stable substrate and the product. Before forming the product, it may show into different state. Implicit in this statement is the fact that all other intermediate structural states are unstable. That means even though they are going to form in different state, transitional state, they are unstable. It is not, if it is formed in this way, we won't consider it as a stable product. Again, it is going to get detached because this is not the final product we need to form. Stability is something released to energy status of the molecule or the structure. Hence, we look at this pictorial through a graph, it looks like something as in figure. That means they are explaining the activation energy without enzyme. Here, this small peak telling activation energy with enzyme. Here is the substrate. Here finally they are reaching the product. Here the energy which is so comes out in the form of a heat. Okay. So here the y-axis represent the potential energy content. X-axis represent the progression of the structural transformation or the states through transition state. That means here different transition state they are going to form. No, that is they are explaining. You would notice two things. The energy level differences between the S and P. Energy level S and P. You can see the difference. Here it is and the product is here. Okay. P is at a lower level than S reaction is an exothermic reaction. Okay. One need not to supply energy by heating in order to form a product. So here we need not to heat the substrate to get heat energy because they are going to release of their own because it is an exothermic. However, whether it is an exothermic or spontaneous reaction as an endothermic or energy requiring reaction, that means any reaction it could be, the S has to go through much higher energy state or a transition state, whether it is an endothermic or exothermic, whatever. But finally, it has to gain some energy, right, in order to convert it into product. That is, in order to show transition state, it requires activation energy. The difference in average energy content of S from that of this transition state is called activation energy. Okay, this is what they are telling. Enzyme eventually brings down this energy. That means energy is not required that much. Barrier making the transition of S to P more easily. That means that much barrier will not be made in the presence of enzyme so that it can easily convert it into product. So hope I am very clear. So here in this graph or here in this whole picture, you need to understand the activation energy. Without activation, without enzyme, with enzyme, x-axis, y-axis, exothermic, endothermic, these things. That's it. Okay? Yes. Now, let us understand. If you have understood this concept, almost we have understood most of the things from the enzyme part. Now we need to learn what are all the factors affecting an enzyme activity. If an enzyme has to work properly, if it has to show a reaction in a faster rate, in a brilliant way, then which are all the factors affecting it? Okay, so that factor we'll learn one by one. Okay, now let's see what are all the factors affecting this enzyme activity. So if the enzyme, if it has to work properly, then mainly three factors affecting a lot. The first factor is temperature. The second one, pH. Third factor is substrate concentration. So now let's study one by one. First, let us talk about how temperature affect an enzyme activity. So I won't read this line immediately. First, let me explain what exactly this temperature influencing, how exactly this temperature influencing enzyme activity. So imagine, let me take an example of our body only. So our body temperature is 37 degrees. Everybody know that. Okay. So in this 37 degrees Celsius, all enzymes do work properly because they know that we need to adjust to this body temperature only. So therefore, they started adjusting. They started working properly. Because of that only, whenever you have a food, it will get digested properly because of the action of the enzyme. If there is no enzyme, as I have told just now, it takes more than a year to get digested. So because of the enzymes only, the food is getting so faster and digestion process occurring. Okay. Now, there are certain group of enzymes, their function will be optimum. Optimum in the sense, it will be in a higher level at an optimum temperature. That means in a fixed temperature. Suppose if I just lower the body temperature, if it become 34 degree or 35 degree, the enzyme do not work properly. Okay. So like that, if my body temperature suddenly start getting increased, if I start getting a high fever, then automatically I started getting tired. I started feeling tired. Why? Because the proteins, enzymes are nothing but proteins only, they started getting denatured. Once it get denatured, you cannot regain it back. Once again, let me take the same example of an egg. So the egg yolk, everybody know, the yellow color part which is present inside, that is nothing but a protein only. So if you keep that egg in a refrigerator, it won't get spoiled. Why? Because in such a temperature, low temperature, in a refrigerator, temperature would be lower. So there, the enzyme activity become numb. Numb in the sense it will be ceases there only. So it will be keep waiting. Oh, when I am going to get a room temperature? Then they start acting properly. Okay, now you just take out the egg, you just keep it outside. Without keeping it in a refrigerator, of course it will get spoiled within 2 or 3 days. Why? Because the enzymes present in that, the proteins, they started showing their action, it will get spoiled obviously. Right? So like that, if I just take a egg yolk, that is the yellow color part, if I just put it on a tawa, then what happens? It will become an omelette. So now, if I want to recover the egg yolk back, can I get it back from the omelette? Of course not. Why? Because in such a high temperature, the protein part has got denatured. So once it got denatured, it is an irreversible process. That means temperature play a very, very important role in enzyme activity. So every enzyme do not work in the same temperature. For example, if I take an organism, imagine this organism, it's a bacteria. So this bacteria name is Thermus aquaticus. So this Thermus aquaticus says, my name itself is Thermus aquaticus. I am going to be present in high temperature area. So it usually survive in a very high temperature like 80 degree, 90 degree, so on. So here there is an enzyme present inside its body. This enzyme name is Taq polymerase. So why I am telling this? Particularly because we are going to study about this in class 12th. If you are a class 12 student, you know about this because we already learned in biotechnology, correct? So there is a process called PCR, polymerase chain reaction. In that, we are going to use this Taq polymerase enzyme. Okay, if I want to get a same DNA in multiple copies, just like a Xerox one, I need to use this enzyme Taq polymerase. So this Taq polymerase, for its work, we need to provide more temperature. For example, around 90 degree, 85 degree. So like such temperature I need to provide, then only it will work properly because they can survive in very high temperature. Now, if I take this enzyme and introduce into a human body, do you think it will work? No, because for such enzyme, the human body temperature will be very low. Where is 90 degree? Where is 37 degree? So obviously it is very, very low, no? So then its action becomes ceases. Just like how we are going to keep a egg in a refrigerator, in that it feels, oh, you have kept me in a human body that is a refrigerator, so I won't work. That means every enzyme is having its own temperature zone. So they are going to work properly. So if I want to generalize these things, then I can put a graph here. So let me write a temperature here. And here in a y-axis, this is x-axis. Here, let me write enzyme activity. Now, let's consider this would be the 0. This would be the 0 degree. So, like that, this would be very high temperature. If I take a human body temperature only, obviously it will be more. Let's assume like that. Now, what happened? If the temperature increases, suppose the enzyme is at 0 degree, now it is not doing any action. Now we have increased the temperature, the activity increases. Now further you have increased the temperature, the activity increases. Further you have increased the temperature, activity increases. So like that, at an optimum temperature, say for example around 37 degree, this is the 37 degree Celsius. I am talking about human body. Now the temperature, when it becomes 37 degree, the activity will be maximum activity. So this temperature would be referred as optimum temperature. So at optimum temperature, activity will be higher. Okay. Now, if I further increase the temperature, then what happens? Automatically activity comes down. Further if I increase, again it will come down. Further if I increase, it will get a denature. Okay. That means if I increase more temperature if increase, high temperature, then what happens? Here it become damage. Denatured at high temperature. So once it get denatured, we cannot get it back. That means it would be irreversible. Suppose even if you have a low temperature, somehow we can reverse it back by keeping it outside or by providing more temperature. But if the temperature exceeds, then it is a permanent damage. Okay. So that is what a line given in NCERT, that activity of the enzyme doubles at every 10 degrees centigrade increases in temperature. Okay, that means if I increase the temperature, the activity double. Further, if I increase, activity double. So like that, when it reaches a optimum temperature, the activity will be very, very high. So this is about the temperature action. Okay. Now another factor is pH. Hope you have understood about this pH, hydrogen ion concentration. So here also the same thing, just like temperature. Let me put a graph for this also. So let me consider this would be the x-axis, this would be the y-axis. So what I need to write here? This is the pH and this is enzyme activity. So here also same thing. Every enzyme will work properly at an optimum pH. There is a fixed pH for particular enzyme. So let me take a human body example only. Digestion chapter has got deleted, but hope you have some knowledge, little bit knowledge about the human digestive system. At least you might have learned in your lower classes. Okay. So now if you just recall the thing, in our saliva also enzymes will be there. Correct? Amylase. So many enzymes are there to break the carbohydrate. So for those enzymes to work within saliva, in that environment, their pH should be neutral. That is around 6.8. So enzyme says, imagine this is an enzyme. So this enzyme says, if I have to work properly, if I have to break the things, then I need this temperature. Then only I will do work. Suppose if you increase the temperature, pH, then obviously I can't work. Or even if you lower the pH, then also I won't work. So this is a fixed pH for these salivary enzymes. Okay. So like that, if I go down to the stomach. So in the stomach, again there are so many enzymes are there. For example, pepsin and all. To break the protein, we need pepsin. So pepsin says, if I bring the pepsin and if I put into the mouth, do you think it will work? No. Why? Because in the stomach, we are going to found acidic pH. Acidic pH means around 1.8 pH is very, very acidic. In such acidic pH only, these enzymes will work. Suppose if it's an enzyme, it says I will work in this pH here because I will active only at acidic pH. If you increase, I won't work. If you decrease, I won't work. So there is a particular pH for particular enzymes to do their function. Okay. So like that, if I further take one more example of a pancreatic enzymes or intestinal enzymes, which are present in the intestine, human intestine. So there the pH will be basic. So basic pH in the sense, it is around 7.8. So those group of enzymes, they will be active at this pH. So if you put those enzymes in this way, in the stomach, they won't work. If you once again keep it in the saliva, they won't work. So what did you understand? If you provide a low pH, then also it won't work. If you provide very high pH, then also it won't work. So you need to provide an optimum pH. If you provide an optimum pH, this is optimum pH. Optimum pH, enzyme activity will be higher. Optimum pH in the sense, depending on the type of enzyme we are talking about. To this pepsin, the optimum pH is 1.8. To this enzyme, the optimum pH is 6.8. To this enzyme, the optimum pH is 7.8. So it differs. Understood? So hope I am very clear about these two factors, temperature as well as the pH. Let's read a little bit about NCERT. Okay? Here, this paragraph together explaining the concept of temperature as well as the pH. And 2 or 3 lines which actually was there in the beginning. Few minutes back I said, no, I will explain it in factors affecting the enzyme. That paragraph also I have put. I will read that also. Okay. First, let's read this paragraph. Factors affecting enzyme activity. The activity of an enzyme can be affected by change in the conditions which can alter the tertiary structure. How they are going to show decreases or increase activity, that is by changing their tertiary structure. So if the tertiary structure itself got changed, if there is no active site, then how come the enzyme will work? So that is what the problem the enzymes are facing when you increase or decrease the temperature or the pH. Okay. Protein. These include the temperature, pH and change in the substrate concentration or binding specific chemicals that regulate the activity. Right. Okay. So first is temperature and pH. Enzymes generally function in a narrow range of temperature and pH. Narrow range means it has a fixed. It is not having a broad mindset. I can work in low temperature also. I can work in high temperature also. No, it won't work like that. It is narrow-minded. If I have to work in this temperature means I should be active in that only. If I have to work in this pH means I should be active in that pH only. That is what called narrow range temperature. So each enzyme shows its highest activity at a particular temperature and pH called optimum temperature and optimum pH. So hope you have understood this. Activity declines both below and above the optimum value. Low temperature preserves the enzyme in a temporarily inactive state. Did you understand? Temporarily inactive state means for time being they will be numb. So they have just tied their hand, their work, they have stopped for time being, and once they come out, then they start showing their action. So that we call it as a temporarily inactive. Just now I told, no, when you just keep the vegetables, fruits, or egg in a refrigerator, for time being they will be inactive. Once you keep it outside, of course, they are going to get spoiled after few days. Whereas high temperature destroys enzymatic activity because the proteins are denatured by heat. Important. Okay, so that's it. We have understood. Now in this paragraph, this is the first paragraph of enzyme part. Already we have understood up to here about the active site. And we need to talk about this line also we have read. Inorganic catalysts work efficiently at high temperature, high pressure, while enzymes get damaged at high temperature. So from here I need to read. While enzymes get damaged at high temperature, say about 40 degrees centigrade. So this we have understood. However, enzymes isolated from organisms who normally live under extremely high temperature. Okay, for example, in hot vents and sulfur springs are stable and retain their catalytic power even at high temperature up to 80 to 90 degrees centigrade. For example, Thermus aquaticus, which lives in hot spring, they can survive in such a temperature. Their enzyme also could able to survive. Thermal stability is thus an important quality of such enzymes isolated from thermophilic organism. Thermophilic means heat loving. Okay, heat lovers. Thermo means heat, philic means loving. Understood? See, this is why this chapter is difficult. This is about the first paragraph. But these points are going to be relevant to the factors paragraph. So how we need to connect the dot. Hope you are understanding, no? So that's why you need a lecturer help. So if you have a proper guidance, how to read, how to understand the concept, then biology become too easy for you. First of all, we should not spend more time on biology. Compared to biology, physics and chemistry are very difficult subjects. I know it. So if you finish this biology within 6 months, within 7 to 8 months, then entire biology will be in your hand. So that you will become a master. But only thing is you need a proper guidance. Okay. So if you follow either my videos or somebody else video, it's not a matter. But if you are following any one lecturer, please do follow them. Okay. So or else you will become in a puzzle to manage the time. Okay. So that's why it's my advice. Do follow any one channel or any one lecturer so that if some information given in one chapter, that will be interconnected to some other chapter, because entire biology textbook of both class 11th as well as the class 12th is just like one chapter itself. Okay. So hope all these concepts are getting clear for you. If you are really liking the video, then what you are waiting for? Just hit a like button and please do share it. Share this video to your friends also. Okay. Tell them to subscribe and tell them even to become a channel member. Okay. Now let's read one more topic about the concentration of substrate. So to understand this, let me explain the part. So this is the third factor we are reading, concentration of substrate or substrate concentration. So once again, bit confusing, but actually very easy, very, very easy. Yes. Let me make you understand one important task here. What is that? First of all, we should know about the rate of reaction. Either it is a physical reaction or a chemical reaction. First, we should know the meaning of rate of reaction. What is rate of reaction means? Everybody is keep telling that, oh, rate of reaction is like this. This reaction is showing this much rate. So what exactly rate of reaction means? Listen to me. Imagine I have given 1 second time. I have given 1 second time for you. For you in the sense for an enzyme. Now within 1 second, suppose from that enzyme, if I am getting 10 product, then I can consider this is the rate of reaction. This is the rate of chemical reaction of that enzyme. Or else, imagine, okay, here I have provided 37 degrees centigrade. Let's assume like this. Now, let me take one more example. Suppose if I have decreased the temperature, that is by 35 degrees centigrade. Then, I have given the same timings, 1 second. In that 1 second, I am going to get 5 product. 5 product. So, at 35 degrees centigrade, the rate of reaction would be 5 product per second. Understood? That means how I can summarize? Rate of reaction means product formation at a given time. The quantity or the amount of product formation at a given time is nothing but the rate of reaction. Okay, that means it will be varies when I decrease or increase the temperature. It would be varies if I increase or decrease the pH. That is what we have learned. Temperature matters, pH matters. So like that, even the rate of reaction will be get affected even if you increase or decrease the substrate concentration also. So how we can understand that? Once again, for that also I need to show up this concept through graph. Okay, so let me draw a graph here. So let me consider this as an y-axis. This would be x-axis. Here substrate concentration, just like pH and temperature. Here let me write enzyme activity. Now imagine we are in such a situation that I have provided optimum temperature here. Okay, temperature is optimum. So if I am carrying this reaction in a human body, the temperature is optimum, that is 37 degrees centigrade. So like that, I have provided even optimum pH also. Optimum pH. So everything is optimum. Now the thing is, how these enzymes are working, it is depending on the substrate concentration. So for that, we need to take number of enzymes also, right? Let's imagine in this reaction, I have around 10 enzymes. So for a rough sketch, I am writing here 10 enzymes. So this is 1 enzyme, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, 10 enzymes I have. Let me write it here, enzymes. Now, listen to me carefully. If you understand this graph, then the rest of the things will be very, very easy. Almost we are at the end of this chapter. Okay. Okay. Now, let's assume here is a substrate. Imagine this is a substrate. Now, let's consider if one substrate combines to one enzyme, then it will convert it into product. Okay. So, this thing is happening in 1 second. So, what I said, where shall I write? I will write here only. So, what is the rate I am getting? In 1 second, I am getting one product. One product I am getting. This is what the rate of reaction. In 1 second, I am getting one product because this one substrate has combined to this enzyme. Okay. Now, I have provided more substrate. Let me make it in red color. Imagine I have 3 substrate molecules. Then what happened? 3 substrate combines to 3 enzymes. Correct? Then how many product I got? 3 product I got in 1 second. Correct? Did you understand? 3 product in 1 second I got. Now I have increased the substrate concentration further. I provided 5 substrate. Now all these 5 substrate combined with these enzymes, 5 enzymes, because I have more enzymes here. Then how many product? Think and tell the answer. I know you can't tell the answer, but at least you are understanding, no? 5 substrate I have given, and these 5 enzymes combined to those 5 substrate. Then automatically how many product I am getting? 5 product I am getting in 1 second only. I am not increasing the time. The time is fixed, but enzymes are more. So rest of the enzymes are free. Now all these enzymes are getting the substrate, immediately they are converting into product. If I am a enzyme, I got a substrate, I converted into product. Again, one more enzyme is there, just like me. She also got a substrate. She also make it converted into product. So like the 10 members we are. So 5 members, still they are waiting for a substrate. Okay. Now I can increase the substrate concentration. So let me show graph here. So I am increasing. The product is also increasing. Again, I have increased the substrate. The product is also increasing. Again, I am increasing. Product is also increasing. Now, what I'll do? I have provided 10 substrate. 1, 2, 3, 4, 5. So these 5 again combine to these enzymes. So how many I got? 10 product I got. So my graph has reached to the peak now. It has reached to the peak. Let me draw it clearly. It has reaches to the peak. Okay. So that means the maximum, the enzyme is working in a maximum velocity. Maximum velocity. Now, the funny thing is, if I just doing the same thing, so 10 substrate I provided, so finally I got 10 product. 10 product I got per second in 1 second. Now, if I do the same activity, if I have provided 100 substrate, imagine I have provided 100 substrate. Do you think the enzyme will work it in the same speed? Obviously not, because enzyme says we got saturated, man. We got saturated. So even if you provide more and more substrate, where is the time and where we are? Only we are 10 members. So only 10 members, how much we could able to accept the substrate, that much we have accepted and we are converting into product. But you are still simply providing more and more. This is enough. We can't do this anymore. That means it has reached to the saturation. Even if you provide more and more substrate also, if you increase the concentration of the substrate also, then the enzyme activity become saturated. Okay. It means what? They have reached the maximum velocity. The enzyme activity has reached to the maximum velocity. Shall I mention the same thing as Vmax? Shall I consider this as a Vmax? Vmax means maximum velocity of an enzyme. Okay. Now, suppose, listen to me carefully. Now, if I just want to get 5 product, where would be the graph? Obviously, it is somewhat here, no? In this area, I have obtained the half the velocity ratio. It means when I come to here, when I come in this area, velocity maximum was there. Maximum velocity is here. Now in this region, I got half the maximum velocity. That means here I got able to get only 5 product per second. But here I got 10 product per second. Here I got 5 product per second. It means this is the maximum velocity. If I come half of the way, then I could able to get only how much? The half of the enzyme activity. Okay. So that means here somewhere else this graph has reached. Here somewhere else the graph has reached. Okay. Now this point, what you are saying? So this point, what you are saying, no? Here this is actually the amount of substrate that you have provided. This will be considered as Km. This will be considered as Km. So Km means what? The Km is equal to, Km means the amount of substrate that you have provided here. When you provided this much substrate, that is 5 product, then the enzyme activity has reached to its halfway. Okay, when you provide more substrate, then it has reached to the maximum velocity. But here the Km value means the half of the velocity that enzyme has reached when you have provided the substrate. Shall I write the same thing here? Km means, okay, Km value means, or Km means the substrate concentration at which the reaction reaches. Suppose, imagine if I have, now you tell me, if I have 100 enzymes, then the Vmax would be reaches only when I have provided 100 substrate, correct? And Vmax by 2, will be obtained only when I have provided how much? 50 substrate. That means here somewhere else I may get Km value. Imagine the graph would be more. Let me show somewhere else. Hope this is very clear. This is very clear. Please write down in your copy. All these things are very important. Very important. Okay. It's given in a graph format in your NCERT, but the question will come from this part. So let me show here one more. So imagine I have 100 enzymes. So I'm writing like this. So let's assume here 100 enzymes are there. Now I have provided first initially 50 substrate. So all 50 substrate had combined here to these enzymes. Now further I had 50 substrate. It become 100. It reaches to the saturation. That means the graph has been obtained somewhere else here. It has reached to this peak. Now this is nothing but Vmax. Vmax. Here 100 product I get per second. Now this would be the half the value. Okay, so if the graph is reaching up to here, then this would be the Vmax by 2. So if I draw somewhere else, I may get Km value. Okay, that's not good. 1 second. So this would be the Km. So here the Km value is 50. Here the Vmax is 100 per product. Here 50 per product. 50 product per second. So this is the Km. Substrate concentration which has reached half of the velocity. Understood? So like that it will be varies depending upon the enzyme activity. At which ratio that you got a maximum velocity, the half of will be the Km value. That is Vmax by 2. Understood? So this much is enough. Don't just get confused by reading something else. Even if you read other publications, other book, the questions won't come from those parts. Okay, trust me, all the questions, everything for your NEET exam will becomes only from your NCERT. So if you understand each and every word of NCERT, trust me, you can't make mistake in your exam. Okay, now let's read the same part what we have done in NCERT part, NCERT textbook. Okay, this is the graph. So hope you have understood this graph. Here they are talking about the pH. So we got an optimum pH. This is the optimum pH. And this is representing the temperature, and this would be the optimum temperature. And look at here, this is the concentration of substrate, which is showing the enzyme activity. So here they are showing the velocity has reaches to the maximum. We got the graph something like this. And this is the Vmax by 2. Vmax by 2. So it has N to the Km. Okay, so this is the Km value. That's it. Very simple. Now let's read this part. Concentration of substrate. With the increase in substrate concentration, that means when you keep increase the substrate concentration, velocity of enzymatic reaction rises at first. First it will rise. The reaction ultimately reaches to the maximum velocity. So finally it has reaches the maximum velocity. We consider it as a Vmax, which is not exceeded by any further rise in the concentration of substrate. That means if you just keep expecting the velocity of enzyme will keep increasing, even if I increase the substrate concentration, that is a nothing but it is a false, it is a myth. It is not true, because once the enzyme has reaches the saturation level, it cannot further increases its activity. This is because the enzyme molecules are fewer than the substrate molecule. Okay, and after saturation, these molecules, there are no free enzyme molecules to bind with the additional substrate molecule. If there is no enzyme, then how can you expect the velocity, increase the enzymatic velocity? So it will be dropped there only. That's enough about the concentration of substrate. Over. Okay, so factors affecting the concentration of substrate is over. Now let's go to another important topic, that is enzyme inhibitors. What are enzyme inhibitors? Imagine I'm an enzyme. Okay, I was doing my work so efficiently, so properly. So I was binding to the substrate, I was just packing it, I was sending into a product, converting into product. Okay, I was receiving substrate, converting into product, substrate, product, substrate, product. Why I am doing this? Because I am so efficient. Now, some guy came. Somebody came and he had tied my hands. Do I do this work properly in the same way how I was doing before? Obviously not. Why? Because my hands have been tied. Okay. So, till he has to release my hands, I need to shut my mouth, I need to shut my activities. Okay. I don't have any other option. It means what? My efficiency has got decreased. I was doing this work within 1 second, I was preparing one product. Now, because of this obstacle, I was just waiting till he has to release me. That means if he has released after 5 seconds, then after 5 seconds only I could able to convert it into product. So, till 5 seconds, the enzyme activity was inhibited. That means somebody came to inhibit this activity. So, that somebody is nothing but inhibitor. Understood? How this action of inhibition occur in enzyme? Who is working as the inhibitor? Let's see this. Okay. So, here in enzyme, our enzyme inhibitor are of two types. Listen to me, very easy topic. There are two types of enzyme inhibitors. One are called competitive inhibitor. That action is called competitive inhibition. This process is competitive inhibition. The chemical is competitive inhibitor. Chemical is an inhibitor, that process is inhibition. So like that, there is one more chemical, it is non-competitive inhibitor. That action or a process is non-competitive inhibition. Example to this is not given in NCERT, but for this one, NCERT has provided given example also, but it has not giving much information. First of all, not given any information about non-competitive, but you need to learn. Suppose if the question comes in an option way also, you should not get panic. Oh, what is this? We have not heard this non-competitive only. So I will teach you. Within 2-3 minutes, we will understand this. Okay. Let's understand first what is competitive inhibition. Imagine this is an enzyme. So this enzyme is very happy. Very, very happy. Why it is very, very happy? Because it can do its function properly with the help of substrate. With the help of substrate means to convert the substrate into product. Imagine this is the substrate. So here is the substrate. Substrate is also very happy because it has a proper site to bind with this active site of an enzyme. So this process is nothing but enzyme substrate complex only. No, this area is called active site. This area of an enzyme is called active site. To this active site, the substrate come and binds. Okay. Now, imagine there is one more guy. This fellow, he is not actually a substrate only. He is somebody else. But he resembles like substrate. Okay. If you just look at the shape of these two, he is also having the same shape. He is also having the same shape. So, if he comes before the substrate, if he comes and bind this active site, then what happens? The enzyme will consider this as a substrate only and it will just keep because it cannot convert it into product. Because it is not its cup of tea, no. So that to convert this substrate into product, so it will be just shut its activity, it will be keep numb. Why? Because this fellow has combined the active site. He has not having the intention of converting into product, but still he is coming and binding to this active site just to avoid the substrate molecule from being bind here. Okay, so this fellow is nothing but competitive inhibitor. Having the shape exactly like substrate. Okay. So obviously what happened? The activity will be slowed down. So if he just come and bind here for 10 seconds, then the enzyme cannot do any function for 10 seconds. So after 10 seconds, if the substrate comes and bind, then it will start showing the work after 10 seconds. That means efficiency of an enzyme got decreased because of this inhibitor. So this process is nothing but inhibition. As it is done by this same kind of substrate, it is called competitive inhibition because having the same shape. Okay. So one such example given in NCERT is This enzyme name is succinate dehydrogenase. Succinate dehydrogenase. It says, I am an enzyme. I am here. I took birth here only for this substrate. So this fellow name is succinate. Succinate. Hence my name is also succinate dehydrogenase. But here he is just resembling like that of succinate, but he is not a succinate. Who is he? He is malonate. Why did you come, man? You malonate. Why did you come? Because of you, my activity has got decreased. Okay. So this is called competitive inhibition. Okay. Now another one is, hope you are writing all those things in your copy, or else you will get confused. Write down everything in your copy. At the time of exam, if you just glance that book, it should be easy for you to understand each and everything. Even if you are not writing anything, just write down in the space which have been provided in your NCRT. That is in the left side and right side of the corner. Some space has given no in your NCRT textbook itself. Write down there only. At least even if you can't read entire paragraph at the time of examination, even if you just glance those things, you should be able to recall everything what you have learned. Okay. Next one is non-competitive inhibition. Non-competitive inhibition, it is quite funny. What is that? Imagine again, this is an enzyme. So this is an enzyme. So this would be the active site. Active site. So here what happened? Look at here. This is the substrate. So this substrate came. He is very happy because he just look around. He won't find any competitive inhibitor. So he very happy and he just bound here to this active site. Okay, enzyme is also very happy. But now the enzyme in a tertiary structure we have learned only active site is not a pocket. Apart from this, there are other pocket, other crevices also there, but we won't consider it as an active site. Correct? Now imagine this would be the pocket, this would be the crevices. Now to this crevices, somebody, he is a villain now. He is a villain. Now he is coming and binding here. He is coming and binding. That means what is the use for him and what is the harmful effect for him also. That means this is the substrate. This is the substrate. Now even if you come and bind to some other area, what is the problem for me? I am okay with this. So like that it is very happy, very cool and very relaxed and binding here. But this fellow, he is a non-competitor. Non-competitive inhibitor. He says, are you happy? Just now, what I will do? I will just do a magic. If I bind here, even though I am not coming here to your way, but I will change this active site shape itself. That means, instead of like this, if I have changed this active site, something like this, how we are going to bind? Because I have changed the active site only. Now, even if you just want to come and sit also, there is no use. Because there is no binding site at all. I have changed this active site. So, like that, he is simply feeling happy. Okay. So, this is called non-competitive. Even though he is not having the same structure like substrate, but it is hindering or it is slow down the enzymatic action. Understood? So here the example for this non-competitive inhibition is not given in NCRT. Let us not worry about this. But you should know the meaning of non-competitive inhibitor. Now the question is, ma'am, suppose if such things happen in our body, what we need to do? Okay, so there is only one way we can escape from this competitive inhibitor. How? If you increase the substrate concentration. Suppose, imagine, I am just writing the substrate something like this. Imagine, if I have increased the substrate concentration, let's assume I have 100 substrate here, only one malonate here. Then who will come and bind first? Obviously, this succinate only, because 100 succinate are there. If not one, somebody else will come. So like that, everybody is standing in a queue so that they're actually avoiding him. So there is only one way we could have, so that if you increase the substrate concentration, automatically we can decrease this competitive inhibitor also. That's it. So this paragraph also over. Let's read this part from NCRT. Actually, I have joined this line because this line is actually indicating the graph presence. If you open your textbook, you can see above this line, we have a paragraph of substrate concentration as well as the temperature and pH. That paragraph is about the factors affecting the enzyme activity. At the last line of that paragraph, we have this information regarding this competitive inhibitor. Okay, and in between, graph is there. Already we have learned that graph, and after that again they have given about the inhibition. So let's take from this line only so that we can understand it better. Okay, the activity of an enzyme is also sensitive to the presence of specific chemical in the sense they resemble like that of the substrate. Okay, that bind the enzyme. That means they are talking about the competitive inhibitors or non-total inhibitors. When the binding of chemical shuts off the enzyme activity, the process is called inhibition. So this statement can be directly asked in your exam. Okay, so when it gets shuts off, when it has the chemical bind to the enzyme activity, shuts off the enzymatic activity by binding to that active site or somewhere else. And the chemical called inhibitor. When the inhibitor closely resembles to the substrate in its molecular structure and inhibits the activity of the enzyme, it is called competitive inhibitor. Just now I told, due to its close structural similarity. Exactly like that only. With the substrate, the inhibitor competes with the substrate for substrate binding site of the enzyme. So substrate binding site means they are talking about the active site. So when it comes and combined to the active site of an enzyme by something else, then obviously substrate will be wait. It has to wait till it move. Okay. Consequently, the substrate cannot bind, and as a result, the enzyme action declines. Example, see, inhibition of succinic dehydrogenase by malonate. Malonate is an inhibitor which closely resembles to the substrate succinate. Succinate is a substrate in the structure. Such competitive inhibitors are often used in the control of bacterial pathogens. Okay, in bacterial pathogens, usually these things will happen. So, hope you have understood this paragraph. Yes, next thing is classification and nomenclature of enzyme. So, this is the last part. Actually, it's not a last part. After this, one more part is there. Hope you are not getting bored, no? Because the chapter is quite interesting. Only the exception is the structural part. So, apart from learning the structure, everything is quite interesting only. So, it's not mandatory that you have to watch the video till the end within one day. So, you can just divide the video for a week, as I already told in the beginning of the class. Okay. Now, classification and nomenclature of enzyme. See here what they are telling. I will explain here only everything. It is easy, but you need to concentrate. Because the question will come. Don't worry, how much I am telling, that much if you learn, that is more than enough. Classification and nomenclature of enzyme. How we are going to classify? There are plenty of enzymes, but based on their function, we are going to group them into 6 classes. Okay. Thousands of enzymes have been discovered, isolated and studied. Most of these enzymes have been classified into different groups based on the type of reaction they catalyze. Okay. Enzymes are divided into 6 classes. Important. Each with 4 to 13 subclasses. Important. So, within one class, again 4 to 13 subclasses are there. But NCERT is not giving information about the subclasses. So, let us learn only these classes, 6 classes. And named accordingly by 4-digit number. Okay. Yes. The first one is oxidoreductase or dehydrogenase. First, I will give example. What is the meaning of oxidoreductase? Imagine, let me write here. Oxidoreductase. What it will do? Reductase. Spelling wrong. Look at my spelling. So, this is an enzyme. Okay, is a very cool and happy guy. So, he is giving instruction to the substrate. Imagine there is a substrate called A and one more substrate called B. So, both are undergoing the reaction. But here, this A is having hydrogen with it. B is not having hydrogen. When it have hydrogen, we consider it as reduced substrate. So, you are reduced. Now, this fellow is giving instruction to this A. You have hydrogen. So, that's why you have reduced. Now, you give it to B. You just give it to B. Let him get reduced. Okay. So, because of that, now A loses hydrogen and B got hydrogen here. Got it? Who is instructing that? This enzyme, his name is oxidoreductase. Because of this, earlier A was reduced, now B become reduced. One who have hydrogen, he is called reduced. One who loses it, one who do not have this, he is called oxidized. Now by giving hydrogen, he is considered as oxidized. He is oxidized. But earlier, without having, he was in an oxidized state. He was in an oxidized state. Now by accepting, he become reduced. Somehow who accept, he become reduced. Now who give, he become oxidized. So that is what the meaning here they are telling. Enzymes which catalyze oxidoreduction between two substrates. Here I took the example S and A and B, but here they are telling S and S dash. That's it. Okay. See, S is reduced. That means nothing but A only in a reduced state. And S dash is oxidized. B is nothing but S dash. They are telling they are in an oxidized state without having hydrogen. Now, the scenario become reversed. Now, S become oxidized and S dash become reduced. So, if you just consider it as A and B, now A become oxidized, B become reduced. It is also called dehydrogenase. Oxidoreductase and dehydrogenase both are one and the same. Now, the second one is transferases. What they will transfer? Here also very simple. Look at here. Let me take the same example. Here A is there and B is there. Okay. Now what happened? Here enzyme come. So this is an enzyme. His name is transferase. His name is transferase. So he is telling, you have one group. Here he is having one group. I am a substrate. I have this pen. This is not hydrogen. This is not hydrogen. Now I am giving this pen. I am transferring this pen to my friend. Are you getting? I am a substrate. My name is A. I have this pen. This is one molecule that I had, but it is not hydrogen, man. This is not hydrogen. But some group I have. Let me consider this pen is some group. Now I am transferring it. Simply I am not transferring. I am not that much generous. Somebody is giving instruction. Who is giving instruction? Transferase. Earlier my friend was simply like this. Now I am giving this pen. I am transferring this pen to my friend. Now this pen is with my friend now. Understood? So, the B is my friend. She was not having this pen. Now, because of the transferase instruction, now A is not having anything. Now, this pen is with the B. This pen is with B. So, I have transferred it. That function, which is so done by the enzyme, we call them as transferase. Okay? Look at here. The same example given. Enzyme catalyzing the transfer of a group. Group means anything. It could be sulfate group. It could be phosphate group. It could be any group. Simply, I took an example of a pen. Okay. But remember, here they have mentioned the group as G. But the G should be other than hydrogen. If it is hydrogen, then we will consider it as a oxidized state or a reduced state. But here it is not hydrogen, something else. Between a pair of substrate, S and S dash. So here I am just taking A and B. They are taking S and S dash. See, S is having this group, but S dash was not having anything. Now, S has lost it, but it came to S dash now because of the transferase. Then the third one is hydrolases. Hydrolase means what? See, in our body there are so many digestive enzymes are there. So all these digestive enzymes, they are breaking the food. If I have to digest the food what I ate, for example, the starch I had, glucose, it has to convert it into glucose. It has to break. The lipid I had, it has to break. The protein I had, it has to break. So all these molecules will be get breaking down with the help of water. With the help of water. When water is added to break down this reaction, with the help of enzyme, such enzymes are called what? Hydrolases. Name itself is telling hydrolases. So, best example we can take here, all digestive enzymes. Digestive enzymes are nothing but hydrolases. See, enzymes catalyzing hydrolysis of ester, ether, peptide, glycosidic, that means bond is breaking. You know how the bond formation occurs. When you just break it by adding water, then all these molecules become released out. Okay, so such enzymes are called hydrolases. Between C and C, C halide, P and N bonds, everything will be get broken down. Now the fourth one is lyases. So what is the meaning of lyases? Let me tell you here. Let me show you here. Imagine there is a Okay. There is a substrate called A and one more substrate is there, B. Let's consider like this. Now this A is having a molecule called X and B is also having a molecule called Y. Okay. Now an enzyme will come. So this is the enzyme. This enzyme come. His enzyme name is lyase. So lyase says, I will break you guys, but not by adding water like hydrolysis, but I will break you without water. Remember, this is important word, without water. Without water, I will break you so that I will release this group. I will make X and Y to get combined in this way. And these two substrate molecule which were there, no, I will connect you guys with a double bond. I will connect you guys with a double bond. I will release this the group that you have attached here. I will leave double bond here, but I will do this action without water. So that's why my name is lyase. Okay. So look at here. These three lines are important. It has been asked in previous years in NEET exam. Lyases means what? Enzymes that catalyzes removal of group. See, group has been removed from the substrate. So these are the substrate by mechanism other than hydrolysis. Other than hydrolysis means without water. Shall I write? Without water. And leaving double bond, important. So three lines you have to remember here. Without a double bond, without water, leaving double bond, removal of group. See, C is given. XY are the group attached here and XY has been removed. That means they have become separated. And plus C and C got a double bond in between them. Just like here. Just like here. So fourth one over. Then the fifth one is isomerase. Isomerase means include all enzymes catalyzing interconversion of optical, geometric, positional isomer. So you have learnt about this in chemistry. Okay. So in chemistry, usually we consider geometrical isomer, right hand, left hand, something like that. I am poor in chemistry, so I won't give much example about this. But even if I try to give example, I can take this one. You will study in plant physiology, but just have some idea. Glucose 6-phosphate is a molecule that is a intermediate compound that will be generated in our body at the time of pyruvic acid formation. Okay, if you are class 12, you already understood about this. So this glucose 6-phosphate now will convert it into fructose 6-phosphate. Fructose 6-phosphate. So everything is same to same. This one also having 6-carbon compound. This glucose is also 6-carbon compound. Here also one phosphate group is there. Here also one phosphate group is there. Only their molecular structure has been changed. Okay. So how it happened? This is by enzyme called isomerase. Isomerase. Okay. So that is what they are telling. Just interconversion. And the last one is ligases. Ligases is for joining. Okay. So everybody is breaking, now he is joining. Ligase means it acts like a glue. You are going to study about this in molecular basis of inheritance. So this is one nucleotide. Okay, let me show like this. This is one nucleotide. This is one nucleotide. These are the nucleotides. These are the nucleotides. Now, every nucleotide had made a bond, phosphodiester bond. Imagine, here there is no phosphodiester bond. Now, at that time, this ligase will come. He says, oh, you don't have phosphodiester bond between nucleotide. I will join it by phosphodiester bond. So, like that, it will join the nucleotides or join the molecules, acting like a glue. Okay, that we call it as a ligases. Shall we read here? Enzymes catalyzing the linking together of two compounds, linking nothing but joining. Example, enzyme which catalyzes joining carbon and oxygen, carbon and sulfur, carbon and nitrogen, phosphate and oxygen, etc. That means by making bond. Just you learn this much, that is enough. Only from this part itself, if any question comes from the classification, it will be asked only from this paragraph. That's it. Okay? Not from anything else. Now we are in the last part. That is cofactors. What are cofactors? Let's learn about this. Okay, what are cofactors? So, if I ask you a simple question once again, what is the nature of enzyme? What is your answer? Is it a carbohydrate? No. Is it a nucleic acid? Except few things, but rest of all are not. Is it a lipid? No. Then, what is its nature? Of course, it is a protein. Correct. So, let me write a protein here. So the protein is nothing but here the enzyme. Suppose if I say you are an enzyme, you are a protein part, but still this protein is upside. Still this enzyme is upside. Why? Because it is telling, of course I am a protein, but I can't do work. But not all enzymes, few enzymes. If there are 100 enzymes, around 50 to 60 enzymes are working properly, but there are few exceptions. They are telling we are unable to work properly. Why? What is your problem? Then it says, in order to work properly, I need one more factor. But it is not a protein factor, but I needed help. Okay. So that is nothing but a cofactor. Okay. So cofactor is a non-protein part. Then let's imagine if it is a cofactor. If it is a cofactor, then this enzyme, there is a protein part of the enzyme, it will call a cofactor to combine to its active site. You just come and sit on my active site so that I will get energy, so that I can do work efficiently like a complete enzyme. Okay. Then this combination, now it is happy. Okay. Then this enzyme combination along with the protein and non-protein part, what you are saying, this we call it as what? Holoenzyme. Holoenzyme. Okay. So holoenzyme is both this protein part plus non-protein part. So we should give a name to this protein part of the enzyme also. So what is the name we have given? Apoenzyme. Don't get confused. Apoenzyme. So this enzyme is nothing but apoenzyme. Non-protein part is cofactor. Then it is a complete structure. We call it as a holoenzyme. This was a question in previous NEET exams. Okay. So that's why it's a very important thing. You have to make a note. So then what are cofactors? So we have understood it is a non-protein part. Is there any bifurcation within that? Of course it is there. So the cofactor are of three types. Shall I move on to the next slide? If you are writing in your copy, please make a note of this. I am moving to the next slide. Okay. About the cofactor particularly. So this cofactor are of three types here. Listen to me carefully. It is very important. I am telling. We are in the last part of this chapter. After this, we'll discuss few questions and we'll end up the chapter. But is it okay? But still I have left one paragraph regarding primary and secondary metabolite. That is too easy, but that also I will do after the completion of this part. Okay. Yes. Cofactors are of three types. One is prosthetic group. Another one is co-enzyme. And the third category is metal ion. Metal ion. Very simple. Very simple. Let's imagine. Prosthetic group is like a best friend or it's like a wife. Let's assume this is the prosthetic group. So she is very obedient. She won't leave her husband in any moment. She will be there with her husband in all the time, in all the situation, whether it is a happy situation or a sad situation. In everywhere, she is with her husband. Okay. She is tightly bound to the husband, permanently bound to the husband. Okay. So let's imagine that husband is nothing but a enzyme. So let's consider he is an enzyme. Okay. So this husband was unhappy because he is unable to do work properly without her. So she came, don't worry, I am with you. I will bound to your active site so that you can work properly. Now he is happy. Now this combination, what you are saying, it is not just 1 or 2 days. It is a permanently fixed. So therefore we can say the prosthetic group, it is an organic group. Of course, it is a part of cofactor only, but it is a organic group. It is permanently and tightly bound to the apoenzyme. Now, when it bound together like this, do you call it as a apoenzyme now? No. Now this combination is a whole enzyme. Sorry, holoenzyme. Okay. Whole means complete. You can remember in this way. That's why I'm pronouncing whole enzyme. But actually, the right way of pronouncing is holoenzyme. It's not a hollow. It's a holoenzyme. Okay. So this is one category. Example part we'll see shortly, but you just know what are these things. Then the second one is coenzyme. So let me consider this as a coenzyme. So this coenzyme is like a hanky-panky girlfriend. Okay. So now she says, okay dear, you are upset without me. I will come and bound to you. I will come and bound to you, but not permanently. If I get one more boyfriend, I will go to him. If he spend money on me, if he give dresses and he'll take me to the shopping, then I will go to him, but not with you permanently. Okay. So she's temporarily bound and she's not permanently bound. Okay. It is just transiently attached to this enzyme part. So such a combination we call it as a coenzyme. Usually whenever we talk about a coenzyme, within this coenzyme part, our vitamins will be there. Okay, we'll talk about that little bit later. But let me write here, it is temporarily and loosely attached to a coenzyme. Okay, got the difference? And this is also organic group. Whenever we talk about any coenzyme, of course, they are also organic group, but it is a part of cofactor. We call it as a coenzyme. The best example we can take here is NAD. Okay, so NAD as well as NADP. Okay, both these coenzymes contain vitamin. So niacin is a kind of vitamin which is present inside this NAD and NADP. Okay, and the last one is metal ion. So metal ion is somehow balancing both enzyme as well as the substrate. For example, imagine this is an enzyme. This is an enzyme. Let's assume here is a substrate. Here is a substrate which is present here. Now, let's assume here it is the metal ion. So this metal ion, from his one hand it is holding the enzyme and from his other hand it is holding a substrate also, so that it is managing both. But of course, for a proper functioning of an enzyme, even we require this cofactor like metal ion also. Understood? So this is the main difference about between the prosthetic, coenzyme as well as the metal ion. Now let's read NCERT, and along with the concept, let us learn even the example also. Okay? Yes. Prosthetic group, example heme group, the organic group tightly and permanently attached to the apoenzyme. See, which one? Here the example given NCERT, heme group. Okay. So here this enzyme called catalase as well as the peroxidase. Here what the example I told, no, here this is an enzyme. So this enzyme is nothing but a catalase. It could be a catalase or it could be a peroxidase. Why this enzyme required? Because in order to convert this H2O2 into water and oxygen, in order to carry this reaction, of course we require this enzyme. But this enzyme says, I can't do this alone. So who is required? I need heme group. So then the heme comes and bound here to the active site tightly. Then it can become happy, it can do this work properly. Okay, so this is what the example about the prosthetic group. So like that, when we come to the coenzyme part, it is also organic group. It is temporarily and loosely attached to apoenzyme. As I said just now, NAD and NADP are the examples. Here it contains what? Vitamin. Let's read here. Cofactors. Enzymes are composed of one or several polypeptide chains. Correct? However, there are number of cases in which non-protein constituent, also called cofactors, bound to the enzyme to make the enzyme catalytically active. Okay, in these instances, means in these cases, the protein portion of the enzyme is called a coenzyme. Three kinds of cofactor. What are those? Prosthetic group, coenzymes and metal ions will be there. Prosthetic groups are, first they are talking about the prosthetic group. They are the organic compounds, are distinguished from the other cofactors in that they are tightly bound to the apoenzyme. See, the words what I am marking here, they are very important. For example, the peroxidase, catalase, which catalyze the breakdown of hydrogen peroxide to water and oxygen. Heme is the prosthetic group. Important. Heme is the prosthetic group and it is a part of active site of the enzyme. Okay, it is a permanently bound. So the one part is over. The second, coenzyme. Okay, so coenzymes are also organic compound, but their association with apoenzyme is a transient in the sense loosely bound. Just now I told, no, I gave the example of hanky-panky girlfriends like that. So usually occurring during the course of catalysis. Whenever the enzyme requires the help of this, then it will come and bind. Whenever the enzyme function over, it will just move to the other enzyme. Okay. So furthermore, coenzymes serve as a cofactor in number of different enzyme catalyzed reaction. Okay. The essential chemical components of many coenzymes are vitamins. So coenzymes can be of different types. Here two examples are taken. One is NAD, another one is NADP. NAD full form is nicotinamide adenine dinucleotide. Write down in your copy, and NADP. Okay, phosphate group has been attached to this, therefore it is called NADP. They contain which vitamin? Niacin, important. Okay, now the third category is the number of enzymes require metal ions for their activity, which form coordination bonds with, see, look at here, with the side chains at active site, and the same time form a one or more coordination bond with the substrate. That means they are going to form a side chain. If I am a metal ion, I am going to form a kind of bond with the substrate as well as even to the active site. Active site, of course, it is a part of what? Enzyme only. So I am holding both the things through my, with the help of a bond. Okay. Example, see, example important, zinc. Zinc is a cofactor for proteolytic enzyme of carboxypeptidase. Write down in your copy, carboxypeptidase. Zinc, it's a metal ion. Metal ion. So this is an important component for carboxypeptidase. So carboxypeptidase is an enzyme name. Zinc is the metal ion. So this example you need to remember. Catalytic activity is lost when the cofactor is removed from the enzyme. So that's why we said if the cofactor is not there in such groups, then automatically the enzyme activity will be stops. Okay, which testifies that they play a crucial role in the catalytic activity of the enzyme. Over, chapter over. Okay, happy? But still, we need to solve MCQ, and after that, one small concept about secondary and primary metabolites. Very simple. Let's solve your MCQ. Come. Okay. Choose the correct regarding active site. Substrate fits X. Active site fit properly to the substrate. Enzyme catalyst through active site show low rate. How come it is low rate? It is wrong. It should show faster rate. Therefore, this is wrong statement. It forms by crevices or pocket made by primary protein only. So, this is wrong. It is actually by tertiary protein. This statement is also wrong. It forms by crevices or pocket by tertiary protein structure. Correct. That means statement 1 and 4 are correct. Here, statement 1 and 4 in option D. So, this is the right answer. Next question. Hydrolysis of starch into glucose. So, starch is a polymer. If it has to break and it become a glucose, then what? Of course, here the enzyme must be involved in that. What type of reaction it is? Is it an inorganic? Inorganic chemical reaction is one where two inorganic molecules become joined. For example, if I want to create a glucose, then two inorganic molecule has to join together, water as well as the carbon dioxide. But here already glucose, already starch we have. It's a polymer of glucose. We are just breaking that to convert it into glucose. Of course, it is a chemical reaction. It is an organic chemical reaction. So this is direct NCERT line. It's not a physical changes. Wherever you see a change where the bond breaking taking place, it is not a physical. It is always chemical. Remember. So of course, not A and B, not even A. Next question. Choose the correct response. For every increase by 10 degrees centigrade, rate is double, correct? But still, let us see the rest thing. Rate decrease by 1/4 by decrease in temperature by 10 degree. No, this statement is not given in NCERT. Let us not bother about this. When enzyme catalyzed reaction are observed, the rate would be vastly lower. The rate would be vastly faster. Therefore, this is wrong. None of these wrong. Therefore, the right one is option A. Next question. Choose the correct response with respect to given equation. What they are telling? Carbon dioxide plus water gives rise to carbonic acid. Correct. Carbonic anhydrase. Correct. Here the enzyme carbonic anhydrase is required for accelerated reaction. Accelerated in the sense to speed up the reaction. Because of this carbonic anhydrous only, we would have able to get 600,000 molecule of carbonic acid just within a second. So this is right. In absence of enzyme, still this reaction is fast enough. No, it is slow enough in fact. 200 molecule of H2CO3 being per hour formed by enzyme accelerated reaction is given. Up to here correct. 200 per hour, it's okay, but not in the presence of enzyme. 600,000 of molecule of H2CO3 is being formed every second in absence given. Therefore, this is also wrong. So, hope you are understanding how the questions will be framed just from the line of NCERT. Now, you are aware about NCERT textbook also of this chapter because each and every line is perfect. I know you are very perfect in biomolecules, but the questions what I am solving here, this is not enough. Try to solve as many questions as possible. Be get perfect in every concept as well as every topic of chapter. Then only you could able to solve any sort of question when it comes in your NEET exam. Okay? Yes, transition state structure is formed when? Transition means what? When it enters into a activation, to gain activation energy. By that time, so many different transition state has been formed. No. Before that, it was a substrate. When it combined with product, then we could able to see many things. I have shown this, no? It may bind like this, it may bound like this, it may bind like this. So these are enzyme product only. I mean, yes, enzyme and substrate only, not enzyme and product. Enzyme is free? No. Enzyme bound with the product? No. It is enzyme substrate complex. Here, substrate structure do not change until the product form. This is also wrong. When enzyme substrate complex has formed, then only they started showing the different changes. Yes, next question. See. Yes, can you see here? Here is a substrate. This is activation energy without enzyme. So 2, this is actually 2 activation energy without enzyme. Here transition state given wrong. Activation energy without enzyme, this is right. Here also activation energy without enzyme, right. One is activation energy with enzyme. Here activation energy with enzyme, this is right. Here again transition state given. And the third one is actually different states which are so formed. It is a transition state. So option C is the right answer. Next one. Which of the following can change enzyme activity? All such activities that can alter the tertiary structure of the protein, correct? Temperature and pH, correct? Substrate conditions, correct? Everything will affect the enzyme activity, all of this. Optimum pH refers to pH at which enzyme activity is lowest, wrong. Enzyme activity should be highest. pH at which enzyme activity is highest, this is the right answer. pH at which enzyme activity started immediately, no. Enzyme activity slowly start gradually down when you exceed the optimum pH. pH at which enzyme activity ended completely wrong. So now all these things are in your fingertip. After reaching Vmax, the enzymatic reaction does not exceed by any further rise in concentration of substrate. Because why? After reaching Vmax, the enzyme becomes saturated. Even if you are more substrate, no use. Enzyme molecule are fewer than substrate molecule, correct? After saturation of those enzyme molecule, there are no free enzyme molecule to bind with the additional substrate molecule, correct? So both A and B is the right answer. Here, after saturation of those enzyme molecule, enzyme get changed in its form. That's not at all happen. Enzyme do not change its shape. When binding chemical shut off, enzyme activity, the process is called what? It is nothing but inhibition process. Inhibition. And the chemical is called inhibitor. Correct? Here cofactor is given. Here inhibition exhibitor given. None of this given. Therefore, all these are wrong. Next one. Enzymes are divided into how many classes? Silly question. 6 classes. Enzyme catalyzing a transfer of a group that is hydrogen between a pair of substrate S and S dash. See, here quite confusion. I put this question purposely because sometimes what happens, the questions will be framed in such a way that the two options could be the right answer. In such things, we need to give a best answer. See, here what they are telling. Transfer of group. whenever it comes to transfer of group, what we need to write? Transferase. Correct? I have something, I am going to transfer. But NCERT says, transferase do not transfer hydrogen. Apart from hydrogen, any group you have, no, you can transfer to other substrate. Then that is the function of transferase. If you are transferring hydrogen, then you are an oxidoreductase. But here this question is telling that is hydrogen between pair of substrate. So here the confusion lies. Then in such cases, here the transfer of group is given, no, just tick on to transferase. But usually such questions do not come. Suppose if it comes, then you can go for transferase because it is transferring. Whatever it is transferring. Okay, whatever it would be. But hydrogen is actually making you to get puzzle in the exam. Okay. Suppose instead of that is, if the question becomes apart hydrogen is if there is no hydrogen, then you can go for an option oxidoreductase. Usually such question do not comes, but even if comes also, you can go to the best option. Okay. Next one. NAD and NADP contain, of course, vitamin niacin. Oh. Which of the following are correct? Heme is the prosthetic group. Correct? Heme is apoenzyme. How come? Wrong. Heme is not a part of active site. Wrong. Heme is a part of active site. Heme catalyzes the formation of hydrogen peroxide from water and oxygen. Wrong. Actually, heme catalyzes the breakdown of hydrogen peroxide into water and oxygen. This is right. Heme is a part of active site of peroxidase. Correct. That means 1, 4, 6. Correct. 156. 156. No. 156. This is the right option. Here 2 is given. Next question. Okay. Done. So now we have end of the chapter. We have finished the chapter. Now only a small portion left in NCERT. That is nothing but about the primary and secondary metabolites. That I am going to teach you. Very simple. What are metabolites? Something which is undergoing metabolic reaction. They are called metabolites. You have carbohydrate in your body, it will undergo metabolism. You have lipid in your body, it will undergo metabolism. You have protein in your body, it will also undergo metabolism. That means the chemicals which are undergoing metabolic reaction in our body, they are all metabolites. Now, if these metabolites, they are doing a proper function, so that I can identify the proper role of the metabolites. I know exactly, oh, are you going this condition? Are you doing this work in your body? Then I can consider such metabolite as primary metabolites. I will give example. You will understand. Wait for a second. See, example I will write here only. You know amino acid. You know about carbohydrates. We know about lipids. We know about nucleic acid. See, we know the function of all these things. Nucleic acid must be there to carry genetic information. Protein must be there to carry the function like enzyme, hormones, all those things. Amino acid must be there to join to become a protein. Correct? Carbohydrate must be there to provide an energy source. That means I know the function. I in the sense, we know the proper function of these metabolites in our body. What they are doing, why they are present. Then such metabolites are called primary metabolites. But in some organism cases, for example, if I take a plant cell, if I take a bacterial cell, if I take a fungal cell, there are so many compounds are present. For example, in plant, there are certain pigments. Okay, I will take few more familiar example. You might have seen the rubber we are going to extract from the plant. Gum we are going to extract from the plant. Certain medicines we are going to extract from the plant, right? Like that, there are so many compounds that will be available from the plants, from the bacterial cell or from fungal cell. But if I just look at those metabolites in the body of that particular plant cell only, the plant cell says, I don't know what these metals are, what these compounds are doing in my body. Are you getting my point? Imagine this is a plant cell. So let's assume in this plant cell, some gum are forming. This is the gum. This is the gum. Some resins are forming. Some toxins are forming. Now I will ask a question to this plant cell. What are the role of these compounds which you are producing in your body? Then plant says, I don't know. Simply they are producing from my body. I will consider them as a waste. Then we human being, what we are doing? We are not wasting anything. So we are extracted this gum. We started using it for our economical purpose. We started extracting this toxin. We started introducing into some other material. Okay. So like that, we started extracting those material which are actually the plant cell, bacterial cell, fungal cell, which are so producing without knowing their function. We consider them as what? Secondary metabolites. So, in general, I can write here proper identifiable function. Proper identifiable function not known in the cell from where they are producing. But if I ask the same question to human being, we say, yes, gum is very important. We can we can use it like a glue to bind the material. We know the function. But plant, they are producing it, no? They do not know the role, why they are producing it. So like that, there are so many pigments which will be released as a waste from their body. Okay. So such things we call them as secondary metabolites. That's it. Okay. Now let's read the NCERT part of this also. And let me tell you what are all the important line which comes from this paragraph in your exam. Okay, especially we are going to get the question from a table. From this paragraph, I will tell you that also. Primary and secondary metabolites, the most exciting aspect of chemistry that deals with isolating thousands of compounds, small and big, from living organisms. Okay, determining their structure and possible synthesizing them. Okay, done. So no question comes from here. If one were to make a list of biomolecules, all which are having organic molecule, they are nothing but biomolecule only. Such a list would have thousands of organic compounds containing amino acid, sugar, etc. The same list what I had made. For reasons that are given in 9.2.10 table that I will show you shortly, we can call these biomolecules as metabolites. In animal tissues, one noticed the presence of all such category of compounds shown in 9.1. 9.1 already we have learnt. You can open the textbook and you can see. These are called primary metabolites. That means these proteins, nucleic acid, lipid, all those things only. However, when one analyzes the plant, fungal, microbial cells, one would see thousands of compounds other than these primary metabolites. So apart from these, there are thousands of compounds they are going to produce. For example, alkaloids, flavonoids, rubber, essential oils, antibiotics. Now we know the use of these antibiotics. We know the importance of these metabolites. But those organisms do not know. Okay. Colored pigments, scents, gums, spices. These are called secondary metabolites. Okay. Here the definition part has mentioned. While primary metabolites have identifiable functions and play a known role in normal physiological process. Okay. We do not at the moment understand the role or functions of all the secondary metabolites. That means no identifiable role in the host organism. Host means who are producing that. However, many of them are useful to human welfare. For example, rubber, drugs, spices, scents and pigments. We are using that. Some secondary metabolites have ecological importance also. For example, we can use gum, we can use spices, we can use a scent, we can use pigments for our purpose. Okay, that is to earn money. And that is economic purpose and even for ecological importance. In the later chapters and years, you will learn more about this. Okay, let it live. Here few things are given in the table. It's very important. For example, see, in exam, by giving carotenoids, anthocyanin, they may give the option like these compounds or see these secondary metabolites belongs to option A, pigments, option B, alkaloids, option C, terpenoids, option D, essential oils, like that. You should know carotenoids, anthocyanin are pigment. It is easy, you can remember. And alkaloids, morphine, codeine are alkaloids. Terpenoids, monoterpenes, diterpenes. You can remember because at the end, it has been end with the terpenes only. Essential oils, lemon grass oil. This also you can remember. Toxin, it is important. Abrin and ricin are the toxin. Okay, here toxin is a small word. End with ion. Toxin, it's somewhat pronounced like ion in. Abrin, in. Resin, in. It is also toxin. Okay, next lectin. Concanevalin. Concanevalin, A. This is also important. Drugs, vinblastine, curcumin, etc. So vinblastine that will be extracted from Vinca rosea. The plant usually are going to use for identifying the pollen grains in your laboratory, So that Vinca rosea, that will be grown in the roadside. So we are going to extract a chemical, vinblastine and vincristin, that is used to treat the cancer. That will burst a tumor in cancer patient. Then curcumin. These are the drugs. And polymeric substances are rubber, gum and cellulose. Remember this table, it is very important. Okay, that's it. Okay, the end. Hope you have understood all the concept of this chapter. All concepts are crystal clear. Even after reading NCERT, even after watching the video, if you get any doubt, please do mention it in the comment box. I will try to answer as soon as possible. Okay? Thanks for watching.